Good morning, everyone. Welcome to the session on drug discovery and translational research for parasitic diseases. We are honored to have with us chairpersons Professor Shamal Roy, who is a renowned expert in visceral leishmaniasis, currently JC Bose National Fellow at IACS Kolkata, and Dr. Krishna Pandey, Director of ICMR Rajendra Memorial Research Institute of Medical Sciences, Patna, who has worked in the field of infectious disease, particularly visceral leishmaniasis and other neglected tropical diseases. I now kindly request both chairs to be seated on the dais so that we may begin the proceedings.
Details are not. Acha, it's just written over there. That will just read out.
So good morning, everybody. So welcome to the session 6 on drug discovery and translational research for parasitic diseases. And the first speaker of this session, Dr. Jeremy Barus from Geneva, MWV Geneva, and he will be talking about drug discovery in malaria and as an antimalarials. And he has authored about 50 plus publications and holds more than 35 patents. With this brief introduction, I invite first speaker of the session, Dr. Jeremy Barus.
Good morning. Is that better? Can you? Oh. Okay. Thank you very much. Can you hear me? Is that good? Thank you very much for the introduction. It's a real pleasure to be back at CDRI in Lucknow with you all. Thank you also very much for being flexible in terms of my speaking slot on the agenda. So I work for Medicines for Malaria Venture, MMV. MMV is a Swiss not-for-profit foundation with the mission to discover, develop, and deliver new anti-malarial medicines. And so we're a product development partnership, and we have products. So with our partners, we have delivered to registration and to patients 18 new medicines, which have had an impact. Over 2 billion courses of treatment and prevention. And I'm a discovery guy, so I want to also advertise the fact that with our partners, we've delivered over 35 candidate drugs over the last 17 years. Now, MMV is a virtual organization, so everything we do is in partnership. So I want to start really just with a huge thank you right at the beginning of the talk, just to say, huge gratitude to all of our partners around the world who are contributing to our mission. Okay, so we have every period of time we actually establish a new business strategy cycle, and we have defined our strategy until 2030 according to these 4 segments of this sort of semicircle. The first segment is the strategy focused on delivering new treatments, in particular a focus on single dose cure. The second segment is actually for preventing infections, and so these are medicines that actually are given to asymptomatics to protect them from getting malaria. Segment 3 Because we believe in the elimination of malaria, we have a strategy to come up with compounds that actually break the life cycle and hence block the transmission. And then pillar 4 is a section called Beyond Malaria, and I'll be alluding to this with some of our tools that move beyond malaria at the end of the talk. Okay, so where are we with case management, with treatment for malaria at the moment? So the standard of care are artemisinin combination therapies, ACTs. These are fixed dose combinations, and there are a number of them actually out there. But you can see, based on the numbers on the slide, that they're dominated by one in particular, which is artemether lumefantrine, Coartem. MMV with Novartis came up with a pediatric dispersible formulation of Coartem back in 2009, I think it was. And since then, over 500 million treatments have been given, which, if you do the maths, equates to one child's life transformed every second since that drug was launched. The numbers are really quite staggering. So we have good treatments, and we've overcome many of the challenges shown in the box on the right. Indeed, you know, Coartem now is approved in first trimester pregnancy, and we've even got an approved product for young infants under 5 kilos. But there remains this question of resistance. What is our strategy in the context of managing resistance, given that resistance to artemisinin and some of the partner drugs is emerging? So we have a strategy which is really focused over different time periods. What do we do in the short term, and what are we going to be doing ultimately in the long term? So in the short term, we have multiple first-line treatments. So I mentioned there are a range of products which have different partner drugs and therefore actually have different abilities to handle resistant parasites. We're also working with Moru and Fosun Marabini to deliver a triple ACT. So this is an ACT which actually has 3 components within it. We also have been working to deliver a product for an actual drug product for single low-dose primaquine. And the idea here is that this is added to an ACT such that it kills the gametocytes, including any resistant gametocytes, and therefore blocks transmission of resistance. And indeed, that this has just recently been announced an approval with the WHO. And we have ganaplacide lumefantrine, which is a combination that has just reported positive results at ASTMH from Novartis. And this drug combination is looking very exciting, and the data supports progress to actually file for registration. And so this is a drug combination that we anticipate at the moment ultimately being the first non-artemisinin combination. So what do we do in the next in the medium term? The short term is all about managing artemisinin resistance. In the medium term, it's really about managing lumefantrine resistance, which is the partner drug within Coartem. First of all, we have a triple combination. So GAN, LUM, CYP, and again at ASTMH, Novartis reported very encouraging phase 2 data for this as a single dose combination. And those data actually support moving to the next phase of clinical development. And then there are a range of very, very interesting combinations which do not involve either artemisinin nor lumefantrine from our partners, carbamaquine, pyronaridine from Merck, the Zydus compound aferoquine from Zydus, and then Novartis are exploring cipargamin for severe malaria. In the longer term, our ambition is to deliver a new single-dose cure, and there are a range of compounds in the pool there which, when combined, have potential for that. Okay, what about protection? So, first of all, the standard of care is with sulfadoxine pyrimethamine amidaquine, or SPAC, and this is given to children monthly to protect them from getting malaria. And it's actually it's resulted in huge use in the key areas of Africa that are relevant, with 75% protection and up to 50 million children being covered by this seasonal malaria chemoprevention. Of note, there are 2 new vaccines which are actually starting to be rolled out, very, very exciting, and which have shown additivity with the drugs. But for the future, our goal is to deliver chemopreventative medicines which have once monthly cover and overcome all the resistance risks associated with SPAC. And within the chemoprevention area, one of the new technologies, new ideas being explored is the notion of a injectable into the muscle that then gives very, very long-term protection over 3 months, ideally even 6 months, and therefore actually can be dosed once and then give protection over an entire season. And MMV has taken 2 compounds. One is a prodrug of atovaquone. The other compound is a compound called ELQ300, MMV055, and clinical studies are beginning with these. Okay, so I'm a discovery guy, as I mentioned. When we think about doing drug discovery, the first thing we think about is actually what is the end game? What are we wanting to deliver? And these are the target product profiles. As I've mentioned, we really think in the context of treatment and prevention. And so we have defined 2 target product profiles, which have a range of associated target candidate profiles with criteria that help scientists understand whether the molecules that they're working on in discovery actually meet the criteria necessary to have impact ultimately in patients. We've built assays with our partners that recapitulate the biology of all the druggable points within the malaria life cycle. And every time we have a compound of interest, we test them in these assays, and it informs us whether or not a compound is active on blood stages or liver stages or gametocyte stages, whether it's prone to resistance generation, whether it kills quickly or slowly. And this gives us insights as to what target candidate profile and what target product profile that compound can be useful in. We've also established clinical protocols, and I'll be showing you some data in our volunteer infection challenge model for asexual blood stage malaria in a moment. So we're constantly updating and making our criteria more stringent because of really what's at stake. And essentially where we are now is that we're looking for single dose cure medicines where with 100 milligrams that will actually clear all the parasites out of an individual. And for chemoprevention, we're looking for a candidate drug with 100 milligrams oral dose will protect for 1 month, huge pharmacokinetic challenge, or indeed chemopreventative medicines which when injected into the muscle provide a long-term protection. And all of these are going to be sort of, well, a number of these are going to be illustrated in some of the case studies I'm going to show you in a moment. Okay, just to set the scene, some of the tools that we have to help us, given the fact that these pharmacokinetic challenges are so difficult, we've established a tool called MMV Solar, which with standard preclinical in vitro and in vivo mouse rat dog data, we can get a human prediction of the pharmacokinetics, of the volume and the clearance. And on the left, this just shows data that we have from both in vitro and in vivo, standard in vitro growth inhibition data and in vivo data in a SCID mouse how well those data, when modeled and transformed, correlate with modeled efficacious data in humans. And the good news on the left, you can see that there's, broadly speaking, a correlation. This gives us a lot of confidence in our discovery phases and assays that when we achieve certain criteria, this will actually mean something in humans. I mentioned about our target product profiles getting more and more stringent over time. And just to emphasize the impact that that has had, this is a slide showing time on the x-axis and the predicted oral single dose on the y-axis. So everything has been normalized to the same scale. To begin with, in the early days, we didn't even have a TPP. Then we introduced a TPP, but it was a 3-day TPP. So you can see that there are a number of compounds with very, very high doses. And this is because they were fine for 3-day, but not for single. And then over time, we then applied a more stringent criteria. We wanted less than a gram or less than 500 milligrams or less than 100 milligrams. And our partners screamed about how difficult this was, but they delivered. And you can see actually in recent years, we've had a number of very, very interesting compounds being delivered, which have predicted doses under 100 milligrams. Okay. I'm going to start with my first case study, and this is a case study of a drug discovery project run with Sanofi. Thanks to Laurent Fraisse, Alain Pellet, Didier Laura, and Sir Simon Campbell, who were involved in this. So Sanofi had an interesting idea. They wanted to pick compounds from their library for screening based on an orthology approach. And what this means is that they looked in their library of compounds, and they selected compounds which actually were targeting biochemical targets in humans which had some link with Plasmodium. And they built up a library and they screened it, and they actually got a very high hit rate, which was interesting. And now one of the hits was this compound here, shown on the left. Very small molecule, a very potent molecule. When you're doing early drug discovery like this, the first thing you always need to do is actually do some experiments to make sure that this is actually what it says it is. And the first problem that they faced was that they resynthesized the compound and it was inactive. They then tested analogs and those were inactive, but they went back to the original sample and it was 12 nanomolar. They did an LCMS. They noticed that this compound was 99% pure, but there was 1% of something else. So they prepped that 1% out, and it was this compound here. Now this, for the chemists in the audience, the chemists will recognize that this is effectively an azodi. Oh, there we go. An azodi linked version of this particular compound, really coming out of, originating from the chemistry that was used to make it. And this compound is 0.15 nanomolar. In other words, 1% of this. So astonishing bit of detective work to pick out the active component. From a medicinal chemistry perspective, this has all sorts of problems. Not least this azodi. So one of the first things that they did was they looked at various linkers, and actually an acetylene linker gives a very nice compound. But again, this is still non-ideal. So what they did is they profiled this compound to understand what its liabilities are. It's very, very potent, but its solubility is very poor. Its permeability is poor. And its hERG was sort of okay, but could be better. So they had a strategy to improve the permeability. This molecule has two acyl guanidines, so they removed one of these to reduce the number of hydrogen bond donors and acceptors. The permeability went up. Solubility was still poor. It was still potent. And they found that actually they could substitute this amide here with this 2-pyridyl component, giving a very, very, very nice balanced compound, but with slightly, with slightly inferior solubility to what they wanted. In terms of solubility, what they noticed was that the melting point was very, very high. So they had a strategy to desymmetrize the molecule. And in doing that, the solubility improved. But in this particular case, the permeability was still poor. So basically taking the learnings from this and the learnings from this together delivered MMV533, which is the candidate drug. So this compound was taken into SCID mice. So these are immunocompromised mice engrafted with red blood cells and Plasmodium falciparum. And an extensive study done to look at both the effect of the compound on the parasitemia over time against the compound concentrations. All of that information could then be modeled and actually to extract out what would be the predicted human efficacious dose in conjunction with the human pharmacokinetic prediction. And so basically a human half-life was predicted to be round about 100 hours, very long human half-life, and a human dose of around 200 milligrams. What actually happened? Okay, so the compound was taken into phase 1. So these are healthy volunteers. First of all, in the single ascending dose, very nice dose-dependent pharmacokinetics with a lovely, beautiful human half-life of exactly as predicted, around about 100 hours. And no obvious food effect in the fed-fasted study. And then they actually ran a volunteer infection study. So this is where you take volunteers, you infect them with Plasmodium falciparum, you see the actual parasitemia increase. When it gets to a certain point, you give the new drug, and then you obviously expect for the parasitemia to come down in a dose-dependent manner. And indeed, that's what you see. Three doses were given. And basically in 20 mgs is not so great, but actually at 100 mgs this is completely suppressing the parasitemia, and this basically all aligned with what had been anticipated, a very, very interesting profile, potential indeed for inclusion in a single dose cure. And really the next step is actually to combine this with other compounds in phase 2. Okay, my second case study, this is from Kip Guy, University of St. Jude, then Kentucky, and now he's in Minnesota. He had delivered with MMV a compound called SJ733, a Plasmodium falciparum ATP4 inhibitor. And this was taken into human studies. And from our perspective, unfortunately, it didn't have a very long human half-life. It was a reasonably potent compound, but actually in human studies, its half-life was about 20 hours. And as I showed you a moment ago, what we were really seeking is compounds with half-lives more like 100 hours in humans. And what happens in humans with this particular compound is you add it to, in fact, it even happens in human liver microsomes, is that this pyridine N oxide is oxidized to an N oxide. And unfortunately, that N oxide is inactive. So it's a major metabolite that is inactive. And so basically, we decided that we were going to explore whether we could come up with a backup to this compound that specifically addressed this N oxide issue, because everything else about this compound looks really, really great. It works very well in both Plasmodium falciparum and in Vivax, in VIS and phase 2. It's just it has a high dose, and as in its current form, it needs co-formulation with a CYP inhibitor to move forward because of its low half-life. So the strategy that the team adopted, this is the structure of SJ733, is they looked at different regions of this molecule and basically came to the conclusion fairly quickly that there were two parts which really needed to be retained. They needed to stay the same. There were a few changes made to this core, but the main focus was this pyridine, particularly since this is the region that is metabolized in human and leads to inactivity. And really the strategy was to put a substituent in the 2 position of this pyridine that either sterically or electronically would reduce that oxidation. Okay, so what actually happened? So it was very nice to see that all of these groups that were made for at the 2 position of this pyridine, all of them were potent. And many others were made, various different potencies, various different levels of intrinsic clearance as well. And obviously these ones down here are starting to look very, very interesting. Now, one point I just want to make before I show you ultimately what happened is to point out that much of the discovery that we do is in a bound world. And I guess what I mean by that is that when we run our malaria growth inhibition assays, we are not just measuring potency. We're also measuring binding to the proteins in the media. And just to sort of illustrate that, here are 2 compounds from this program, which are identical apart from 2 motifs. This is a 4-fluorophenol, and here's ethanol. Ethanol and 4-fluorophenol, very, very different chemically, very different lipophilicity, and so on. On the face of it, though, you would say these are within biological error. They're 3-fold difference in terms of the EC50 in a growth inhibition assay. However, if you measure the binding of these 2 compounds to the protein in the media and you correct, you actually find out that this is sub-nanomolar, this compound on the left, and this compound is a little bit more potent because it's so much less free. So it's only when you look at the unbound potency that you really get a sense of the true structure-activity relationship. And we have started to think about this in the light of everything that we do, not just potency, but also intrinsic clearance, volume of distribution. So here I've actually put in the unbound EC50, and you can see that the rank order is not the same between, if you look at the compounds, the rank order is not the same once you take unbound concentration into consideration. And basically, once you factor in unbound volume and unbound intrinsic clearance and the ratio of those, which is a surrogate for half-life, there are 2 compounds that really, really stand out. And in fact, it was this compound, MMV609, with a cyclopropyl ether, spectacular pharmacokinetics in animals. So 26-hour half-life in the mouse, 23 in rat, 41 in dog. It's predicted to have a half-life way over 100 hours in human, very, very low human dose prediction, very, very potent compound. Okay, I'm coming to the end of my talk, but I want to just say a few words first about MMV Open. So MMV over the years has been making boxes, physical boxes of compounds available to the community for free. Just to say that there are still some boxes, most of these are actually exhausted. Not sure what's happened to my pointer, but the Global Health Priority Box, which in collaboration with IVCC is still available if anyone wants that. And MMV we've been doing a lot of work in the machine learning, the AI space, and I wanted to just highlight a few points there. One is that we've built a machine learning model which is available on the Kemble website for free. It's built on over 5 million compounds and data from Novartis, AstraZeneca, GSK, and so on, who modeled their data with us. And this allows anyone to put a virtual structure into the website. You then get a score, and then that score tells you the likelihood of that compound being active on Plasmodium falciparum. And we actually use this to help design the compounds that we will acquire for our libraries, because we've shown that it gives a 10-fold increase in hit rate. Using the whole technology of pattern recognition, the sort of thing that you would have on your phone to unlock the phone, we've actually got funding from GHIT to do a cell painting imaging project, really where the goal is to create open access models which recognize the parasite pattern linked to a specific compound mode of action. And so this is going to be delivered at the middle of 2027. But the aim is that there will be algorithms for different pathways, different modes of action, that whenever we've done a high throughput screen or got phenotypic actives, we can image according to the protocol, basically insert it into the tool. We instantly find out, is this something new or different? And if it's different, that could then be prioritized for follow-up. And then we put a lot of work into actually delivering a medicinal chemistry design tool. And this is a tool that's actually freely available for anyone working in global health. Doesn't just have to be malaria, it could be LF or whatever. It's a gated access, so you have to basically contact me, and we can then arrange for you to have training and so on, up to a certain number of licenses. But effectively, what this tool does is it allows users to put their data into the tool. It models that data, and you get information on the quality of that model. How does my chemical structure predict the potency on the parasite, for instance? Then it has generative design, where it creates virtual chemical structures around your front runner, and it scores those virtual chemical structures against the model. And then you can triage it based on your particular design strategy, on your TPP. And ultimately, it then picks 20, 30 compounds for synthesis based on compounds that are predicted to be good against your TPP, but also that will improve your models in the next design strategy. In other words, there would be a learning loop, there would be active learning. And this is going to be delivered at the end of March next year. And then we're actually in discussions with the Gates Foundation to deliver the next version of this, which will introduce also co-folding that will enable people to work with biochemical targets and sequence to structures, basically. Okay, so this is the end of my talk. I'm extremely grateful once again for your attention, and thank you very much.
Thank you, Dr. Burrows, for your wonderful journey for excavating antimalarial drugs. So now the floor is open for a few pressing questions.
Pushkar.
Do you know the target for the MMP first drug you mentioned?
No, we don't. Great question. We don't. We couldn't select resistance, which is obviously great news. But the corollary is that we don't actually know the mechanism of action. There are several very slight markers which David Philip found, but that it's the only things that we have at the moment are very vague hypotheses.
One quick question. So it's been a few years since the two studies from Kip Guy and another one from GSK came on this screening of compounds to back-to-back paper in Nature, and AI has developed significantly. So has there been a revisit on prediction of some of the targets using the new AI algorithms for the compounds for both the GSK as well as this thing?
So there's been a lot of work on looking at the overall data, as I showed, but I don't know that the newest algorithms to do the chemoinformatics to extract targets has been done.
Thank you.
A great talk. My question is around on the same line. Is the in vitro resistance selection is also integrated with the selection of the compounds? In vitro selection for the resistance, is it also integrated with the selection of the compounds, and are they comparable to artemisinin in that?
So, with every lead compound, we've got a sort of like a standard protocol that we apply to look at in vitro selection and of de novo resistance. What I would say is that once upon a time our mantra was that the frequency of resistance was likely connected with the target. In fact, what we've seen in some very, very nice examples recently is is that it is possible to find compounds on a particular target with very, very low risk of resistance, in fact. And so there are a whole myriad of factors that ultimately, affect the resistance frequency. And our goal, what we prefer, are compounds which we call irresistible, where effectively we cannot select resistance, similar to what was observed originally with artemisinin.
Hi, it was a nice to talk. I actually didn't get the relevance of actually looking for the unbound EC50 for these drugs, because at the end all these drugs have to work in the blood where you have these proteins present, right? Why do you actually look at this?
So, it's the unbound concentration in plasma, which if we assume a fully reversible diffusion of compound, will ultimately lead to efficacy. And for instance, the plot that I showed where there was a very, very nice correlation, what I didn't say was that the theory underpinning that correlation was that the unbound concentration in vitro equals the unbound concentration in vivo equals the unbound concentration in humans. And in fact, the fact that there is a correlation demonstrates that is the case with the compound selected.
Jeremy, great to see how much work has happened through MMV, and I think it's an example for many other diseases perhaps to follow. We have similar challenges for novel antibiotics and for drugs for other neglected diseases. So it's really hard, very, very amazing to see how well the pipeline for malaria has built up. So congratulations for being a big part of that. I wanted to ask you a question more generally. Obviously in this conference, we have a lot of people working on basic biology, trying to identify new targets, new mechanisms, and so on. And somewhere all of this also has to feed into the pipeline. That's what new target discovery and validation is about. And yet, a lot of the molecules that are in the pipeline, or at least what I gathered, you are coming from a phenotypic approach and then deconvoluting the target subsequently. And sometimes you can't, right? Ganoplacide, you don't know what the actual target is. So where do you see a place for both approaches to merge? How do you see target-based drug discovery tools and so on, which are evolving in the field of drug discovery in general, merging with malaria drug discovery efforts? Is there a happy mean where the biology can also play a big role in guiding compounds?
It's a great question, and in short, absolutely. So I think a lot of the early progress has been made with phenotypic screening, as you say, and until recently, actually, the biology, our knowledge of good targets, and then actually having expressed protein and conditional knockdown data and so on that really confirms attractiveness. This is all very, very new, and I believe that target-based discovery for malaria is going to be a key component of the future.
Thank you.
Hi, Jeremy. For this AI-based modeling, phenotype-based modeling, what is the input data, or would you ask scientific community to provide the data, like different knockout studies and all that?
So, in terms of the medicinal chemistry design tool, which I described, the input data is the data that the scientist using the tool brings into the tool, and that typically it can be biochemical data, but it can also be in vitro growth inhibition data.
And for the phenotype model, for the parasite,
for the overall global phenotype model, I guess that model isn't attempting to make any to separate out compounds having different modes of action. It's just simply saying, is a compound likely to show activity on malaria. It has no information at all on the biology associated with that. So this, I think, really fits with Pushkar's first question. Actually, I think there's plenty of things that could be done to actually, look at the biology associated with these compounds.
Quick question.
Jeremy, always a pleasure listening to you. Just a couple of thoughts. I mean, if we know the target, can we predict the resistance? One, if we know the targets, if we have a short listed of candidates. And then secondly, the unbound concentration, can we apply that to the other therapeutic areas as well? It will be true for other indications. And in the clinical model where you inject the volunteers, how different will it be in the clinical setting? Because it's a control, you know how when you have inoculated and you are giving the dose. But in a true clinical setting, how different it would be?
Okay, I've got to remember those three questions. So can you predict resistance based on the target?
Yeah.
No, you can't. And I can talk to you over coffee to expand on that. What was the sorry, my brain is fried.
Unbound concentration,
Oh, yes, absolutely. I mean, the reason that we came to look at malaria in this way is because this is how the whole pharma industry look at, human targets and human disease. So it's not new. It's just that no one had really looked at it in the context of malaria. So as long as you have drugs which are passively, reversibly distributed, absolutely, it doesn't matter what the pathogen is. And then the final point was
a clinical setting.
So I guess the data that we have is that when you model the volunteer infection study data and you model the phase 2 data in patients, actually the Emax and the in vivo EC50, they're comparable. Okay. So it's actually good enough translation.
Thank you.
Okay. So the last pressing question from my co-chair.
Great talk. Which, according to you, should be the best drug for pregnancy, especially in the first trimester?
So, I mean, the only drug, well, it's not the only drug because there are others, but Coartem is approved for use in first trimester for treatment. And I guess what I'd say is it took a long time, decades for observational data in pregnancy registers for the data to be there to give the regulators the confidence for that.
And one last point, ICMR has made a drug and it's now freely available in our country, Piperacin plus Aterolane. Do you have any experience with that?
I mean, I know Aterolane very well because it
Yes.
came out of an early MMV program. So this is, I mean, it's essentially a synthetic peroxide with piperacin, and I think the critical questions will be what is its efficacy in the real target population in kids, and how does the drug actually perform in relation to artemisinin resistance.
Thank you. Thank you.
Thank you.
Okay. Thank you, Dr. Burrows, for your provocating talk. Thank you very much.
Thank you.
Thank you. Thank you, sir, for such an insightful and enriching presentation. May I now request the chair, Professor Shamal Roy, to kindly felicitate our speaker as a token of appreciation.
Is Dr. Saman Habib. She is the chief scientist at CSIR-CDRI, an eminent molecular parasitologist. Her
pioneering research on malaria parasite biology, population genetics of malaria susceptibility, and organelle targeted nucleases for Plasmodium genome maintenance, and has advanced novel therapeutic strategies. So she'll be talking on malaria.
Good morning. Am I audible? So there will be a shift of gears a little bit as we move from Jeremy's extremely exciting talk to, make us understand what's happening in the drug discovery development field. I move away to take you to the parasite and to its biology, and I'll talk today on some relatively recent work on organelle-targeted nucleases for genome maintenance of organelle genomes in the parasite. Now, for students here who don't work on malaria, just to let you know that the malaria parasite has two very important organelles, the mitochondrion, of course. But the interesting thing is that it has a single mitochondrion in most of the stages, except the gametocyte, where it has about 4 to 5 mitochondria, or sometimes even more. There's a single epicoplast, which is a plastid-like organelle, and evolutionarily, it's been a very exciting discovery made about 20 years ago, and which told us that there was this organelle that came probably from red alga and early cyanobacteria. And so it's a secondary endosymbiont. It has 4 membranes surrounding it. And it also offered, sort of made people think that if you could design drugs against proteins that target to the organelle, then you would be actually having good targets for malaria. Anyway, what I'll talk about today is that the genomes of the epicoplast and the mitochondria are necessary for maintaining integrity of the coding sequence during replication and under exposure to genotoxins. An alteration of DNA repair proteins, including those that target to organelles, have implications in generation of resistance to antimalarials. And there was this paper that came out in 2016 that showed that the falciparum mutator phenotype, which is amplified resistance to multiple drug strains that fail to induce DNA repair upon MMS damage, carry mutations in a lot of DNA repair genes. And some of these genes actually target to the mitochondrion or the epicoplast. They were putatively targeted. They were not established to be targeted there. And there has been an observation that one mutation in an exonuclease, and I'll talk about this exonuclease later, is linked to high IC50 values for etovaquone and proguanil. So clearly there is interest in exonucleases and DNA repair proteins, and there was absolutely no understanding of how DNA repair works in organelles some years ago. There is now understand a little bit, we understand a little bit now. Now, as I said, there is this mitochondrial genome, which is the smallest mitochondrial genome known in nature. It's only 6 kb in length. It codes for a fragmented ribosomal RNA population and for 3 exons that are important for the electron transport chain in the mitochondria. And it's a repeated genome, so there are 6 kb tandem repeat copies. In Plasmodium, it's a concatemer and probably replicates. It's been shown by an early paper on replication that it replicates by the rolling circle mode. The epicoplast, as I mentioned, has a circular genome of 35 kb. It has 4 membranes surrounding it, and most of the proteins required for replication in both these organelles are nuclear encoded and organelle targeted. Now, we started looking at organelle repair, and we realized that there is a group of proteins that are important for base excision repair. And if you look at the conventional base excision repair pathway, the first step of base excision repair is when you have a damaged base, which can be an oxidized base that's removed by DNA glycosylase, and then an apurinic or an apyrimidic site is then cleaved by an endonuclease, and then this would proceed either by the long patch pathway or the short patch pathway, and in the long patch pathway you would also need a Fen endonuclease to remove the flap. Now, to cut a long story short, we did analysis. So we analyzed what would be potential epicoplast or mitochondria targeted proteins. And we shortlisted a group of them, and then we characterized them and localized them. And our results showed that both the AP endonucleases, which are critical for the Burr pathway, go to the mitochondria. So what you see here in red are mitochondria targeted proteins. What you see here in green are epicoplast targeted proteins. And we realized that most of the important base excision repair proteins go to the mitochondria, and we established their activities. We showed how the pathway functions, and we now believe that the base excision repair pathway is a major pathway in the mitochondrion and is not active in the apicoblast. And there is reason to believe this because we don't find any AP endonuclease going to the apicoblast. These are the only two AP endonucleases in the parasite. What was interesting was that we found an exonuclease or a FEN. This is both an exo and a FEN, which was very, very different from known flap endonucleases. And I'll describe this protein now. So what this protein is, we call it PF-exo or PF-exo-FEN. And this is a protein that I show here. It's a very, very large insertion. It's got limited identity to the Mycobacterium smegmatis FEN-A. And if you model it on FEN-A, you find that it has 3 metal binding sites. It lacks a 4th metal binding site. And this insertion, this large, more than 150 amino acid insertion, is obviously not modeled, cannot be modeled. It's quite disordered. Early reports have suggested that it's essential, might be essential for falciparum, and the knockout has shown slow growth in the blood stages in P. burghae. Satish, in collaboration with Satish, who's done a conditional knockout in burghae, it seems to suggest that it is essential for parasite transition from the liver to the blood stage. So you don't see a transition happening. If you look at the distribution of this, we find that this insertion is found only in the parasites that belong to the Labarenia subgenus of Plasmodium and are not found in other parasites. It localizes to the apicoblast, so it's very typically an apicoblast targeted protein. We made the recombinant protein, generated antibodies against it, and then by IFA shows where it goes. And when you start looking at activities, this is where the interesting part. It is a 5-to-3 exonuclease. It works on all these targets, so it can work on these target templates. These are FRAM labeled templates. It can cleave DNA in the 5-to-3 polarity. It can cleave rhesus DNA. It can cleave blunt end DNA. It can cleave nick DNA. And it can also cleave single-stranded DNA in the 5-to-3 polarity. It is a flap endonuclease. It's a very, very efficient flap endonuclease, and it can cleave this flap that I show you here. So it generates this sort of flap. But when we did this experiment, we realized that it's not only cleaving the flap, but once the flap is cleaved, the flap is further cleaved, and you get this very, very small molecular weight over here, a small stretch, which tells us that this might also be a 3-to-5 exonuclease on single-stranded DNA. And when we checked this on single-stranded DNA, we found that it cleaves the single-stranded DNA also in the opposite polarity, in the 3-to-5 polarity. It also cleaves DNA-RNA hybrids. And putting all this together, this seems to have a very, very expanded substrate range. This distinguishes it from all known exonucleases in nature. So this is like really unique. It has all these activities. It can work in reverse polarity on single-stranded DNA, and therefore is very different from the whole repertoire of exonucleases that we know of. So it is a novel exonuclease, and we published this last year. Now the question here is, how does this function? If it's got a similarity to MS FEN-A, which is a flap endonuclease, and FEN-A does not show this diverse sort of substrate specificity. We did a lot of mutations, and we found that there were these 2 metal binding sites that were very critical for the exonuclease activity, and disruption of any of one of these metal binding sites resulted in complete loss of FEN activity. And both and mutations in M1, M2, and M3 were required to cause reduction in the exonuclease activity. So clearly there were different regions participating in the exonuclease activity and the flap endonuclease activity. Now, the interesting observation, and I showed you in that phylogeny that I had showed earlier, is that the burghae protein does not have this insertion. So we thought, let's check on the differences in activity between the burghae protein and the falciparum protein. We did that. We find differences. The differences are that there is exonuclease, and the burghae protein is actually active on rhesus DNA, but it's very, very poor activity on blunt end DNA. It is a flap endonuclease, but the terminal product generated by the flap endonuclease, the size is very different. It is also very poorly active on a 3-to-5 template. And it works on RNA DNA hybrids, but its efficiency in the 3-to-5 polarity is very poor. So there are differences, clear differences between the falciparum and the exoproteins. Then another interesting observation was that when we modeled it, we thought that there was a possibility of a 4FE4S cluster forming because of the position of the cysteine and histidine residues over here. And when we mutated these individual residues, we realized that the 4FE4S cluster was disappearing when a histidine that was present at the end of this insertion was mutated. So clearly the formation of the cluster on this protein, the iron sulfur cluster, was determined by the presence of a histidine residue in the insertion. And since the P. burghae exo did not have this insertion, the P. burghae exo also did not bind FES clusters. So there was very clear differences between the presence and absence of the FES cluster on the burghae and the falciparum protein. Now, so now we have these differences. We also know that there are differences in activities. So what was it that was causing this difference in the observed activities? So we removed the FES cluster by chemical means. That was not the reason for the differential activity. We mutated a D2N, which was different in the two proteins. That also did not result in any difference in activities. However, then we decided to remove this LCR insertion because we thought that perhaps this is what is influencing the differential activities of these two proteins. So does the presence of an LCR determine different activities in these two homologs? So what we did was that this is your entire PFXO protein. This is the insertion. This has a 4FE4S cluster. We made a deletion where we removed this insertion and therefore also removed the clusters. We call it the del-ins. We took the Bergheim protein, we took the falciparum insertion and put it into the Bergheim protein. And when we did that, the cluster came back because the histidine residue was there now. And then we tested activities of all these. And so this is the original one, which is showing you this expanded DNA substrate repertoire. The PBXO, which has much more limited substrates. Then the del-ins, in which you remove the insertion, this starts behaving more like the Bergheim protein, loses several activities. You put the insertion from the falciparum protein into the Bergheim protein, you get all these activities, which were not there in the Bergheim protein earlier. So then PFXO becomes a novel epicoplast exonuclease with a functional LCR that diversifies substrate recognition. This was a very interesting observation for us, but for many of us working in the falciparum area tend to ignore these LCRs, these regions that are present in a lot of p-falciparum proteins, thinking that they don't have any function, that they're just there and they just cause problems for expression because they're so disordered and they are difficult to work with. But clearly they have functional relevance, and I'll show you our analysis further. So, here it is. So now what we did was we had a substrate which was this blunted DNA on which the PFXO was active, and the PFXO del-ins without the insertion was active, but the binding of that enzyme to this substrate was poor. So when we did this analysis, we found that there is so when we compare the DNA binding of PFXO intact and the PFXO with the deletion of the insertion, the KD values are very different. The KD value of the insertion of the del protein is much higher, telling us that the affinities for the DNA substrates change when the insertion is removed. It does not bind DNA so well. And here on substrates where DNA binding, where activity was not seen on the with the del-ins protein, it also did not bind the CNS substrate, and this we showed by NSAS. So removal of the LCR therefore abolished DNA binding to flap and one nucleotide gap substrates, and also reduced the affinity to blunt end DNA, telling us that the LCR had a very critical role in substrate recognition. So this gave us what it was doing. Now, what was the function of this exonuclease? That's our next question. So if this is a FEN and an exo, it can work in DNA repair, but it can also work importantly on DNA replication. And in the malaria parasite, the epicoplast has a DNA polymerase, which is a very, very large protein. It's called PFP-REX. It has a 5' exonuclease, 5 to 3' exonuclease and a polymerase domain, but it also has a helicase and a primase domain, and this gets cleaved, and these then become independent enzymes, and this is a 220 kilodalton, extremely large protein. And then you have this polymerase domain. So we made this recombinant protein, we raised antibodies against it, and then we did pull down experiments with the exo antibodies on the one hand and vice versa with the PFP-REX polymer domain antibodies on the other hand, and we showed that both pulled down each other from the falciparum lysate, indicating that there was interaction between PFP-REX and exopol in vitro, and also when we pull them down from the parasite lysate, we got the interaction. So clearly during replication, this seems to have importance. And there is no other flap endonuclease or RNase H predicted for the epicoplast. So clearly this seems to be the main protein. Now we therefore hypothesize that there are these possible functional roles of PFXO. It's a work on RNA primer removal. This will be like RNase 2-like activity, RNase H activity would remove the RNA primers during replication. It complements the absence of a 5' to 3' proofreading on PFP-REX, so that would be an important thing for repair during replication. This is a 5' flap removal activity, would be important for removal of flaps during DNA replication. Its ssDNA cleavage activity can function to restart stalled replication forks during replication, and then it might also help to resolve R loops like the human RNase H2 does, because there is no other homolog in the epicoplast with all these activities. And prediction also suggests that it might even work on for liquid-liquid phase separation, but this is something that we have to establish, and we haven't done that so far. So the larger question, what does it tell us about LCR function and disordered domain function in plasmodium proteins? There have been many hypotheses. People have suggested that these large asparagine-rich sort of insertions can also provide alternative sites for glycosylation and therefore contribute to a variable antigenic landscape for falciparum. These may also work as tRNA sponges to slow down translation, because if you had a lot of asparagines in an insertion, the tRNA recognizing those codons and with the anticodons recognizing those codons would be limited in quantity, and therefore translation would slow down. And when that happens, you might have better protein folding. So this might aid in that. These are all hypotheses. Some experiment from Ajali Fujia's lab telling us this. These might also be recombination hotspots that contribute to generation of diversity in antigenic loci. And work from our lab and from 2 other labs has shown from other proteins, not this one, is that they also affect catalytic activity, might affect stability, and we have shown that in the ISCS case also affect domain interactions of proteins. And the work on the exo tells us that they also have roles in DNA binding and can expand DNA substrate recognition in the parasite. So clearly they are not useless extensions or insertions. And if you look at the phylogeny again, as I said, these Plasmodium 2 subgenera we are looking at, the Lavarania subgenus and the Plasmodium subgenus, which infects primates. And as you can, and the exo-LCR insertion is found only in parasites of the Lavarania subgenus. And all these insertions are quite different. So the insertion is present, but the sequence is not absolutely conserved. But what is conserved in the insertion is the histidine residue that forms the iron sulfur cluster. So clearly this is very, very important in all these. So, and this LCR renders, as I said, unique functions and expands the DNA substrate repertoire of an apicoplast enzyme. So a very unique and very interesting exonuclease, something that's not known in nature so far, and with an LCR that does this function. I move a little bit away from the apicoplast to come to the mitochondria. So once we had this interesting exonuclease, we asked the question of what were the exonucleases now functioning in the mitochondria during mitochondrial DNA repair and replication. And if you look at the gamma DNA polymerase, people have not worked on it. It's not been sort of purified, and therefore there is no biochemical analysis of the protein, but there's a predicted protein that goes to the mitochondrion. And this gamma-like DNA polymerase does not have an exonuclease domain. So it has a polymerase domain, but you would expect a 3 to 5 exo domain on the polymerase. But this 3 to 5 exo domain, there's a big, huge N-terminal sort of region, but this region does not have any exodomain. It is very, very disordered, so we don't know what that does. So there's only a polymerase domain, there's no exodomain. So we don't know what exonuclease, and the exonuclease would be required for replication and repair. Now, Mrinal is here. Mrinal's lab has shown that in terms of repair, RAD51 and Bloom helicase, these are 2 DHBR proteins, they do go to the mitochondrion as well as to the nucleus in certain stress conditions and at late stages of the parasite. We identified one mut S. So for MMR, we identified a protein that was a mut S that could go to the mitochondria, and we needed to establish the fact that it was going there. So these seemed to be two possible mechanisms of repair in addition to Bur in the mitochondrion, but we only had a mut S. We had no other protein of the MMR pathway putatively going there. So we looked at these 2 exonucleases. These 2 exonucleases are conserved only in certain alveolates, and the Burghi orthologs are essential for survival of the parasite. You look at these structures, very different from each other. Quite a lot of insertions are not known, but then there are some conserved exonuclease domains over here and over here that you can see. Both of them go to the mitochondrion. So as I said, we made the recombinant protein, raised antibodies, did IFAs, and both of them are very, very clearly localized, as you can see here with the mitochondrial marker dye, and do not go to the epicoplast. So you do not see any co-localization with the epicoplast marker PFHU. If you look at their activities, I'm not showing you data, just tabulated it here. PFXO MIT1 only cleaves single-stranded DNA. So it does not cleave. It has all the 3 exonuclease sort of marker domains, but it does not cleave double-stranded DNA. On the other hand, MIT2 cleaves single-stranded DNA, but also has activity on double-stranded DNA in both polarities. So this is a very interesting protein also. So two important proteins conserved only in alveolates. This one is bidirectional exonuclease on single-stranded DNA. This one is bidirectional on double-stranded DNA and cleaves single-stranded DNA only in the 3 to 5 polarity. So two different functions. And with Satish's lab, Pragya, Pratis's student, did this knockout. We made a knockout of MIT1. And this is Pragya's data, in which she showed that if you have a knockout line in Berg hive for MIT1, which is a single stranded DNA exonuclease, and if you inject sporozoids, this is a conditional knockout line because you couldn't make a direct knockout. What happens is that you do not see transition from the liver to blood stages. And in the very, very rare cases where you see something coming up in the blood, so which has the pre-patent period is very long. And actually, these are parasites in which the excision hasn't happened. So basically, there is a complete block of transition from the liver to the blood stages. Similarly, they took merosomes. The number of merosomes comes down a little bit. And when you check infectivity of merosomes in mice, you also find that there is no conversion from the liver to the blood stages, like 0 here out of 5. And in the rare cases where you do see conversion, the pre-patency is very long. And these parasites are ones in which the excision has not happened. So MIT1 then seems to be very important for conversion from the liver to the blood stage of the parasite. What does MIT2 do? Now, MIT2 knockout has not yet been made, but we looked at interactions of MIT2 with other replication proteins. And firstly, we tried to understand whether MIT2 has a role in the MMR pathway. For this, we looked at mutase. Look at the kind of protein you have as mutase. It's so complicated. It's got these large numbers of insertions, so many disorders. This is an AlphaFold model. You can't even make out what's happening here with the protein, but you do have a DNA binding domain that's conserved. You have some conservation of the core domain and the ATPase domain. But what is very interesting with mutase of Plasmodium and Toxoplasma is that both these have an additional endonuclease domain. And this endonuclease domain is a GIY-YIG endonuclease, which tells us that if this mutase is functioning in the MMR, you may not need an additional endonuclease. So, and that this would function as the endonuclease required for MMR. And when we did interaction analysis using antibodies against these two proteins, we find that we could pull down mutase when we use the MIT2 antibody, indicating interaction. With Mrinal and Abhilasha, Mrinal's student, in fact, Mrinal and I discussed this in the last ISP, and in this ISP we have data and also a publication, so that's good. And we did a pull down using RAD51 to see whether there was interaction with RAD51 and this MIT2, and Abhilasha showed that there indeed was interaction and that the RAD51 antibodies could pull down PF exit MIT2 from the lysate, and that therefore this protein could also be involved in the DSBR repair in the parasite. So what are we looking at? So basically in the mitochondria, this is a long 6 kb repeat concatenameric structure, and what we are saying is that MIT1, which is only working on ssDNA, what does it do? I mean, this would not, so it's very difficult to envisage a role for an exonuclease that's only a single-stranded DNA cleaving protein. You can't locate its function in replication. You can only locate its function in, not even in repair. So what we think is happening is that when mitochondrial DNA replicates, it's a very old paper from 96 from Peter Preiser's group, who had done EM studies on replication of the mitochondrial DNA, and they showed that during that process, a lot of single-stranded DNA puddles are formed. And these puddles must be cleared from the parasite. And we think that this exomit is very important for clearing these puddles. And if these puddles accumulate, then these might be toxic, and therefore you won't have conversion from the liver to the blood stages. In repair, now we know that this protein exomit2 can have a role in DNA replication, but we yet have to establish this through its proofreading activity on double-stranded DNA. It can help in replication, but it certainly seems to have a role in mitochondrial DNA repair in the DSBR. And so this can have an end. So once you have an end, once you have a cleavage like this in DSBR repair, this might have a role in end resection to generate a 3' overhang, after which PF-RAD51 and other proteins would come in to repair the protein, the DNA. In mismatch repair, we think PF mutase is the enzyme which recognizes a mismatch. It has an endonuclease domain itself. So this endonuclease will then cleave the DNA close to the mismatch. And once the DNA is cleaved, the 5-3 exonuclease and the 3-5 exonuclease activity of MIT2 can come into play, and the MMR process can move forward. So that's our understanding currently. But I want to leave, this is my last slide, I want to leave you with a thought. So we have done knockouts with Satish's lab on all these proteins. And what we have seen is that when we look at the base excision repair proteins, we find that we can make direct knockouts. So direct knockouts are possible, the parasites survive. So in fact, in APN1, you can actually, we don't see any difference. So APN1 is not the major protein, not the major AP endonuclease. APE1 is the major endonuclease. And we see delayed patency over here. But we see perfectly normal blood growth. OGG1 also a similar phenotype. We see a normal blood growth, but we see delayed patency. So clearly the liver replication is affected. But with both the exonucleases that we made a knockout of, we couldn't make direct knockouts. We could only make conditional knockouts in Burghi. And both these knockouts could not move from the liver to blood stage. They were completely blocked in the liver stages. So the exonucleases clearly seem to have a very important role. And we think it's because their role is not only in repair, but also in the replication process. And for MIT1 particularly, the role is primarily in clearing of ssDNA buckets. So what would happen then is that once you have schizogony in the liver, and as you know, the schizogony in the liver is a very huge process. You have a number of parasites, large number of parasites are made here. So what would happen is that because we have multiple copies of the genome, some of the merosomes or merozoids that are formed in the liver would have, there'll be differences in the nature of the mutations. So if there is an error in repair, some of them will have normal DNA, and the ratio of the normal to the mutated DNA would be something that would determine whether a merozoid is degenerate or whether the merozoid survives. Okay, so you will have ones in which mutations have accumulated in a large number of genome copies would actually degenerate or would have very low infectivity or slow growth. So this is called organelle heteroplasmy. Because you have multiple copies of organelle genomes, there will be heteroplasmy in organelle DNA. And because of this heteroplasmy, you will see different kinds of phenotypes. And we know that mitochondrion, that was the understanding, that the estimates have ranged in terms of mitochondrion genome copies to more than 20 copies in Plasmodium. But there is now an estimate from Florentine Enard's group that suggests that it might only be pentaploid. But even if it is pentaploid, organelle heteroplasmy would come into play. In the epicoplast, estimates have varied from 1 to 15 copies. But again, this paper seems to suggest that epicoplast might have only one haploid genome. This is surprising to us, and I think more experiments need to be done to understand whether this is actually the case. But if this is the case, if there is only one haploid genome, this might explain the drastic effect of exofen that we see in parasite survival. I would like to end here by thanking my students, Tribeni, who did all the work on the exofen and the work on exomit1, Shivani, who did the work on exomit2, and Anupma, who had initiated this work when she was a PhD student in my lab. My collaborators, Satish, who's done all the knockouts that I showed in this, and his students, Mrinal, with whom we did the AGAT51 interaction, Stuart, who's helped us initially with localization of OGG1, and Imran, my colleague at CDRI, with much of the modeling. And funding from CSIR, of course, the JC Bose Fellowship from the NRF SERB, and the DBT, which actually funded this project. Thank you very much.
Nice talk as always, and we do have time for 2-3 questions. If
someone very thorough and very nice talk. My question is, how does these various exonucleases get the specificity, whether they have to act at the mitochondrial level, or they have to act at the apicoplast level, or the normal genome?
So that's targeting. So, that specificity is not a result of their structure, it's a result of their targeting. So these are so I didn't explain that, but they have targeting sequences, and that's how we initially sort of made this list of putative organelle proteins looking at the targeting elements. So the exofen that I talked about has a very clear epicoplast targeting sequence. It's also now part of this epicoplast proteome analysis has been done. It's part of the epicoplast proteome as well. So, it's clearly an epicoplast protein. So initially we had shortlisted it based on its N-terminal bipartite targeting sequence. And the activities, of course, those are because of its structure and the way it interacts with DNA, and also the presence of this LCR in the middle. That's more structure related. But the specificity of organelle is not because of that. Okay. That's because of the way it's started.
The other question, they're recognizing DNA with the help of some complexes at least, or they will not be because
they bind to DNA in vitro, Pavan. All our experiments that I showed you here are in vitro,
but right? I think all of it, when exonucleases are there, they must be having some other helicases or other.
Oh, of course, the complex will be much larger. Yeah. So if we are envisaging a role for this, that's why we did a pull down with the PFP-REX, because the idea was that does it actually interact with the complex? Clearly if it's working in replication, it's not going to go and bind DNA alone. There's such a big replisome complex at the DNA replication fork, which is why the interaction with PFP-REX was checked initially. And it does interact. So that's clearly there. So there is an interaction. We now have to look at the larger complex in the epicoplast to see what proteins are part of it. So in addition to these two.
Similar to this line. So now in the mitochondrial, is their complex different than apicoplast, or in the genome it is different. So have somebody really try.
Nobody is on anything in organelles, and you know why? Because
Yeah.
they're so tiny,
Yeah.
they're so difficult to purify.
Yeah,
So all you do, so
I
people are
do agree
trying.
with
Somebody's here is waving his hand, and I know who it is. so people are trying to do it, but to look at replication forks and especially in the mitochondria, where there's not a replication fork, it's a rolling circle mechanism in Plasmodium, unlike toxoplasma. So toxoplasma is a very different mechanism of replication of the mitochondrial genome, but this has a rolling circle mode, and we really don't understand what happens there. And we think that, and this DNA polymerase, as I said, is so different. If you look at the structure, it doesn't have an exonuclease domain. So then which exonuclease works in replication, right?
Question? Very nice talk. In this 4Fe4S cluster containing protein, is it oxygen sensitive?
Yes, it is. Yes,
so,
it is. So we do all this work in a glove box.
Okay.
Yeah.
And did you do EPR on that? Electron paramagnetic?
No, we didn't. So we just did a UV phase, and it's such a clear 420 nanometer peak, and this peak is not there in the bug head. And we can actually oxidize it and remove it chemically, which is what we did when we were testing the differences in activities between these.
Also, did you take a native protein mass spec? The native.
We have native protein mass spec from the Parasite.
No, or from your system, whatever you're purifying. After you reconstitute, did you take a native?
No, we didn't. No.
This is
so
very
what we've done is purify it. We purify the recombinant protein.
Yeah.
And the recombinant protein expressed in E. coli has a 4Fe4S cluster, like we've seen.
You don't need to reconstitute it.
We don't need to reconstitute it.
It's a stable. It's a stable.
Yeah, it's a stable cluster. We purify it. It's fairly stable during the purification. The last steps of purification, where it's more exposed, we shift to a glove box. And it so this is a stable, relatively stable complex compared to other proteins. So we have worked on the ISC pathway. I mean, iron sulfur cluster has been a favorite thing for me for many years. So unlike the ISC proteins in the mitochondria, which are all iron sulfur cluster biogenesis proteins, which the oxidation is much faster there. You can actually they're more unstable, the iron sulfur cluster. But here the iron sulfur cluster is not very unstable. Either in the structure it is more internal, that's possible. Maybe the large insertion actually masks it and prevents the oxidation from happening, a possibility. But we haven't looked at that. It might be more internal in the protein in terms of the structure.
So if it gets oxidized, the activity is lost.
Yes. If you oxidize, no, activity is not lost. The activity is not lost. I showed that. I said that when we remove the iron sulfur cluster, the activity is not lost. So it's not critical for activity. What we think might be happening is that in vivo, so this has been shown for some DNA replication proteins. The iron sulfur cluster is not important in vitro for their activity, but actually what happens is in vivo is that this is required for charge transport. So the recognition of a DNA break or a DNA lesion is dependent on the charge transport that happens via the cluster on the DNA protein complex. So that's the that's why you may not see their criticality in vitro, but they would be very important in vivo for recognition of the lesion sites.
Very interesting.
Yes.
Yeah.
Thank you very much.
Suman, given this apicoplast exonuclease, given the bidirectional nature 3 to 5 and 5 to 3, in terms of structure function correlation, how we can't anticipate that it's just recombination hotspot?
What is a recombination?
It's a recombination hotspot, given the function in terms of structure
that
function.
That is not a recombination hotspot. What I was saying was not for in the context of the exosomes.
Why can't think of that it's just a recombination?
Well, it could be, but you have to have a lot of data to understand recombination hotspots. What I was saying in terms of recombination hotspots was that the LCRs present in falciparum, in the whole genome, there are a lot of LCRs in various positions on various exons. And that's been proposed to be a possible site for recombination, and that would give you the antigenic variation that you see in falciparum. That's what I was saying. This structure is very important. We haven't been able to get a cryo-EM yet, but there is a group in the UK which has contacted us and is interested in looking at the cryo-EM structure for this. So we hope that they will do it.
Any more questions? One last one. That should be the last.
Very nice talk, Saman. Is MIT2 specific to epicomplexans, what is known about
it? It is only present in certain alveolates. It's specific to, and it's present in chromera. So it's not only for epicomplexans, also for chromarins. So it's there in chromera and vitrella. And in the epicomplexan group, it's not present in all epicomplexans.
Oh.
It's not present in cryptosporidium, which also tells us that cryptosporidium has a mitosome, doesn't have a genome. So okay, so it's not going there. Huh?
In toxo?
In tox is present.
It is.
Yes.
And can you speculate the effect of MIT2 knockouts? You haven't been able
speculate. We'll have to do it. I don't want to speculate.
Thank you very much.
Thank you, ma'am, for your valuable talk. May I now request the chair, Dr. Krishna Pandey, to kindly felicitate speaker as a token of appreciation. Also, there is a quick announcement for Flash Talk participants. Please submit your PPTs in Hall 2. Thank you.
So the next speaker is Dr. Pushkar Sharma. So I have the great pleasure to invite Pushkar. He's a chief scientist, staff scientist at the National Institute of Immunology, and his research focuses on dissecting the eukaryotic cell signaling pathways with a particular emphasis on Plasmodium falciparum. Pushkar, floor is yours.
Good morning. We have no chiefs. We have only lesser mortals, staff scientists at NII. No chief scientists like someone. So, correction. So at the outset, I'd like to thank Satish, who's also very Sushil, as we learned yesterday. Thanks, and Radha and the other organizers for putting up this great show and for this invitation. I think my last visit to CDRI was at the old campus, even though I'm just a few hundred kilometers away, and that was at a SBC meeting, and I believe SBC meeting will be held this year. So that is not a hint for anything, but just a bit of history playing out. As he suggests, as Dr. Shamal Roy, who I've known for many years, and glad to be speaking in a session chaired by him. I mean, we work on eukaryotic signaling pathways. The reason is that because I still had little interest in neuronal signaling, which was from my old, 25-year-old postdoc days, and Plasmodium qualifies barely to be a eukaryote, so that's why we have it on my website. So what we have been doing for last few years, almost 24 years, is trying to carve out the signaling pathways in malaria parasite, and that stems from my interest in signaling per se. And what we have been doing mainly for last is that looking at second messenger mediated signaling, which are mediated by second messengers such as calcium and phospholipids and phosphonucleotides, which have actually taken us into very adventurous and interesting territories in terms of things which we were not very equipped to learn, but then thanks to very smart people in the world, they collaborated with us, such as involvement of calcium in the host cell invasion, and its crosstalk with cyclic EMP signaling pathways we showed a few years back. And very interestingly, we have been working on phosphoinositide signaling both in Plasmodium toxoplasma, and we have and it regulates interesting things like phospholipid biogenesis, which was earlier not known, the connection between the two, and Cyril Botte helped us out in that regard in France. And then also in recent times, we have also looked at phosphoinositide mediated phosphoinositide 3-phosphate signaling regulates hemoglobin trafficking and involved in non-canonical function of autophagy related pathways, which is namely apicoplast biogenesis. So that was a bit of adventure as well. And somewhere down the line, we actually got interested in division, was one of the mutants for some of these things actually exhibited defects in parasite division. And something which I wanted to do, where I started my lab at NII, but then we got entangled in this and due to other reasons, but for last 3 years or 3 or 4 years, we've been heavily involved in that, and this is what I'm going to talk about today. If I can have the lights down a bit, and because there will be a lot of images. If not, that's all right. This crowd doesn't need an introduction to the development of the parasite life cycle. But what I really like to emphasize is the role of division, which is most fundamental as fundamental as a process can be for any organism. And during liver stages, the parasite, as you learned from several talks this morning and yesterday, from one cell it develops into from one spore it develops into almost 10,000 merozoites over a period of few days. And then when these merozoites come out and they invade the red blood cells, they subsequent to asexual division, you get up to almost 30 odd merozoites, which is in case of falciparum in 48 hours. And then some of these guys, actually, some of these parasites, they decide to differentiate into male and female gametocytes, which upon upon a blood meal, the male gametocyte actually, as we learned from Shraman's talk yesterday, undergoes rapid 3 rounds of replication within 15 minutes to give 8 microgametes, and so on and so forth. And then, of course, you have meiosis, and where cell division again plays a very important role. So we thought we like to address, but there's some very peculiar features of each of these stages of division, each of these stages of parasite, and I would like to highlight a couple of them. As we know, as well known, as we know from textbook knowledge of studies done in mammals and yeast, from one cell you get two daughter cells, and then of course they separate from the mother, and you have 2 daughter cells which go and replicate one more time. Of course, you have 1 round of replication, DNA replication, followed by nuclear division or karyokinesis, cytokinesis, and you get the daughter cells. In budding yeast, slightly differently, but you achieve the same fate. In the case of Plasmodium, it's a different kettle of fish in many ways, and including cell division, especially during asexual stages, there is asynchronous division, which we also call schizogeny, where a single merozoite can, as I mentioned, can give up to 30 odd merozoites. And what happens is, after rounds of after DNA replication, there is karyokinesis, but the parasite does not undergo cytokinesis. So within the same cytoplasm, the parasite continues to divide, and it's only after 1 round of final division you have cytokinesis. And what also happens is, while this is going on, some parasites, while they're still dividing their DNA or undergoing division, the others have still not started doing it. It's only towards the end you get some kind of a synchrony. There is no nuclear envelope breakdown like you see in mammalian cells. No chromosome condensation, some of these things which will become relevant to the talk. As I mentioned, asynchronous division. The spindles are intranuclear. Centriole, so the centriole is there only in male gametogeny very briefly, and there's no centriole. So centrosome, what we call a centrosome plaque. And during male gametogenesis, as I just mentioned, within 15 minutes, the parasite DNA replicates 3 times, and you get spindle poles, you get the kinetochores along this, and what you get is 8 exoneme, and subsequently you get these male gametocyte with 8 of the after exflagellation, which again fuse with the female and give you zygotes and so on. And of course, I won't go into the meiosis and so on and so forth. So what I'm trying to essentially say that is that the nature of cell division in the parasite is very different from other organisms. It's non-canonical, and within for the same parasite with 2 different hosts, it is again very different. In order to appreciate this and to start doing this, what we started doing was, since parasite is a small cell and mitotic apparatus is even smaller, so we had to start adapting ultra expansion microscopy, which has been developed in the field in the last 2-3 years, and where you can see a 5 times magnified, almost 4.5 cell. What you can appreciate is, so we have labeled the spindles with tubulin. Centrin labels the central plaque or mitotic organization center, MTOC, and of course in some cases we'll use for pellicle antibody for GAP45, which is a glidosome complex protein. And as you can see, if the parasite starts dividing, it has these hemispindles. This is just before division, and it has only one central plaque or MTOC. And once it starts dividing, undergoes mitosis, there is duplication of the of the CP or central plaque, and you have mitotic spindles, which spans these two dots, which are the centrosomal plaque. And subsequently, it has these intrapolar spindles as the parasite continues to divide. And once the division has occurred, each of these parasite get their hemispindles black, and so on and so forth. Subsequent to cytokinesis, you get subpellicular microtubules, which stabilize the structure of the brazoid. And of course, so we also can use NHS ester to label the protein dense regions such as ROP3s and mTOR, which you'll see in subsequent slides, and so on and so forth. So Anu in my lab established these studies, okay, and which we have been using to great benefit. So what we have been, in order to address the problem of cell division, what we're trying to do is looking at mitotic kinases and phosphatases is relevant. So if you look at the kinome from my good friend Christian Dorick, published here, 80 odd protein kinases, and there are a family of protein kinases which have been speculated in other organisms to be involved in cell division or been proven to be the case, but in Plasmodium the information was not that clear, at least how they are doing it. So we are interested to be specific for this project in Aurora kinases, Aurora related kinases, which is the subject of the talk, NEMA related kinases, and CDK cyclin dependent kinase related kinases, classical CDKs, and so on and so forth. And of course, we are interested in a phosphatase, which I presented at the last NCP meeting, and where we showed that this is involved in cell division as well as sexual differentiation of the parasite. So the criteria were, if they were essential, they must be doing important things. They must be expressed in the regions, stages of the parasite relevant for division, which is schizons, and of course not investigated previously. And if we have information about the localization, which may be relevant for division. So some of these criteria, we shortlisted some of these candidates. While we are doing it, our good friend Rita Tiwari at University of Nottingham, she has been looking at cell division and including kinase cell division. So we thought, why should we should join hands and actually look at the pan life cycle division process. So where she's actually looking at sexual division using Plasmodium burgeri as a system during the in mosquito stage development. And Keshav Prasad, my longtime collaborator, who's been helping us out, and invaluable support in terms of omics, including phosphoproteomics and so on and so forth. And we wrote a proposal, which was the idea was we could sell to India Alliance, and we got team size grant, which we have been operating the last couple of years. So coming to the subject of today's talk, which is Aurora kinases, which are bona fide mitotic kinases. They are present either on the during cell division in most organisms at the spindle poles, the kinetochore, centromere, and so on and so forth. Shown here is IFA staining from this paper showing the presence of Aurora A kinase and at the spindle pole, and Aurora B kinase at the equatorial at a metaphase plate during the pro-metaphase, and there's a dynamic change in localization when the cells divide, and so on. Plasmodium has this is just for Plasmodium has 3 Aurora kinases, and 3 of them are essential because they cannot be knocked out for blood stage development. And there is in last of 2 or 3 years there has been a great activity, and I will call it a race as far as Aurora kinase is concerned. Rita published this paper on ARC2 during mosquito development, and recent paper from various groups led by Burkholz in South Africa, and with Elizabeth Winsler and others, they actually targeted Aurora kinases and so on and so forth. There are 2 papers in bioRxiv, including one of us on ARC1 with Rita, and other one by Matthew Brochet's group, looking at ARC1 and so on and so forth. And some of these results we're going to talk about. So what we did was, straight away coming to the talk, since they were essential, we used dimerizable rapamycin dimerizable induced tree recombinase. We introduced LOXP site using either CRISPR-Cas9 or selection link integration with the loci of ARC1. And by using rapamycin, we could excise this locus, and we could knock down ARC1 and ARC2 as shown here reasonably efficiently. We looked at the parasite growth rate. This thing, so in both the cases, ARC1 and ARC2. So when I say ARC1 minus, it means rapamycin added, and so on. So there was a significant defect in parasite growth during asexual development. Of course, the question was then, what is the reason for this growth? Because as we know, ring to trophozoite to schizone transition happens. This pointer is not working, but still. So when we try to look at the defects during parasite development, something which stood out was that the nuclei, which is not very clear, which are very nicely segregated in control parasite. In the case of both ARC1 and ARC2 knockout parasites, they were all clumped together. They were bulky masses. Simply, they were unsegregated, and so that was the first hint at something may be happening to the division of the parasite. So we looked for more details. First and foremost, so light कम हो सकता है, अगर नहीं है तो कोई बात नहीं. So we looked at the localization of ARC1 and ARC2. Interestingly, so we have co-stained for a reason ARC1 and ARC2 along with histone, phosphoserine 10 and serine 28 antibody. You can immediately see ARC1 is expressed only in a few merozoites. And you can also see it is only in those merozoites which also has phosphoserine 10 histone. It has not been known. In the case of Plasmodium, we have actually identified serine 10 phosphorylation as a very important, very pinpointed marker for parasites undergoing mitosis. In the case of ARC2, they are present in all merozoites, so that was a major difference, and so on. But then we went to expansion microscopy and we tried to see more into details where is it localized. So we co-stained ARC1 in this case, and we can see by now you would know that these small guys, these are the mitotic spindles and these are hemispindles. So these are the parasites which are dividing. So going along with the serine 10 phosphorylation, this thing, so it is present only in this parasite and it is present as in these dividing parasites too at the polar ends, and so on. And we could see that in these parasites, which has most of these parasites undergoing division with mitotic spindles. Others it is absent. In the case of ARC2, it is in the parasites which are before division. It is present somewhere near the mitotic organization center or central plaque, and we'll come to that localization subsequently in my talk. But also when the parasite divides, the single dot converts into two dots, which is basically almost like at the spindle poles of these at the poles of this mitotic spindle, which is almost like ARC2. And this is just summarizing the schematic which I already described to you, so I don't go into this detail. Now, what happens to when you deplete ARC1 and ARC2? In both the cases, we what we could see is that, as you can see, the spindles are lost. So these are the control parasites, and you knock out ARC1 and ARC2, the spindles are gone mostly. And what you also see is these accentuated number of these green dots, which is actually central plaque or mTOC. So which basically, and along with this, you have seen these bulky, unsegregated masses of nuclei, which are present in both these knockouts, which I tried to describe earlier as well. And what is happening is centrosome duplication or central plaque duplication continues to go on as a nuclear division, but nuclei do not divide, so it continues to be hanging around these things and so on and so forth. So there was no impact on this thing, but it also tells you that collectively there was a defect in karyokinesis because the nuclei were not getting segregated, and that was due to the loss of these spindles, and that is why there was a disorganization with these mTOCs, which were present in these large amounts, and also sometimes slightly away from the nucleus. Now we could quantitate that and this thing. Going forward, what we did was we wanted to see that the next step. I'm going a bit faster now because I'm running out of time. We looked at cytokinesis. To cut a very long story short, so these are basically very nicely labeled GAF45 labeled segmented schizons or segmenters which undergo cytokinesis, beautiful rough trees labeled by NHS ester. But when you deplete ARC2, you can see all. So these parasites TEAM2 or ARC2 or ARC1, they cluster together, and they also have these bulky massive nuclei, and several of them do not have the parasite membrane. So these data and others suggested that these parasites have defects in cytokinesis, and that probably is because of the defects earlier seen in karyokinesis and so on and so forth. Now, if you can turn the first video on. the first one, we looked at what's happening to these parasites, and you can see this is a control parasite which will egress. It will come out of red blood cell, disperse, and all these merozoites are flowing around, ready to invade the next cell. Next video, second center. So look at this parasite and look at the time. It will come out of the red blood cell, but it seems to cluster around right there in the center, right here, and it will continue to do so. Third one, and this was the ARC1, and this is the ARC2. This thing, it comes out of the RBC, but these parasites tend to cluster together and continue to do so, and so on. So, well, this is what we had been observing. So these parasites which have undergone defects in division, nuclear division, and subsequently cytokinesis, just will not be able to go and carry out the subsequent life cycle. Now, so Rita, in the meantime, and I should introduce Ruji Zishan, who's now at CDRI, earlier in Rita's lab. They contributed to understanding ARC1 in the Burgess system. And to cut a very long story short, what they did was they made GFP line. They found and they looked at ARC1 localization during male gametogeny. Within the first 1 minute, ARC1 comes across as a single dot coinciding with the NDC80, which is the kinetochore protein, and subsequently it just lines up as an arc in 2 to 3 minutes along with the NDC80 and so on and so forth. And when they did an expansion microscopy, they found that it was nicely lined up at the spindles. The ARC1 was and so on, and also at the spindle poles. When they depleted ARC1, they found there was a great defect in exflagellation, and subsequently also oconase conversion, as well as there was significant loss in number of poses. Going forward, then they looked hard into performing several expansion microscopy and super resolution microscopy, and they found that the spindles, which are beautifully formed in the normal parasite, the spindles, just like the blood stages, were lost. The sizes were small, or they were not formed properly. They also found defects in exflagellation, beautifully exflagellated the parasites right in the control here in the ARC1. When the ARC1 is present, when you deplete ARC1, there is a clustering of basal bodies or the mTOC, and shown here, and exflagellation does not occur. So this is a similar kind of defects we saw during the sexual division in the case of PF, the falciparum ARC1 mutant. Now, next, so next we wanted to know, why is this happening? What are these enzymes doing? ARC1 and ARC2, how are they regulating these important processes? So with Keshav, we did comparative phosphoproteomics. It is pretty daunting task. We had to get material, but then Keshav Akila, a very bright student in his lab, did the thing. So we looked at global phosphoproteomes when these kinases are present and when they are absent, and compared what are differentially phosphorylated proteins. To cut a very long story short, something which stood out straight away, the significant number of proteins which were differentially phosphorylated, or phosphorylation of these proteins which were altered due to ARC1, ARC2 depletion, was centromere kinetochore proteins. And shown here is in the schematic the ARC1 and ARC2. So several the complex, the kinetochore complex has been identified in Plasmodium by Matthew Brochet and others, and we found several of the proteins in this complex are ARC1 and ARC2 targets. Very interestingly, ARC2 not ARC1 also targets or phosphorylates. Its depletion results in this thing of centromeric variant of histone H3. And also interestingly, while we find these proteins, only 40 of these proteins with ARC1 and ARC2 targets were common. That means they were acting, despite having some similarities in function, they were acting mechanistically different because they're acting on different proteins. We could do we have done an IP immunocomplex, as well as we have validated that and shown that ARC1 in this case is part of this kinetochore complex, as shown by co-IP with other antibodies. Now, so I'll just take you briefly to what happens to some of these targets to dwell more into the mechanisms. So centromeric histone H3, which is a centromeric variant of histone H3, one of its primary job is to assemble the kinetochore complex, and which in turn is important for spindle assembly. So serine 40 is an H3 which is conserved in yeast, but not in the human this thing, and I'll come to that the significance of it in a minute, was found to be this thing. So what we did was we first looked at localization of ARC2 and SNH3, which we found in close proximity during division when the parasite, this merozoite divides. And as you can see in the zoom version, this is not expansion, this is just normal IFA, and they were close together, and we know that they interact, and so on and so forth at the beginning of division, and the parasite divides, and of course it divides into the two different foci, the ARC2 does. And then we tried to answer the question, what happens to SNH3, centromeric histone, which is present very nicely at the centromere. So it is, so we got first this antibody for in common, now we have made our own, and it's present at the centromere. This is centrin, which is the centromere plaque antibody. These are control. When you deplete ARC2, it is ARC2 was the SNH3 was totally gone from the centromere. What we also found, it also was degraded. So clearly, ARC2 depletion had an impact on SNH3 localization or organization of the centromere, and it was lost. We went a step further, and we made a mutant of SNH3 where we mutated this serine 40 to alanine, and we found that the mutant did not localize, the phosphodeficient mutant did not localize in the centromere. So that was which confirmed the fact that phosphorylation of SNH3 at serine 40 by ARC2 is important for its centromere association. But in the case of ARC1, there was no defect on SNH3, and which goes into the fact that it was not found to be altered during the phosphoproteomics. What Anu did was he made a double transgenic line where she overexpressed either the wild type SNH3 in ARC2 background, and to cut a long story short, if this was ARC2 plus and this is ARC2 minus, upon overexpression of the wild type, she found that there was significant complementation of the defects seen in growth, and with the mutant, she did not see that kind of complementation. Now she went ahead and looked at the spindle biogenesis and other things, and he found that, and she took this parasite. In the case of the wild type SNH3 overexpression, the spindles which were lost, of course, they continued to be lost in the case of ARC2, but a significant number of parasites recovered their spindle, and that's why you see the complementation. And similar thing did not happen with the mutants, which was not able to bring the spindles back. Since I do not have much time, I will run through this slide, but except to show that we have gone on to do NUF2, which is the inner kinetochore protein, which is phosphorylated by both ARC2 and ARC1, albeit at different sites. And if you deplete ARC1 and ARC2, NUF2, which comes as two parallel lines as shown by Liantelli, recently we also saw the same thing, which basically resembles the kinetochore, which is indicative of the kinetochore. But when you deplete ARC1 and ARC2, the kinetochore is lost. So it is lost on the kinetochore, and we now have stronger evidence for this. Now, so just to summarize, so what we have shown is ARC1 and ARC2 dynamically localized in the centromere kinetochore axis, and they phosphorylate key proteins such as centromeric histone 3, NUF2, and I'm sure pretty others, and so on, which contributes to kinetochore centromere complex formation, which is critical for spindle assembly, which is very important for karyokinesis, and which is obviously critical for parasite division. Elko Tromer, who is also a collaborator in ARC1 study with Rita, has actually annotated ARC1, which is like aurora kinase B, as part of the chromosome passenger complex, and that is also very interesting, which also is found by Matthew Brochet in their preprint. Lastly, what we have done is we actually we have been trying to target the aurora ARC1 and ARC2 using known aurora kinase library, which we got from Select, and we found several compounds, Ankita Parasati in my lab, which had reasonable IC, which were effective with the parasite. Shown in green are the ones, two compounds which were recently identified in the study I just mentioned. We found 3 additional compounds, and they have IC50 in nanomolar range, and they can all kill parasite during development. One of the very interesting things we wanted to see if it is working against ARC1 and ARC2, we found several of these compounds, while they inhibit parasite growth, they did not have effect on centromeric histone H3, except one of them, AKI603, which just like the ARC2 mutant, also displaced it from the centromere. And of course, we saw the defects in spindles and so on and so forth. I will end here and say Anu is the brave lady in the lab. She was almost single-handedly handled the ARC1 ARC2 story with Nisha support Ranjana, an next postdoc in my lab. Ankita has joined hands in the ARC2 so inhibitor story and so on and so forth. With Rita, as I mentioned, and Ruji and Zishan in the lab, we've been looking at the other for not only for the other kinases and trying to get a more holistic picture including in the mosquito stages, how the division happens. Krishnan Pal Karmodia has been helping us out with a lot of epigenetics, which is turning out to be very interesting. And of course, Keshav, as I said, is a very important piece in the puzzle with proteomic support. India Alliance, DBT, NII, all I appreciate for their financial support again. Thank you very much for your time.
Thank you, Pushkar. Now the floor is open for 1 or 2 quick questions.
Hello.
Hi.
Yeah. A very nice talk. So I want to know that how much domain conservation is between ARC1 and ARC2, and did you try to complement in ARC1 knockout background to put ARC2 back and see that whether it can compensate? Although you are showing that it has substrate specificity, but whether one can.
So the reason why we didn't do this experiment, because both of them are essential. Both cannot be. So Parasite cannot survive by either of these. So while it may go, but so putting R2 back, I do not know if it will bring it back, because they are both essential independently.
Right.
Even though they seem to be performing overlapping functions, but the reason is, but they're performing functions mechanistically acting differently, and so on and so forth. Now going back to what was your first question?
Yeah. So how much domain conservation? Because you were showing initially
so
they
when
are
you
very
see
much.
so domain conservation, so there is a kinase domain, and these enzymes have something called Degron domains, which because of which actually they undergo degradation, and that's probably the reason why you see them in one parasite, not the other. As far as kinase domains are concerned, they're nicely, very well conserved. Of course, there are subtle changes, which, as a recent study suggested, where inhibitor selectivity can come from, and so I'm not going to get, but very subtle. But the kinase domain is very well conserved, but the other features are different, and which may include, some of them include targeting as well as degradation.
Okay, but kinase domain, if it is so conserved, then how you are showing that they are phosphorylating different residues?
So that is the name of the game. So they are also localizing differently. For instance, if you see ARC1, it is right present at the outset, even before cells start undergoing mitosis. ARC2 is not present, and when ARC2 is dividing, and it's also present more at the centromere, where ARC1 we did not. So these are the those are the kind of changes. And I personally feel although we have to prove that, it may also we have to do a degradation of one of the kinases when it's not needed, the other one takes over. Right. It may, but we have to still do an experiment for that to prove that.
Thank you.
Pushkar, very nice talk. So,
That will be a new question, not the last one. Yeah.
My curiosity will always be there, that what are the substrate, particularly in terms of the cytoskeletal element, which actually on these kinases acts so that the spindle formation is stopped.
The substrate, we don't think it's on cytoskeleton. We did not find many. It is in the centromeric proteins and kinetocoproteins. And what is happening is, so you have centromere, you have inner chitochore, you have itokinocor, then you have spindles. So spindles, and that basically these complexes are important for assembly of spindles. So if the basal complex, if I can use the word, is not formed properly, you will not get spindles, and that is why we don't see the spindles. Not per se. We do not have any evidence. I'm not. If there are any spindle formation assembly proteins are the target, that we do not know. But this is what our data suggests.
So, what is in other eukaryotes people have thought of, because aurora kinases have been very well characterized in other.
Yeah, so I find it very boring to say this already exists in other this thing. So what are you doing?
No, it's not that. I'm talking about the substrate.
No, I'm telling you.
So what kind of substrate?
No, I find it very boring. Yeah. But the interesting thing with these damn guys is, with kinases, is that they may be aurora, they could be Kapoor kinase, right? Sorry, yeah, I mean, it doesn't matter. My point is just that what is there in the name? The point is that the substrates. So if you look carefully and did not show the data for NUC2, there are subtle differences in the phosphorylation site. So usual suspects are present, but they are not conserved in other organisms. And actually, we are doing a lot of phylogenetic analysis because we have a lot of targets for a lot of kinases now. And likewise for CNS3, it is there in the yeast and it's not present in other organisms, including also probably in toxoplasma. So it is a mechanism, and that is why the process of division is very different, even though the Kapoor remains the same or the Aurora remains the same, but the substrates, either the substrates are different or the sites of target are different.
So Pushkar, keep it for tea time something, right? Okay, we will stop here.
Thank you.
Thank you, sir, for an insightful presentation. May I now request the chair, Professor Shamal Roy, to kindly felicitate our speaker as a token of appreciation.
The fourth speaker of this session is Dr. Asif Mohammed, group leader at ICGEB, New Delhi. His research focuses on organelle dynamics in Plasmodium falciparum and the identification of key proteins essential for cellular homeostasis as potential drug targets, including studies on parasite proteases, organelle development, cellular stress, and parasite cell death. So, Dr. Asif, the floor is yours now.
Thank you. Okay, first of all, I would like to thank the organizers for inviting us here and having a wonderful arrangement for everyone. Another thing is, sorry, light is fine. Another thing is, it is very difficult to give a talk after Pushkar because you can't impress the audience more. But I will try to be brief. So I am from ICGB, and most of you know about ICGB. It's a intergovernmental organization. We have about 69 member countries and 3 components, one in Trieste, one is in Cape Town, and the other one is New Delhi. So I am from New Delhi, as you know, and my group at ICGB, we work on parasite cell biology, especially linked with organelles, and we try to understand different metabolic pathways in these organelles, those which are different from the human host, so that they can be ultimately targeted. And we work on ER mitochondria, several pathways associated with ER mitochondria, including membrane biogenesis, the contact machinery which is involved in crosstalk. Of course, we have done mitochondrial fission machinery and a number of protein linked with cellular homeostasis and proteases. And same for food vacuole, which is very important and critical for parasite. We have worked on different proteases. We have worked on different lipid metabolism pathways, which I will talk today. Organelle homeostasis, and then based upon some of the targets, we select out good drug targets and we try to develop inhibitors for that, which can be ultimately developed into new anti-malarial. Just to give you one example of a protease system, which we have been working for last many years, which is a ClipQY protease system. This is a prokaryotic proteasome-like system, which consists of ClipQ protease and ATPase. So this is involved in regulation of protein levels in the mitochondria. So this is localized in mitochondria, and we have done detailed study to do knockout and showed that it is critical for parasite survival. It plays critical role for mitochondria function, and if you knock out the parasite, will die. So for the protease, we standardize the protease activity. We set up in vitro activity assay, and Jeremy explained very well today. So there's a MMV malaria box. We screened this box, and we identified one of the hit compound from the screening based upon activity assay. And this compound also show activity on falciparum. That's how the malaria box was selected. And the compound showed about 700 nanomolar EC50 on the parasite, while having a very good selectivity index on the mammalian cell. So in last few years, we have worked on this compound with the help of different medicinal chemists and tried to develop this compound further. In the first set, we identified 3 candidates, which are about 100 nanomolar EC50. We developed these further for physicochemical properties and then improved them further. And now we have these compounds which have EC50 at 1 nanomolar, while maintaining the selectivity on the mammalian cell model. The selected lead compound, because it is a mitochondrial target, show effect on both the liver stages, so it can show reduction in the parasitemia in the liver stage, show very good efficacy on mice malaria model. So we can clear the parasitemia completely in the mice. The mice will survive forever. There is no recurrence, as compared to control, which die in 10 to 12 days. So now these compounds we are trying to take forward. But today I will talk about some of our work done on lipid metabolism machinery and membrane biogenesis. This project we have been working for many years with Cyril and Grenoble, Cyril and Yoshiki, and of course Pawan and Dinesh are long-time collaborators. So Parasite has this fatty acid biosynthetic pathway which is in the apicoplast, but that is not very active in the blood stages and mostly in gametocyte stages. But the parasite also has the scavenging pathway where it takes the lipids from the host milieu or takes the complex lipids from the host and then use it to develop the lipid for its own purposes. And we showed that a phospholipase which is present in the parasitophorus vacuole membrane, this is the site from where the parasite will take up nutrients and other material from the host. It is localized in the PV, you can see here, as well as in TVN. And again, not going into detail, we did a detailed knockdown study, detailed activity assay, lipidomic analysis, and showed that this protein active in the parasitophorus vacuole space, and this when the parasite takes up complex phospholipids, it cleaves it, ultimately developing into using the fatty acid. The fatty acids are utilized by the parasite for development of neutral lipids, which are then utilized for membrane biogenesis or phospholipid synthesis. For this phospholipase, we also developed in vitro activity assay. We screened the library, we identified two compounds which showed IC50 and EC50 very close to each other, and the phenotype showed by these two compounds was very similar to what we see in the knockdown. So again, these two compounds now we are trying to develop with the help of medicinal chemists and with our bioinformatics person Dinesh to take these compounds further. The neutral lipids formed by the strategy, they are then utilized for phospholipid synthesis, mainly PC and PE, and there are three important phospholipids which are required for membrane biogenesis, and the third one is PS. We studied in detail how PS is synthesized in the parasite. So there is one protein phosphatidylserine synthase which is present in the parasite, PSS. Usually there are two kinds of PSS, PSS1 and PSS2. PSS1 synthesize PS from the serine, which is probably available from the hemoglobin degradation using CDP DAG. But the PSS2 which can do interconversion of PS and PE by base exchange. And we try to see what this PSS is doing in the parasite. So first we tried to localize, we did GFP targeting. The protein was localized in the perinuclear space, which you can see here. Which is the ER region, and you can co-localize with the ER marker, and we showed that it is indeed in the ER. The ER in the parasite has some extension towards the cytosol. Most of you know that. Usually there is one or two extension going into the cytosol, and by detailed analysis we showed that these are the sites where mitochondria interacts with the ER, or these are the ER mitochondria contact sites. And then using this parasite, we studied in detail how the ER mitochondria dynamics with respect to this contact changes across the cycle. So you can see here in the ring, there is a perinuclear space, elongated structure. The mitochondria is attached here. As the parasite grows into trophozoite, the elongation increases into the cytosol. The mitochondria also gets elongated and remains bound to this structure. And when you have two extensions, the mitochondria bites from both the sides. And in the late stages, when the ER is around each developing nucleus, the mitochondria also gets branched and then attached with each of the nucleus site. And you can see it more clearly here in the ring stage, trophozoite and schizoid stages. But usually reviewers are not happy with what images you show, so they ask us to show the same images, which you can see here. Again, perinuclear space, the extension and mitochondria is bound here. In the late trophozoite stage, I hope it works, yes. There are two extension and a mitochondria is bound to two sides of the ER. And using all this data, we did the we showed how the ER mitochondria contact dynamics across the asexual blood stages. And by using pull down, and I'm not showing the data, we showed that most of the phospholipid biosynthetic machinery is localized in the ER of the parasite as compared to other system where ER and mitochondria both participate. So that's then we tried to see if this protein is important for the parasite. So we tried conditional knockdown, where you C-terminal tag the protein using this selection linked integration strategy. We tagged it with a glymas ribozyme so that you can cleave the RNA by using a glucosamine based induction. So the fusion protein is again localized in the mitochondria. You see expression in the transgenic parasite as compared to wild type. And if you knock down by using glucosamine, the parasite growth is significantly inhibited. You see down regulation of the protein in the parasite, and the parasite growth is almost 90% reduction, suggesting that it is very critical for parasite survival. With respect to morphology, you see in the control ring, trophozoite and schizoid development, but here ring to trophozoite development happens, but the schizoid development is significantly inhibited. Now, because it is an ER, we checked what happens to the organelles. So, in the control in the late stages, as you see, the ER is like a network structure. Mitochondria gets branched and later gets divided. However, in the knockdown, the ER is collapsed. It gets fragmented. It collapsed onto the nucleus sites, which you can see very clearly here. The mitochondria, although get elongated, but doesn't get branched, and rather gets fragmented, and you can see it more clearly here. You can see the mitochondria gets fragmented, and this is the quantitative data to show the same. Then, to understand its functional role, we did 3 sets of assays. One, we expressed the protein, made the recombinant protein, and showed what is the activity assay in vitro. So, as I told you, PSS1 type can convert PE to PS and PC, whereas PSS2 type can convert PE to PS, as well as a reversible reaction can happen, and this can be controlled by ethanolamine presence. And this is what we showed, that our protein can convert PE, can generate PE as well as PS, and this reaction can be inhibited by ethanol in a concentration dependent manner, suggesting that it is indeed a PSS2 type PS synthesis as compared to PSS1, which uses serine coming from the host. PSS1 we could not identify in the genome, maybe some protein is taking that role. Second, we did total lipidomic analysis of the parasite under knockdown condition. So here to show you that the total fatty acid content did not change. However, there was a change in the phospholipid profile. You can see here the PC level goes down, PE level also goes down, and surprisingly the PS level are not going down. In addition, there was a increase in the combination of scavenged or long chain fatty acids in the parasite. So we thought that because PS levels are maintained, these long chain fatty acids scavenged more from the host might be going to the PS. So we did that assay by using a labeled fatty acid, and we when we checked, we thought it might be going to neutral lipid as well, and then they are utilized for phospholipid biosynthesis, but there was no uptake in the neutral lipids. However, there was increase in uptake in the PC levels. And based upon all this data, we proposed this model. So in the normal case, there's a Kennedy pathway where DAG is utilized for PC and PE synthesis. There can be interconversion of PE and PS. PS can be also synthesized from serine coming from the host. However, when we knock down this PSS, which is present here, doing this step PS to PE interconversion, so there is a reduction in PS to PE conversion. When the PE is reduced, there is a pressure on this DAG so that the PC also get reduced. The PC can be complemented from outside by using LPC and LPCAT system. However, that was not sufficient to complement it fully. The PS levels are maintained because the PE can be converted into PS anyway using PSD. And because of that, there is a disruption in phospholipid biosynthesis, and the parasite is not able to survive. Coming to a linked story, but slightly different, we worked on some transporters which are associated with lipid homeostasis. To give you the background, Dr. Malhotra's team, they tried to identify novel transporters in the parasite, and what they did was they purified the food vacuole and identified the membrane bound protein in this by proteomic studies, and based upon that they identified a number of transporters. One of the transporter was aquaglyceroporine protein, which is again a transmembrane transporters, and this is what we were interested in. As you know, the parasite has to take up a number of nutrients from the host, and a number of transporters are there in the parasite membranes, and aquaporins and aquaglyceroporins are known to take up either the water or soluble salts, and aquaglyceroprotein takes up glycerol, and both of these are important for any cell survival. So we first try to localize the okay, and when the glycerol comes into the host in any cell, it can be utilized for the synthesis of neutral lipids, and it also has a correlation with the glucose and glycolysis, so that there is a critical balance between the two. So to understand the role of this aquaglyceroprotein, so there is a transmembrane protein, has several transmembranes, so of course it is a transporter, and we did a GFP targeting and showed that it is not only localized in the plasma membrane of the parasite. Around the parasite you can see very clearly here, but also in the food vacuole membrane. To prove this further, we purified the food vacuole and showed that the GFP is around the food vacuole membrane as well. So it is localized in both the sites, the plasma membrane and the food vacuole. Okay, again here we did a knockout using a glymas ribozyme strategy. The knockout shows a growth inhibition as compared to control. Here the parasite was not able to develop very well from ring to trophozoite as compared to control. This tag protein is localized again in the plasma membrane. And here again we did lipidomic analysis, because we know that it might be involved in lipid biosynthesis. In the knockout, the total fatty acid contents were reduced, especially C16 and C18 showed a significant reduction, and together these two form a large lipid molecules. And if you see the total lipidomic profile, there was a large difference between different neutral lipids as well as phospholipids in the knockout parasite. To summarize, there was a reduction in neutral lipids, TAG and DAG. If you see the phospholipid, there was a major reduction in PE. So again, to see the effect, and Pushkar has already explained ultra expansion microscopy, so we wanted to see what happens to different membranes in the knockout. And in our lab, Madhi has standardized this strategy. So what you can do, you can fix the parasite on a gel and then denature, and then when you soak the gel in water, it expands, because the cell is bound with the network of gel. It also expands, and it is 4 to 5 times expansion. You can see very clearly the organelles in the parasite, as you can see here in the control. So this is a Shidon stage parasite. You can see clearly the merozoite, the membrane staining, and you can also see the rhopteries developed here. But when you have a knockout for this aquaglyceroporins, you can see that the membrane development was not very clean, although the merozoite were trying to develop, and you can see very clearly the rhoptery development here. So suggesting that it was the membrane development that was affected. Again, sorry, I'll just take some water. Because glycerol uptake is associated with glucose uptake as well as glycolysis and TCA cycle, and Dr. Hima Balram explained very well about the TCA cycle, how that happens in the parasite. We checked what happens to the TCA cycle, and we showed that in the different metabolite, the malate and alpha ketoglutarate, which alpha ketoglutarate is here, and which is basically the starting point in the TCA cycle, they are reduced as compared to citrate, which showed no reduction, and suggesting that there was an effect on the TCA cycle from here. And alpha ketoglutarate, because it comes from glutamine and glutamate, we checked its level, and glutamine glutamate level are reduced significantly. But when we checked other amino acid levels in the parasite, they were also reduced, only 4 or 5 amino acids which did not show any effect. So this includes both the amino acids coming from the food vacuole and those which are coming from outside exclusively, including the isoleucine. So suggesting that there was a change in catabolism of amino acid, because that is linked with the TCA cycle, and because there's a disruption in the glycerol and glucose homeostasis, there's a change in TCA cycle which changes the amino acid level, and this is very similar to what happens under cellular stress, the catabolism of amino acid, and we checked what if it is also playing a role in stress tolerance and all. As you know, that has been shown that starvation and fasting can induce more glycerol uptake, and where aquaporin plays important roles. In Parasite, it has been shown that isoleucine starvation, the amino acid which is coming exclusively from the host milieu, if you give a starvation, it can induce hibernation-like condition in the parasite. So it can undergo hibernation for almost 72 hours. However, in between, if you provide the complete media, the parasite can recover back into the cycle. So we first checked what happens to aquaporin expression levels under this hibernation condition. So we showed that they get upregulated significantly in the parasite. And in the knockdown, if you do the knockdown and do the revival assay, so you can see here, this is the growth in complete media. So as compared to control, knockdown grows about 60%, and it undergoes hibernation. In control, if you provide complete media after 24 hours, you can see revival, of course 70 to 80% of revival. However, in the knockdown, if you provide the complete media after 24 hours, there is only 10% recovery, suggesting that this glycerol uptake is critical for survival under the stress condition. So based upon all this data, what we think is that aquaporin involved in glycerol uptake, which is critical for neutral lipid synthesis, which are required for phospholipid biosynthesis, because there's a balance between PE and PS. When the PE level reduce, the PS there's more conversion here, so PS levels remain same. PC can be made from LPC coming from outside. So mainly it was the PE level which were affected. Because there's a critical balance between the glucose and glycerol uptake, we think that affected the TCA cycle, and that in turn affected the amino acid level in the parasite when we do the knockout. So I'll stop here. Most of the work which I presented was by Omer, Monica, Mujahid, Madiha, and Pragyan, and some of them are here. And lot of lipidomic work we did with Yoshiki and Cyril and Grenoble, and this work was funded by Indo-French grant, and of course we are funded heavily by DBT and DST. And I collaborate with Pawan, Dinesh. Activity essay was done with Lawrence. We collaborate with Volker and Sumit as well. And thank you all.
Finished well before time, so we can have one or two questions.
Asif, yes, very nice work.
Thank you.
So I just clarification I need. So this aquaporin is present both in the FVM, food vacuole membrane, as well as in the plasma membrane.
Yes.
So of course the one is the lipids coming from the host or the cell culture milieu. That's where we have the plasma membrane localized aquaporin. But the lipid because the lipid drops are present in the plasmodium food vacuole as well.
Yes.
Okay. And where the catabolism can take place, the lipid can also come from the FE food vacuole membrane. Is there a way you can disconnect them?
Okay, so we have shown that lipid body, which is associated closely to food vacuole. So we have shown that there's another phospholipase which is involved in producing neutral lipids for bringing it to lipid body here, which is LPL1, which we published earlier. So complex lipid will come, it will be degraded, and neutral lipids will be generated, but that comes through an endocytic process. Aquaglyceroprotein is only involved in glycerol uptake. We don't know what is its function in food vacuole, so we are working on it, maybe involved in some metabolite transfer between food vacuole and the cytosol. But for the neutral lipids with the food vacuole, there's a separate endocytic process. More like hemoglobin uptake, but not exactly same.
You said you see a change in malate levels.
Yes.
Is it going up or?
Malate is going down.
Going down.
Yes. Well, it is going
Does
down.
it get reflected? What about aspartate levels?
Aspartate level also goes down. If it is not here, yes,
Both go down.
both go down.
One more, if not, then thank you very much.
Thank you, sir, for your intellectual talk. May I now request the chair, Dr. Krishna Pandey, to kindly felicitate our speaker as a token of appreciation. We extend our sincere thanks to all the speakers and delegates for engaging sessions. We will now take a short tea break. We have 2 more sessions remaining, so kindly be back in 10 minutes so that we can start timely. Also, there is a special announcement for posted participants. Please assemble in the posted area by 12:30 p.m. as the evaluation will start from 1 p.m. onwards. Thank you. The short tea break has concluded. We request everyone to please take their seats as the session is about to be resumed.
Okay, sorry. So Dr. Vishal Trivedi and his group is exploring the use of Newcastle disease virus specifically to target malaria-infected red blood cell, revealing a novel strategy for antimalarial intervention. Dr. Vishal Trivedi, please.
So, can you? Hello? Okay. Okay.
You're audible, sir.
Okay. So, first of all, I am thankful to Satish and the organizers for inviting me and discussing my recent talk or recent research actually from IIT Guwahati. And this is just an observational study. There are so many mechanistic questions which we still need to be addressed. So I was very happy when I see this poster actually, the conference poster, and the most exciting part was the artemisinin molecule. So that gives me an impression that we can talk about the discovery of antimalarials. So in this particular talk, we are talking about how we can be able to even control the malaria with the help of some of the other organisms which are probably be having the similar requirements as the malaria. And one of the such, oh, sorry, oh, wow, change, okay. Okay, and one of the such organism is the virus, the Newcastle disease virus. They are लेजर पॉइंट का काम नहीं कर रहा है प्रेस ओके ऊपर वाले से और बदलने के लिए भैया okay so Newcastle disease virus, and I will not go through with the introduction part because this is very trivial thing, and from the morning only we are talking about the malaria, so I will skip all these slides. We know that malaria life cycle and all that. In fact, this is the only parasite which most of us are studying when we are in high school actually. So there's a huge importance of malaria in our society. And this is also, I'm not going to talk, this is very, basic thing, okay. What is important is to understand that if there will be a multi-drug resistant parasite, we have multiple options. We can actually have an option of identifying the new targets. These are the things which we are listening from the morning, that how we can actually be able to characterize the different proteins so that we can be able to use them as a target. Then we can also talk about the drug-induced toxicity and drug efflux so that we can, overcome the drug resistance. And the most important part is where actually most of us are trying to work is the drug delivery part. Vaccine is anyway another part where we are also trying to see whether we can take care of the drug resistance with the help of vaccines. But our focus was on drug delivery, okay, and we were looking for the organisms which are actually having the specific affinity for the malaria parasite. So for that, we thought of targeting the blood stages parasite, okay, and we were looking for the molecules which will specifically interact with the RBCs which are infected with the parasite. So and when we do that, okay, we found that the viruses could be one of the choices, and So this is the Siddharth whose work I am presenting right now, and so viruses are already been known to have a the vehicle so that you can use them for delivering the different types of molecules to the molecule. So we thought, why not we explore the NDV, and what is unique about the Newcastle disease virus is that it is not infective, or it does not cause any disease into the humans, because it is a poultry virus. So it does not even replicate also, but it has all the vehicles and all that machinery which is required for the so this I will skip, okay, because of time. So this is we are just showing the hypothesis that the why we think that the Newcastle disease virus is good for utilization, because the RBC surface is having a lot of sugar molecules, and the Newcastle disease virus has a unique receptor on their coat which actually required for identifying the sialic acid. And we thought that the merozoite, which is actually recognizing the RBCs with the help of the sialic acid modifications, and we can also use the virus which is also requiring those same sugar molecules. So for doing this, what we did is we tested how the NDV is going to affect the parasite viability. Okay, so what we did is we tested the parasite not for one cycle but multiple cycles, and as you continue the virus incubations with the parasite's growth reduces over the course of time, and what we found also is that because it is a the molecular interactions, so it is very rapid and it does not require the re-incubations, because even if you do one round of the virus infections, the virus will go and bind to the RBCs, and then it is actually going to disrupt the parasite viability. To test whether it is a virus specific effect or whether it is a NDV specific effect, we tested the different types of viruses. So we tested the NDV, influenza, the IBV, the Japanese encephalitis viruses, and as well as the duck viruses. And what we found is that most of these viruses are not affecting the parasite viability except the NDV. So there was something unique about the NDV, and so when we started the work, we thought of coming with the three different types of hypotheses. One, the NDV is binding to the sugar molecules present onto the RBCs, and that's how it is competing with the parasite for the invasion. The second is probably the NDV is binding to these receptors, and that's how it is lineating the signaling molecules, and that signaling molecules could be not allowing the parasite to proliferate. And the third is we thought of that maybe the NDV is entering inside the RBCs and interfering with the parasite replications. So test the first, we thought of because we were trying to use the virus, so we thought first we should test whether the specificity is there or not, right? So what we found is that the NDV is binding to the infected RBCs with more strong affinity compared to the normal RBCs, and that gives us the hope that probably this difference can be exploited for developing the for utilizing this particular virus, not for only to use as the anti-malarial agent, but also to deliver the drugs specifically to the parasite infected RBCs. And then we also did a lot of the epifluorescence imaging. So this is the epifluorescence imaging. I'll show you the confocal imaging as well. So that actually shows that the NDV is very specifically binding onto the periphery of these RBCs which are infected with the viruses. And then we also did the same experiment in the mice experiment. So I'll show you the mice experiment also, where it shows that the parasite is when we are doing the colocalization studies with the POLI and the NDV. So we labeled the NDV with the antibodies, and what found is that it is actually going and localizing with the parasite. And then we did the experiment in the mice also, right? So what we found is that if you are doing the mice experiment, it is actually co-localizing with the parasite. And so this is one of the experiment where what we did is we did the experiment in the mice with the 3 different conditions. So in one conditions we infected the mice with the POLI, and then we injected the only the plain lysate, or we injected the NDB, which is heat inactivated, and we injected the virus, which is the live virus. Okay. And we did this in different types of conditions, like the pre, post, and the before also, or during the infection as well. So what you see here is that if you are doing the infection, like for example, if you do the POLI infection, allow the the pastemia to go to some extent, and if you do the the NDV injections through the tail veins, what you found is that as soon as you give the NDV injections, the parasite the growth reduces, and that come down to a level, but it again goes up, right? So if you keep doing that injections, because the parasite is probably the NDV is getting cleared from the circulations, so it is again going up. So if you continue these injections for multiple rounds, probably you will not be able to see the growth. And then we thought of what is the mechanism of this this blockage. So what we did is we synchronized the culture and we tested the NDB effects onto the ring or the Schiedon stages, and what we found is that it is specifically affecting the ring stages. So it is affecting basically the invasion, and that actually gives the impression that it might be affecting the invasion steps. So then we thought of whether the NDB and the RBC binding is essential for this. So we look for the many of the NDB proteins, okay, and what we found is that if you are actually pre-treating the NDB with the proteinase K, or if you are treating the RBCs with the proteinase K, which means if you either remove the protein part from the RBCs or if you remove the protein part from the NDB, you are actually going to disrupt the interaction of the NDB with the RBC. So that actually proves that NDB-RBC interaction is involving a protein-protein interactions. We also did the experiment because we know that the NDB has the hemagglutinin proteins, and that is present on the coat proteins. So we thought that whether that particular protein is involved. So what we did is we actually pre-treated the RBCs with the neuraminidase, and neuraminidase is specifically going to remove the sialic acid from the RBCs, and as you see that there will be a direct shift of the NDB binding. And similar kind of experiment we did, but instead of adding the neuraminidase, we also treated the RBCs with the sialic acid, right? And if you add the sialic acid, which will actually going to compete for the binding of NDB to the RBCs, that also is going to have the similar kind of effects. So to test this, what we did is we overexpressed the HN protein into the bacterial expression system. We purified the protein, and then we tested the proteins. Okay, so instead of viruses, we tested the proteins. Okay, and surprisingly, the protein was also inhibiting the malaria parasite growth, and that's the same thing we observed even in the culture images also. So because the activity was very low, right? So we thought of why can we increase the activity of this HN protein. So we did actually the protein engineering part, right? And so the basic of this protein engineering is that the sialic acid binding recognition is actually require the different types of the recognition fragments, right? So you actually require these scaffolds for any protein to bind the sialic acid. So you actually require so if you can actually have the different types of modules, you can have SF1, SF2, SF3, and SF4, and what we found is that different viruses actually have the different types of these binding modules. So if you see the sialic acid, these binding modules are distributed throughout the molecules, and the HN protein, which is present in the NDV, is only having the SF4 and SF1, right? And whereas the other viruses proteins are actually having the more of these binding modules. So, and these binding modules can be incorporated. So, if we incorporate these binding modules, we probably could actually increase the efficiency of the binding, and as a result, we probably could actually get the better anti-malarial activity. So, what you see here is that this is the binding of the sialic acid from the different viruses. And for example, the influenza virus is actually having the binding in the range of nanometer because it has all the 4 different types of modules. So the logic is that we can actually, if we can reintroduce these bonding modules, probably we could be able to we could actually increase the efficiency. So for that, we did a lot of mutations. All these are like in silico mutations, and we tested whether their binding is affinities and all that. And then we actually did all these studies with the help of the molecular dynamics. And what we found is that this mutant of I175Y is showing a better stability, better binding compared to the wild type. So then we actually and what we found is that if you have the wild type, you basically have the 2 different types of modules, the SF1 and the SF4. But if you do these mutations, like for example, if you do the serine to D, right, you are actually going to incorporate the SF3. And if you are going to do the I175Y, you are actually going to have SF1, SF2, and the SF3. Right, so these additional binding modules, when you are going to incorporate into the proteins, you are actually going to have you expect that they are actually going to have the better anti-malarial activity. So this I will skip, right? So we actually tested also whether these mutants are having the activity as the wild type. So we tested where they are binding to whether they are showing the normal activities, and there are differences in terms of whether they are showing the other kinds of whether they are affecting the NDV replications and all that. And then we also tested their effect inside the chicken. So whether these viruses, whether these proteins, or whether these proteins are good enough to inhibit the activity of NDV. So this is the NDV infected chicken, and this is the wild type. So, and compared to the wild type, you see that the effect is very much enhanced, because when you are injecting a protein, it is actually going to bind the sialic acid, and it does not allow the virus to go for the multiple round of applications. So that will prove that these mutants are much potent compared to the wild type proteins. And then we actually overexpressed, we purified these mutants. So these are just the experiment to show that they are actually better in terms of the binding as well. So this is the experiment where it shows the the binding of the wild type versus the I75Y. And this is the experiment where we actually compared the wild type versus the I75Y. Although still it is not very significant, it is not very super duper what we expected actually, but there is a hope that probably we can actually do even multiple more mutations. We can actually try out many more mutations to bring down this efficacy to a certain very high level. Okay. So this is the summary of the story, that we are actually, this is the RBC, and we are working with the different types of hypotheses, because as I said, this is the observational study so far. We have not even worked out the mechanistic part, where we actually trying to now see whether the NDV is actually entering inside the RBCs, and whether NDV is doing some kind of signaling alterations, and because of that it is actually causing the the disruption of the parasite replication, and that's how it is actually going to have the anti-malarial activity. What we hope is that can we have the HN coupled liposomes, and can that be anti-malarial agents? So, and at the end, we actually know that the virus is the interacting with the RBCs, and it is interacting through the surface, the sialic acid molecules, and what we don't know that is whether the parasite is entering inside the RBCs or not, or whether it is interfering with the parasite replications, and what could be the mechanism. And then we also don't know if there is a effect in terms of the virus having the signaling events, or whether the virus is, because we also got the the positive results in terms of the mice experiment also. So we don't know if the virus is actually doing some specific kind of immune system activation, which is also a very interesting part which we would like to explore. And one of the very important thing is that NDB, as I said, NDB we have chosen because NDB cannot replicate and cannot proliferate into the human system, but it actually can replicate into the chicken system. So can we test the effects directly into the chicken, and we can see whether the NDB infection is disrupting the parasite replication inside the chickens. At the end, this is my group. So what the work you have seen is been done by the Siddharth, and all the virus related experiment what we have done, that is we have done in the Professor Sachin Kumar's lab in our department itself, and this is our department, and this is our beautiful campus of IIT Guwahati. Thank you.
So thank you. Floor is open for one or two pressing questions.
Sir, hello.
Over there.
Yes.
Okay. Sir, when we want to simulate protein and DNA, in this case, suppose we are selecting a force field EMBAR. So protein DNA simulation, EMBAR force field or for both molecule, or for protein different force field we need to select, and for DNA we need to select different force field.
I think we should prefer to ask for these technical stuff outside, I think you're asking MD related experiments.
Yes, sir. I am asking, suppose I want to do protein and DNA simulation, protein and DNA interaction in computer. So in this case, I need to select a force field, better force field, which can mimic my parameters. So can I use a single force field for both protein and DNA?
You can use the EMBAR force field if you like to.
EMBAR, sir, both for protein and DNA.
You can use for both.
Okay. Thank you, sir.
So I have a quick question here. The chairman.
Oh, okay.
Okay. So in one hand, you are showing that the sialic acid is the molecule.
Yes.
But other hand, when you have cloned the protein in the E. coli perhaps, and also showing the inhibition.
Yes.
So theoretically, the sialylation of that protein is absent when you expressed in the E. coli expression system.
Okay.
But in both the cases, it is showing. So it is inhibition is coming by sialic acid as well as by the protein in both the cases.
Okay.
So how do you explain this?
No, I think there's a misinterpretation, right? Sialic acid is a ligand for this particular protein, so it will go and bind to the sialic acid.
Right.
It does not allow the parasite or the merozoite actually go and bind to the sialic acid.
Now, when you express the protein in E. coli, so there is a different type of glycosylation. Whether the sialic acid is present on that.
No,
on the protein?
proteins. We are not talking about sialic acid present on the protein of the NDB.
But when you expressed in E. coli, the protein is also inhibiting the process.
Yeah, but because that also has the similar kind of affinity for the sialic acid, which is present on the RBC surface.
Oh, okay, okay.
So it is actually blocking the receptor what is present onto the RBCs rather than it has nothing to do with the glycosylation of the protein.
Okay, okay, okay. Thank you. Thank you.
Okay, thank you.
Okay, don't run away.
Thank you, sir, for sharing your expertise with us. May I now request Professor Shamal Roy to kindly felicitate our speaker as a token of appreciation.
Professor Sunanda Bhattacharya. She is from the University of Hyderabad and is the leading expert on Plasmodium shaper-1 system and genome maintenance in malarial parasites. Ma'am, for your presentation, please.
Hello. Good afternoon, everyone. First, I would like to thank the organizers, mainly Dr. Shotish and Dr. Saman, for inviting me here to present our work. So today I will discuss the crosstalk between Plasmodium heat shock protein 70 and heat shock protein 90 chaperone systems in client maturation. So in our lab, we are doing work on protein folding machinery. So first, why it is so important to study the chaperone systems in malaria parasites? As we all know, 1/4 of the proteome contains proteins with asparagine repeats, which has an increased propensity for aggregation. Plasmodium proteome also suffers significant temperature shift, and that is how host tries to disrupt the proteostasis of the parasite. And also they encounter high oxidative stress due to their own metabolism, which causes a lot of modification of its amino acid, leading to unfolding and aggregation. So as you can understand, Plasmodium proteome is highly dependent on chaperones. So before going further, I would like to give some very basic idea how in eukaryotes protein folding occurs. So as you can see that when the nascent polypeptide chain is synthesized from ribosome, without the chaperone, they undergo aggregation. So the first co-chaperone which they encounters is the J-domain protein, which are basically co-chaperone of HSP70, and together they fold most of the matured proteins. However, it is found that a significant proportion of the proteome needs additional folding, and HSP70 system cannot fold them completely. So those partially folded proteins are transferred to another chaperone system, which is HSP90 chaperone system, through the help of different co-chaperones, and HSP90 binds and gives them maturation, but remain associated with them, keeping them in an mature inactive state. When the function of these clients are necessary by the cell, then HSP90 is dissociated, and that is how the matured active protein is released. So if we see the HSP70 and 40 partnership little bit in detail, we find that HSP70 has two distinct domains. One is N-terminal domain, which is the ATP binding domain, which is also called nucleotide binding domain, and the substrate binding domain. So J-domain protein is basically working in twofold. It brings the unfolded peptide to HSP70, and through its J-domain, it causes the ATP hydrolysis enhancement of HSP70, which causes a conformational change in its substrate binding domain. And when this ADP is bound, it is again exchanged by this nucleotide exchange factor, so that the substrate binding domain is now open and the folded protein can be released. Now, as you can see, it is very interesting to note that if you consider the cytoplasmic isoform of HSP70, nucleotide exchange factor, and type 1 JDP proteins in human and parasite, In human, there are multiple isoforms. But in Plasmodium, all of these are having only one isoform. So we are trying to understand that if we can block the assembly between the J-domain protein and the HSP70, what will be the impact in the proteome of the parasite? And to do that, we took one very well characterized replication protein of the parasite, which was earlier done by Devapom Chakraborty's group from University of Southern Florida, that is ribonucleotide reductase. So this protein we have chosen because this is a very essential replication protein, as you all know. So what his group has shown, he has studied the total holoenzyme and found that one of the particular subunit PFR2 is essential for the catalytic conversion of NTP to DNTP, and which is why all these substrates for DNA polymerase can be produced. So it is very important replication protein, and it can be inhibited by hydroxyurea, which is a potent inhibitor of ribonucleotide reductases. And he has shown that this protein is majorly present in cytoplasm. So as you can see here, this is the infected parasite within the erythrocyte, and you can see, as I said, 71, that means the HSP70 cytoplasmic 1 is only one present in the cytoplasm. However, so far it is reported that there are two kinds of JDB protein which are present in cytoplasm. One is the ortholog of East YDJ1, we call it as PFYDJ1. Another one is CIS1. So we wanted to know whether there is any specific requirement for any one of these J-domain protein to fold ribonucleotide reductase. So that is the question which we asked, but today I will only talk about the story of YDJ1 due to the lack of time. So as you can see, J-domain protein YDJ1, its domain organization is like that, which has a J-domain, and within that there is a very important 3 amino acid rich motif, HPD, which is found to be evolutionary conserved. And this HPD motif has been shown in our other systems that they are actually responsible for the connection for the JDB protein with HSP70. And there are these kind of regions are there which are important for binding the native, binding the nascent polypeptide chains. So what we have done, we took yeast as a surrogate system, and we took YDJ1 null cell, and we cloned the full-length PFYDJ1 in a centromeric vector, which is giving only one copy, and made a mutant of that where D is changed by asparagine. And then we created a positive control where SCYDJ1 being cloned, and all these 4 isogenic strains were exposed to potent inhibitor of ribonucleotide reductase, that is hydroxyurea, and we measured the survivability. We found that PFYDJ1 expressing cells are able to survive in presence of HU, so they can bypass HU mediated toxicity in comparison to YDJ1 null cell. However, the DN mutant can't. It is behaving same as that of YDJ1 null. And when we probed at the SCR2 level, we find that YDJ1 expressing cell can stabilize SCR2 same as that of Saccharomyces cerevisiae YDJ1 copy, but this DN mutant cannot. So we wanted to see that why that is happening, that this DN mutant fails to bypass HU mediated toxicity. So we measured the interaction between this mutant with the 70 and compared with that of the wild type by our Y2H assay, and we found that this mutant indeed cannot interact physically with HSP70. And further, we purified all these proteins and checked that in presence of wild type PF, HSP70 and YDJ1, YDJ1 dependent ATPase activation is working, but in presence of the mutant copy, it cannot activate the ATP hydrolysis of HSP70. So together we are finding that if there is a particular mutant which cannot be associated with HSP70, then the stability of the model protein can be, stability of ribonucleotide reductase can be hampered. So we thought that can we just hamper this interaction using some small molecules. So we saw the literature, there is a molecule 116-9E, and it was earlier shown to disrupt the association between DNA K of bacterial HSP70 and DNA J. So this is the biphenyl residue which actually blocks and sterically hinders the association between J domain protein and HSP70. So we thought that if we add 11690 in the purified protein mixture of 70 and YDJ1, what happens? And as expected, we found that there is a dose dependent reduction in the ATP hydrolysis activity in presence of 11690 when wild type these two proteins are added, and also by Y2H interaction we find that in presence of this inhibitor these two proteins cannot interact. So then we thought that why don't we check what will be the consequence of adding this particular chemical within the parasite, and let's see what is the outcome. So we did co-purification study to check the interaction between PFR2 and YDJ1 and 70. So as you can see, this is the parasite lysate was passed through the purified protein, and we could trap the stable complex formation between R2, YDJ1 and 70. But when we took the parasite where it was pre-incubated with 11690, we found that although there is a stable association between YDJ1 and R2, but we cannot find 70 to be associated at the same level. So we found that approximately 1/3 amount of 70 is not able to associate, 2/3 cannot able to associate when this particular chemical is added, and that is not due to loss of its expression, because Western blot analysis shows that in presence of 11690 there is no change in the expression of these co-chaperones or chaperones. So then we wanted to understand that what will be the consequence of ribonucleotide reductase level then, because in that condition we are finding that 70 cannot be associated with 40, and as we expect we are also finding that there is a dose dependent reduction in the Plasmodium R2 level in 116 treatment. Further, we have done the LCMS analysis, and we have measured the metabolites of all these metabolites, and we found that there is a drastic reduction in the DNTP levels within the parasite when they are treated with 11690, but these are the some control metabolite which are not dependent by ribonucleotide reductase, where we do not find any such change. And when we treated this parasite with this inhibitor, we observed that there is a complete arrestation of the parasite at the tropozoid stage, indicating that there is a replication arrest within the parasite, because DNTP cannot be produced as much and the R2 stability is also compromised. So then we wanted to understand that what will be the consequence if we inhibit the PFR2 abundance along with we inhibit the catalytic activity of PFR2. So as earlier Devopom Chakraborty's group has shown that benzohydroximate, which is a derivative of hydroxyurea, it has a IC50 of 23.85 micromolar. We also found in the similar range. But when we add the 11690 within benzohydroximate together, we found that there is a potentiation factor of 70.15 fold. And similarly, when we take only 11690 and add benzohydroximate, we find there is a potentiation factor of 55.7, and these two chemicals synergistically interact with each other. And I would like to tell one particular observation done by Andrew Truman group, where he has found that in human cell line, 11690 and hydroxyurea combination is showing potentiation of 1.57. So as you can understand that within the parasite, most probably as they are having only one isoform, the effect is much more significant, and we are finding about 70.5 fold potentiation. So we conclude that inhibition of this complex assembly sensitizes the parasite towards ribonucleotide reductase inhibitor. So the second part is, as I said, that not that all the proteins could be folded by HSP70 chaperone system, and there is another chaperone system HSP90, and as you can see that HSP90 undergoes various conformation change along with the ATP hydrolysis through which the HSP70 dependent client which is uploaded on HSP90 can be folded properly. And you can see that there are a huge combination of different co-chaperones which are involved in this particular chaperone cycle. Now, recent study by Johannes Buchner's group has shown some interesting data where it was found that HSP90 chooses a specific group of co-chaperones for folding a specific subset of clients. That means all the clients are not important for folding all kinds of proteins present within the cell. So that opens a new area, because earlier people used to inhibit only HSP90 chaperone directly, its ATPase activity. But now, if we can understand which particular co-chaperone is important for folding of which client, then that co-chaperone can be targeted in order to reduce the proteostasis of that target protein. So we wanted to understand that what are the, so if we compare with respect to Plasmodium co-chaperone, we find that they have very poor sequence conservation with that of the human homologs, which present a promising avenue for targeting anti-parasite drugs. So what we found is that we wanted to ask whether PFR2 is a direct client of HSP90 also. So what we have done, we have used two different inhibitors of HSP90, 17-AAG and radicicol, and we found that in both the condition, R2 amount is getting decreased in a dose-dependent manner. Further, in presence of Mg132, it is getting restored. That means it is undergoing proteasomal degradation in inhibitory condition of HSP90. And also we found that in 17-AAG treated condition, there also occurs the DNTP levels are going down, and there is a arrestation of the parasite at the tropozoid stage. So we wanted to understand that any co-chaperone of HSP90, can we tell that that is essential for R2 maturation? So what we have done, we took the parasite culture and treat it with two different doses and measured the expression of all these co-chaperones of Plasmodium, and we found that only AHA1 is induced by several folds. As you can see this picture, AHA1 expression at the protein level is induced by several fold in a dose-dependent manner when we inhibit ribonucleotide reductase, indicating AHA1 might be one of the important factor for R2 stability. Further, we find indeed R2 is found to form a complex with AHA1 and HSP90. So we wanted to see what happens if the parasite is not having AHA1, what will happen to the proteostasis. So we are very thankful to Professor Mrinalkanti Bhattacharya's lab. They have generated the AHA1 knockout strain by CRISPR-Cas9 mediated knockout, and we use this particular strain and we found that PFR2 level indeed is very less. So here we are not inhibiting HSP90, just one of the co-chaperone being deleted, and we show it undergoes proteasomal degradation, and we find that AHA1 deleted strains are more sensitive to hydroxyurea, and also we found that they are also having very low ploidy levels compared to that of the 3T7 cell. So together we conclude that we have found out two important co-chaperones of HSP70 and HSP90 chaperone system, which are the important factors for proteostasis of one of the important malaria protein, ribonucleotide reductase, and we show that if we can block the assembly of 70 and 40, it can causes replication arrest within the parasite. So I would like to thank all the funding agencies and all the students and my collaborators, Professor Mrinal Bhattacharya and Professor Johannes Buchner. And today all the work of 70 which I presented is done by Iman Al-Khatib. And Shraddha is also giving a poster. She has done the HSP90 related work. And Boibhav is also giving a poster. I would ask you to go to his poster. He is trying to find some selective inhibitor of one of the uncharacterized HSP90 of Plasmodium. And with that, I will be happy to take any questions.
Thank you, ma'am. We can allow one or two questions because you have finished within time.
Your results of the synergistic effect of the inhibitors of the co-chaperone. That was really interesting. I was wondering, have you tried this with some antimalarial drugs?
We have not tried yet.
Yeah, because also what are the substrates of some of these folding proteins? If they turn out to be some of the drug targets, then this strategy of synergistic
Right. We have not tried
drug
yet,
discovery would be very interesting.
One more. Sir has come.
Sunanda, here. So,
Ma'am.
from the
Oh,
okay.
Yeah.
So my question is that your HSP90, it can act in a selective manner for certain proteins,
Right.
So how this type of specificity is coming
Yeah,
out?
this is an excellent question. It is not known that how out of the whole proteome, only a very selective group of proteins required additional folding from HSP90. That is not known yet. But for the co-chaperones, some selectivity people have shown that for all the kinases, they require CDC37. So without CDC37, kinases cannot be folded, at least in yeast and human. But very interestingly, Plasmodium don't have CDC37. So we really do not know in Plasmodium, so many kinases are there, how that activity is being compensated. Maybe CDC37 like proteins are there, but they have very poor homology. As I am telling, all co-chaperones are very less sequence homology is there with respect to human. Yeah.
Okay. Wonderful talk. I really enjoyed it. So I was trying to say, did you try to check the interactome of all these proteins, co-chaperones?
That is the part which we want to take now.
It's already there in PlasmoDB,
Mhm.
and it may not be correct or not, but at least it gives some idea to us whether with each co-chaperone they are doing a different functions.
Right.
So if you can find out that interactome for each of the co-chaperone and then put HSP there in that prospect, you can really have prediction of the functions at each HSP70 and co-chaperone and the pathways also. Based on that, you can go into that and develop some of the inhibitors.
Right, right. Thank you so much.
Thank you very much. That concludes the session, I think.
Thank you, ma'am, for your remarkable presentation. May I now request the chair, Dr. Krishna Pandey, to kindly felicitate our speaker as a token of appreciation. We thank both chairpersons and invite Dr. J.K. Saxena to facilitate chairpersons. We extend our sincere thanks to all the speakers and delegates for a stimulating session. Your valuable perspectives have truly enlightened our audience. We will start our next session of Flash Talk shortly. So all participants of Flash Talk are requested to kindly assemble at the backstage. Thank you.
Good afternoon, everyone. Welcome to the Flash Talk session of 33rd NCP Day 2. We extend a warm welcome to all our judges and the delegates present with us today. Moving quickly with the Flash Talk format, each speaker will have a total of 5 minutes. A reminder buzzer will sound at 4 minutes and the final buzzer at 5 minutes. Also, there will be no question and answer for this session. We once again request all the participants to immediately line up backstage. We may now call upon the first participant, Ms. Plabita Paul. Good afternoon, everyone. Am I audible? Good afternoon, everyone. I am Plabita Paul from CDRI under the supervision of Dr. Satish Mishra.
I'm here to present my work on the topic Plasmodium berghei PPT is required for maintaining sporozoid shape and liver stage infection. So before going into the main findings, I would like to point out a specific stage of malaria life cycle, that is sporozoid stage. Sporozoid is highly specialized form which is required for the mammalian infectivity. For the infection, sporozoid needs a specific cylindrical shape and its compact IMC complex and as well as the apical regions. So I would like to say that there are several proteins which are within the apical region and which is essential for the infectivity. I would like today I will say how we selected and identified a specific protein that is highly upregulated in sporozoid and later liver stage development. We disrupted this gene by double crossover homologous recombination strategy, which reflects that it is dispensable in blood and mosquito stages. The main difference comes here, where the sporozoid is getting bulb-like structure within the salivary gland itself. That is a very unusual case because it generally occurs after the invasion within the hepatocyte. These PPT knockout sporozoids are devoid of circular movement, as you can see in this figure, and when we inject this sporozoid within mice, we got 3 days prepatent delay. The prepatency is nothing but the reappearance of the parasite within the blood stage after the sporozoid infection. We also got reduced liver load after 48-hour post-infection without any invasion defect. To find out the reason behind why the liver burden was reduced, we performed in vitro analysis after the infection into the HepG2 cell, where we found that the merosome number was highly reduced and it fails to rupture the PVM at later time point, which implies us that PPT is critical for egress of merozoites. The PVM rupture and the host actin modulation are highly correlated with each other. Here you can see within the wild type parasite, all the forms which is required for the host modulation are present, which is highly devoid in the case of PPT knockout parasite. So PPT knockout actin fails to accumulate within the parasite. So it implies that it do not efficiently modulate the host actin during the PVM rupture. So to localize this gene or protein within the parasite, we tagged this gene with 3XHA, which precisely localize within the apical region of the sporozoid as well as the merosome, as you can see in this figure also. Here, the RNA sequencing analysis also revealed that PPT is important for membranal integrity because all of the proteins which are present within the glidiosome and IMC complex associated proteins like myosin A, actin 1, profilin, all are downregulated in the case of PPT knockout sporozoids. To validate this RNA sequencing data, we also have done the IFA analysis with anti-PIC5 and SIMP, which is active marker for the IMC complex. In PPT knockout sporozoids, these two markers were highly absent. And in the conclusion, I would like to say this PPT is newly identified apical annuli protein, whose down regulation also affect the expression of the microneedles protein and the apical region protein. The PPT is essential for sporozoid shape maintenance and IMC complex development, and it also plays a very crucial role for the egress of merozoites. Last but not the least, this PPT knockout parasite failed to efficiently modulate the host actin during the PVM rupture. So this is all about a specialized gene that is essential for egress, and it is localized in the apical annuli protein, which give us a impactful insight about the sporozoid biology. Thank you so much.
A quick announcement. All the participants in evaluator or poster session are requested to go for lunch beforehand, as the evaluation will start from 1:30. So our upcoming participant is Dr. Kamraj Chinnaperumal.
Okay, thank you.
Very good afternoon, Ronald. Today I talk about the discovery of antimalarial compounds from Angelus murmurus and Caesium aromaticum through in vitro and in silico approaches. This is my overall graphical representation. My research focus on the major 5 objectives like to evaluate the antimalarial activity and traditional used medicinal plants against the chloroquine sensitive Plasmodium falciparum 37 strain, and to isolate active compounds from the methanolic extract of Angelus murmurus and Caesium aromaticum, to structurally characterize it from FTIR and proton carbon NMR analysis, and to assess the cytotoxicity of isolated compounds through ELA cell using MTT-SA, and to predict the molecular docking interaction and drug likeness properties of safety and the safety profiles of the isolated compounds. This is a global scenario of malarial incidents. Methodology. We have collected 62 different medicinal plants from the eastern and western Ghats of the Tamil Nadu, from the suggestion of traditional healers, entry with the traditional healers. And the preliminary study we have published in Journal of Ethnopharmacology and as well as the Parasitology research. Based on the preliminary study, we have selected least acceptive values. We got the least acceptive values of 11 plants. First in this study, we document with the 2 plants, that is Angelus murmurus and Caesium aromaticum. Yes. We have around 176 approximately collected in the fraction and subfraction. Based on the subfraction, we pulled together to the based TLC analysis, and we have finally got 4 purified fraction. That is FC3B and FC3D and FD2B, and another plant is FCC compound 4. Those fractions, final purified fractions showed the least ICP values and highest growth inhibition activity of the Plasmodium falciparum. And as well as this, those compounds showed the least cytotoxicity in a dose dependent manner, toxic effect. And first fraction, so we conclude the based on the proton carbon and LCMS HRMS analysis, and we conclude this bus compound is the leafiol, and second purified fractions for the beta cystosterol, and third one is 1,3 dimethyl pyrimidine and 2,4 one is the dione, and 4 is the ezonal. And then we next for the agreement with the in vitro study, and so the molecular docking analysis and the bioradar availability and bioradar plot analysis. And in our compound, the two compounds showed the Lipinski not violating to the Lipinski rule. That is a 1,3 DMP and ezonal fully compared with the Lipinski rule. Conclusion: the 4 biotic compounds, that is leafiol, betazoline, and the 1,3 DMP and ezonal are successfully isolated from those 2 plants, and also showed the good parasite incubation and the least cytotoxicity effect less than 100 microgram per ml, and the favorable pharmacokinetic profile also documented. And there is a integration of ethnopharmacology and in vitro bioactivity and in silico validation established this the plant derived it's a collection and a validation and documentation. This is a study. Thank for the funders ICMR and CSIR-CDRI and and also my research collaborator and my research scholars. Thank you.
The next participant is Dr. Smritikana Datta.
Good afternoon, everyone. Today I will be talking about a novel function of Plasmodium falciparum artemisinin-resistant determinant KELCH13 as an oxidative stress sensor under heme-regulated proteostasis. So, malaria is one of the most devastating disease worldwide, and artemisinin emerged as the combating for this disease in 2003, and for that in 2015 UU2 got the Nobel Prize. Now, how artemisinin is working? It stimulates alkylation of the parasite targets and protein lipid DNA, but it's a sporadic event and in connection with the heme, and it prevents the hemozoin formation, that is, it prevents the heme detoxification. Now, there are several artemisinin resistant parasites spreading worldwide from 2008, and now it's spreading like hell. And after this, Plasmodium falciparum KELCH13 is the only confirmed marker for the clinical artemisinin resistant, and most of the mutations are dominated in the propeller region. Now, interestingly, in human, the Plasmodium falciparum KELCH13's homologue is KEAP1, and KEAP1 is known to degrade NRF2 and sense oxidative stress. So, what is happening in the normal condition? So, KEAP1 is interacting with NRF2 and it facilitates its degradation in the normal condition, but under oxidative stress, KEAP1 releases NRF2, which goes to the nucleus and then combat the oxidative stress. So, we want to know whether this kind of NRF2 mediated regulation is also true for K13 or not. That is why we selected the mammalian cell line, that is DU145, and interestingly in DU145 the inheritant, the KEAP1 level is low, and when we expressed the KELCH13 in this DU145 mammalian cell line, interestingly we could find that the NRF2 level is lower compared to the GFP line. And interestingly, when we also did the NRF2 and KELCH13 binding in using the recombinant protein in the in vitro condition, we could find that the NRF2 is binding with the KELCH13, and after this, when in the in cellular condition, when we did the IP using the KELCH13 GFP line, we could pull down NRF2. And interestingly, for the next portion, when we treated the KELCH13 expressed DU145 cell line with the H2O2, and in the H2O2 treated cell, we could see the upregulation of the antioxidant responsive genes. Those are NRF2, HO1 and NQ1. Same thing we could also find from our real-time PCR data analysis. And after this, another important thing, that is heme. Heme also induces the oxidative stress. Heme also induces the oxidative stress response, but heme-mediated oxidative stress response or increasing or decreasing in the protein level, this condition is not happening for the KEAP1 or either in the GFP line. So, we wanted to check whether heme is doing anything with in relation with the KELCH13 and NRF2. So, what we did when we are, so NRF2, the localization is generally in the cytoplasm, but when we are treating the GFP expressing cells with the heme, the expression still remains within the cytoplasm. But when we are treating the cells with the KELCH13 GFP expressing cells with the heme, it's moving to the nucleus, which is evident from our IFA data. So here you can see this is the heme depleted media, and the NRF2 is expressing in the cytoplasm, not in the nucleus. But when we are treating the cells with the heme, it is moving to the nucleus. And the next thing, so we wanted to know, as we are seeing KELCH13 is also playing a role with the NRF2 to combating oxidative stress. So, we generated the biotin ligated line for this KELCH13, and firstly we tried to identify whether KEAP1 stress mediated genes are there or not, and we could identify, and then we selected the biotin enriched fraction for the KELCH13 express line, and did the mass spec analysis, could identify 290 genes, 290 proteins, and among them 275 are unique for only the KELCH13 interactome line. And for the clustering analysis, when we did the interactome analysis, we could find 20 top clusters. Among them, top 4 clusters are related to the oxidative stress sensing. And so after this, we did the pathway analysis using the KELCH13 gene, and there the majority of the proteins are involved in localization and also the transport, and they are mainly for the organelle localization and the protein trafficking. And for this we also did the localization of the K13 to the lysosome, and finally we could find this K13 is getting degraded mainly in the lysosome through the CMA pathway. So this is the overall finding. Thank you.
The next participant is Ms. Shweta Sharma.
Okay, so a very good afternoon, everyone. I'm Shweta Sharma. I'm a PhD scholar. So, a systematic review, which I'm going to talk about in brief today, was done under the supervision of my supervisor, this work, Dr. Amit Sharma, and my senior, Dr. Kojong Foucault. So, as you can see here, the title is, this review is all about the transfusion-transmitted malaria and Plasmodium species and the risk associated with its safety. So this is just another way of transmission that we generally don't talk about. And let me begin with a very simple thought. Let's imagine a patient is receiving blood in hospital and what he might be expecting. Safety, certainty, and of course a second chance at life, right? And what if I say that the blood might be carrying parasites in it? And let me also tell you one uncomfortable truth, like in our screening system, while blood bags are well screened for viral and bacterial pathogens, parasites are generally overlooked. So as we are celebrating here 33rd Congress of this parasitology, so I think it will be worth discussing about this topic. So let's just try to understand three major terminology. Residual risk. It's nothing when we miss the parasites, even though we use primary screening tools, RDT, microscopy, etc. It will be called as residual risk, and it can be appeared as two terms: TTM, transfusion transmitted malaria, and post-transfusion malaria, PTM. So the difference is just if malaria infection is just immediate just after receiving the blood, it will be called as TTM, or if it is happening after some interval, it will be called as PTM. So this study is based on this analysis is based on 122 studies, including 6.5 million individuals data. And look what we have found. This figure is just showing that a spike in publication studies from 2014 to till now, and still northeastern part and some other states, Bihar and Jharkhand, are lacking the study and publications from this region. Now, this slide is just showing that nearly 70% of studies were designed as retrospective, while 94.3% studies were only talking about the adult age group, and very few were focusing on the children and pregnant women and neonates, which is important to discuss and to cover. And here types of patients, 91% studies were focusing only on the donors, but inproportionately they were not very clear about the voluntary or the replacement. Replacements are nothing, just it can be your relative or your friend. Now, this slide is telling you the primary screening tools for RDT 46% and peripheral blood film PBF 26.7% tools were used to screen malaria. And Maharashtra recorded more cases using microscopy, and Uttarakhand and Telangana, like states, recorded malaria cases using RDTs. So this slide is all about the comorbidities. Only 4.9% studies were talking about this, and of course, for the obvious reason, comorbidities were beta thalassemia and sickle cell anemia, because they need a regular transfusion, need of regular transfusion. If you can see here the table, only 3 studies out of 122 were actually talking about directly this issue, transfusion transmitted malaria. So this figure will tell you that 74 districts in India are lacking blood banks. So irrespective of malaria, this is a concern if we are talking about the elimination phase, and 13 districts are malaria endemic. So you might be thinking that 0.87% by microscopy and 2.3% by RDT donors still shown the malaria infection, but I think the country like India, even a small fraction can lead to big change and a problematic situation it can be. And no study checked for the TTM risk. So here are some suggestions. We need to switch from microscopy and RDT to nucleic acid based amplification techniques for sure, because this is low density infection, and better reporting strengthen blood banks. And of course we can also use expired blood bags for genetic screening and to know about the more factors of malaria susceptibility. So in summary, I can say that this is very dangerous and we generally overlook, and let's make sure that the every drop of blood that saves a life is also free from malaria. Thank you.
The next participant is Mr. Jadav Prasad Ramachandra.
Okay, good afternoon, everyone. My topic is mitochondrial cytosolic iron sulfur cluster trafficking and protein interaction in Plasmodium falciparum. As we all know that iron sulfur proteins with the iron sulfur clusters performs various roles, as in DNA synthesis, repair, the ribosomal assembly, and the cellular iron metabolism and homeostasis. It may be present in the various forms. These iron sulfur clusters cannot be scavenged from the environment. It has to be synthesized within the cell. So the eukaryote possess three biosynthetic iron sulfur biosynthesis pathway. The sub pathway is present in the apicoblast, the ISC pathway present in the mitochondria, and the CI pathway present in the cytosol of the cell. So this cytosol that lacks one key enzyme, that is cysteine desulfurase, is needed for the iron sulfur assembly. So the CI pathway has to be dependent on ISC pathway for the continuous supply of sulfur intermediate or whole iron sulfur cluster through PF81. So we want to check the interaction between the PF81 and the CI pathway components. So we choose the two early CI pathway components. One of them is NBP35. It having the transmembrane domain and the conserved cysteine residues, which responsible for the iron sulfur binding and ATPase domain. We recombinantly purified the NBP35 and we raised the antibody against this protein in the rabbit. We performed the IFA. It shows the co-localization with the mitotical rate. So it suggests that it's localization near to that mitochondria. It also having the conserved cysteine residues. So we performed the in vitro reconstitution assay, which shows the binding of 4V4S cluster. The 420 nanometer peak suggests this 4V4S peak. So it also having the ATPase activity, which needed for the transfer of 4V4S cluster. So we perform the kinetics for the protein and we get the efficient catalytic efficiency of this protein. So as we move to the second protein, this is SCF101. It also having the conserved cysteine residues for binding of iron sulfur cluster with the ATPase domain and a large insertion in between. So we recombinantly purified this protein and raised the antibody against this protein in the rabbit. We performed the IFA. It shows their localization with the MR, and we use the apicoblast marker as a HU, which is localized in the different cellular compartments. So this protein also binds to the 4V4S cluster in reconstitution assay, and it also having the ATPase domain. So we perform the kinetics for the protein and it showed the moderate catalytic efficiency of this protein. So the main focus of us is to find out the interaction between PF81 and the cytosolic proteins. So the intermembrane extension domain of this protein is protruting towards the intermembrane space. So first we wanted to see their interaction with the NBP35. So we incubated this protein with the NBP35 and it forms a complex, while it not form the complex with the C-terminal domain. To see its interaction with the other CI pathway protein, we incubate to perform the in vivo complexation assay, and we find out that PF81 interacts with the NBP35 and forms a complex, but it could not interact with the CFD1 or SSP101 alone. But when we incubated these all three proteins in equimolar concentration, it forms a larger complex. To see the in vivo interaction, we use PFNTA recombinant as a bait, and we passes lysate through it. It specifically pulls the NBP35 out of lysate. We use the negative control as a E. coli ingate, which you can see that most of the protein comes out in the flow through and watch. So this interaction is very strong. To see the interaction between the NBP35 and SF101 CFD1, we use recombinant MBP35 as a bed and passes the lysate through it. It specifically pulls the CF101 or CFD1 from the lysate. So this data has been published last year in reported journal PLOS Pathogen. When we come to the summary, we can find out that the PF81, which localized in the inner mitochondrial, can transfer the 4S cluster to the NBP35, which forms a complex with SF101 by using the various client proteins. We can transfer this iron sulfur cluster to the cytosolic iron sulfur proteins and nuclear iron sulfur proteins for their maturation. In collaborative study with the Dominic, we have found out that the TG81 knockdown shows the elevated expression of CDGSH1, while there is a when we overexpress the TG81, there is down regulation of CDGSH1 and 2. This inverse effect shows that the this inverse effect shows that the collaborative manner transport of this iron sulfur cluster from CI pathway from intermembrane space to the cytosolic client proteins. And we are going to study this in the Plasmodium falciparum in upcoming experiments. Thank you.
Our next participant is Dr. Gyan Prakash Srivastav.
Good afternoon, everyone. Basically, I'm working on Brugia malai, and this is the in silico computational data. I want to understand about the transcription pattern of stage-specific and sex-specific expression in Brugia. So this is the genome fact. First, like the genome sequence was completed in 2007. The total genome size is approx 95 MB. The total number of coded gene is more than 1000, and the non-coded gene is approx 472. It also contains pseudogene, gene transcript, and other are also annotated. The mitochondrial genome is size about more than 13,000 base pairs, and it total contains 12 protein coded gene, 2 RNA, rRNA, and 23 tRNA. Still, we not have the categorized gene in the database. So first, I want to introduce about the life cycle of Brugia. The first infection after the 0 to 4 day post infection, the first larval stage is there. We can see in mosquito. Then it followed by larval day 2, that is after 4 and 8 post infection day, and then the third larval stage, that is a infectious stage for mammals, that is come after 8 to 10 day post infection. It then L3 enter in mammalian body, and then go to L4 stage and differentiated into male and female, and then the same embryo, mature embryo, that the life cycle is continued. So I'm interested in L3 because this was the infectious stage as well as male and female transcriptome. So what I do, I fetch up the data from the database and RNA sequencing database, and I select only 50 predominant differential expression gene from L3 stage, and we found that the signal transaction, many signal transaction proteins, cytoskeleton proteins, and EV candidates, extracellular vesicle candidate, and housekeeping genes are there. One transporter protein is also mapped, but a number of proteins are still unknown, which are uncategorized. In this, we found that the single transaction proteins are in our study, we found that the single transaction proteins are involved in transmission of the signal and transmission of signal to understand that the L3 is either entered in a mammalian host or not. The cytoskeleton synthesis proteins are synthesized to molding the L3 in L4, and the EVs candidate are involved to understand the host internal environment. Then we go for reactome study, and we found that maximum of the proteins are belong from extracellular vesicles, means extracellular vesicles are involved to understand the internal condition of the host. Similarly, when we go to see the adult male differential gene, we found that different type of signaling transduction proteins and cytoskeleton proteins are synthesized, and new EV candidates are here. It means that these EV candidates are involved in modulation of L3 to male, or either male or female. These are the sex-specific EVs, and still many unknown proteins are there which are uncategorized till now. Then again we go for rectome analysis. Similarly, we do the same analysis with female Brugia worm, and we found that signal transduction proteins are there, but these are again different and different EVs. It means, it concludes that the stage-specific differentiation and sex-specific differentiation of proteome are there, and the gene are sex-specific. And in future, we can use this strategy to understand the stage-specific biological function, sex-specific gene expression, understand the identify the drug targets, and definitely we able to understand the regulatory pathway, that how they are, and definitely, and at the end we able to understand the host parasite interaction. Thank you.
Our next participant is Dr. Vikas Ranjan Giri.
A warm afternoon to everyone. So today I'm going to talk about how the RNA modification is gender biased in case of Schistosoma japonicum, which we call as blood fluke. So if we'll see different Schistosoma species, basically they call the disease known as schistosomiasis, and in schistosomiasis the major cause is the deposition of eggs. So in case of Schistosoma, basically belongs to the trematode, and trematodes, other trematodes, unlike other trematodes, basically this is dioecious. So due to this dioecious nature, the male is more towards the metabolic regulation, whereas the female is more towards the egg output or the reproductive development. So recent reports, several reports have suggested that the RNA modifications have several regulatory roles in different biological functions. So here we aim to study what are the role of these RNA modifications in case of Schistosoma japonicum. So first we use dot plot to detect whether the RNA is modified or not, and more specifically we isolated the mRNA and then subjected to mass spec, and then find out that yes, the RNA modification, that is type is M6A type, is highly enriched in the mRNA of both the male and female worms. Then when we see the distribution of those modifications, they are little bit biased towards the 3' region, however they are falling on the CDS. Then the conserved region is conserved motif is AAACA. Then we find out that most of these modifications are towards the oogenesis in case of the female, whereas in the male they are more towards the regulation of catabolic activity. So which further push us towards what are the regulators or the writers that are basically responsible to regulate these modifications in Schistosoma. Then we find out the M3 and M14 that are mostly enriched in the gonad region as well as in the female, whereas in the male it is more towards the tegument. Then we want to see if they have a bias regulatory role in case of male and female. In case of female, we find out that this regulate the reproductive development. When we generate the knockdown, M3 knockdown female parasite, then we find out that the gonads are severely hampered, which leads to the decrease in the egg output as well as in the delivery of the eggs. So that was also further proved by the decrease in the proliferative cell by EDU staining. Then we search some genes which are severely affected by the changes in the overall modifications. Then we find out that most of the genes are associated with vitellocytes. This was the first part of the story. The second story was how it works in the male. In the male, we found out that it's mostly dispersed in the tegument. Then we go for the single cell sequencing. Then we find out that in case of most of the transcripts that are modified are related to the tegument progenitors. So when we select one gene, that is glutamine synthetase, we also found similar result that the tegument of the worm is severely affected by the loss of several critical ultrastructures of the worm. So the summary suggests that the modifications regulate in a gender biased manner in case of cystosoma, where it regulates the glutamine synthetase and it regulates the tegument or the ultrastructure of the male worm, whereas in case of the female it regulates the oogenesis of the worm, which results in the decrease in the oviposition. Thank you.
Our next participant is Dr. Ajayta Realich.
Good afternoon, everyone. I am Dr. Ajayta Realich, working as a scientist at IVRI Regional Station, Palampur, and today I am here to present my work on the topic Unveiling Haplotype Distribution of Bovine Cryptosporidium Isolates in North India, inferred by 18S rRNA sequences. So, brief introduction. Cryptosporidium is an epicomplexan parasite with a wide host range including humans and livestock. And till date, around 44 parasite species are recognized as valid, and among these 12 have been reported from bovines. Globally, the bovine cryptosporidiosis is the majority of infections are caused by the 4 main species, that is Cryptosporidium parvum, Cryptosporidium bovis and rene, and Cryptosporidium andersoni. Now, these species, they do have an age-related distribution, difference in the pathogenicity, and zoonotic potential. And among these, Cryptosporidium parvum is the most pathogenic and is a causative agent of neonatal calf diarrhea and is of utmost zoonotic importance. The disease is widespread in cattle globally with a global pooled prevalence of 25.5%, and C. parvum is the most frequently identified species. And in India, there are several reports around the disease has been reported from 16 states with a high prevalence. And the molecular characterization studies from the country have reported C. parvum from North Indian states, C. parvum, rene, bovis and andersoni from southern states and West Bengal, and C. Swiss-like genotype from West Bengal. Now, regular monitoring of the disease is important because of the public health as well as economic issues, and the comparative studies on the different species from bovine host are lacking. So with this, we have designed the study to detect and identify the Cryptosporidium species and the risk factors in bovines of Uttarakhand, Himachal Pradesh and Uttar Pradesh, and to evaluate the haplotype distribution and genetic diversity among the detected species in India and their comparison with the global isolates. So we have collected 1417 samples from the calves from the animals of different age group, irrespective of the diarrheal status, both from male and female, and the animals reared by individual owners as well as farms from these 3 states, and we identified 11.78% positivity after MZN stained fecal smear screening, and we identified age as the associated risk factor, where we could detect odds of Cryptosporidium infections were 3.5 times higher in calves less than 3 months of age and 2.9 times higher in animals of 3 to 12 months of age than adults. Now, 4 different species, they were identified in the studied population. They were Cryptosporidium andersoni, bovis, parvum and rene. And these species, they have followed the age-related distribution patterns which are there worldwide, but we found Cryptosporidium rene incidence comparable to that of parvum, and we could find all these 4 species in the calves less than 3 months of age, where the high incidence of Cryptosporidium bovis and rene has been reported along with parvum in this age group. We have submitted 21 unique sequences out of this study in the gene bank, and for global genetic diversity comparison, we have taken or extracted 91 sequences available in the database. The phylogenetic analysis was conducted using Eimeria zwerni as an outgroup, and broadly we found that Cryptosporidium parvum, Cryptosporidium bovis and Cryptosporidium rene, they have clustered together, and Cryptosporidium endosonii, it formed a basal clade. The haplotype distribution, it was found that 34 haplotypes are present globally among these 4 populations with 4 species specific haplogroups, and within these haplogroups there is recombination or gene flow which was detected by the reticulated patterns, and there was substructuring within C. rene, C. bovis and C. parvum. It was further confirmed by various statistical indices like high genetic diversity was identified, and genetic diversity and neutrality indices, they have shown the population expansion among the global populations of all these 4 species. And genetic differentiation indices, it has shown strong genetic differentiation among these species. So to conclude, we found 11.78% prevalence with 4 main species detected in the bovine populations of 3 states of North India, and high interspecific divergence was there, but there was gene flow and no geographical structuring within the species. Thank you.
Our next participant is Dr. Sudeep Ghatani.
So good afternoon. Yeah, I'm Sudeep from Sikkim University, and today I'll be talking about a very neglected trematode parasite known by the name Artificainostomum malayanum. To begin with, we saw 2 problems working with this parasite. First was the taxonomy of the species. This species is known by different names. In India, it is known by the name Artificainostomum cephalotyphex, and in Thailand and other Southeast Asian nations, this particular species is known by Artificainostomum malayanum. So, but again, there were several authors who suggested that these 2 species are same, but again there are other people who said that these 2 species are different. So we just wanted to see through COX1 phylogeny whether these 2 species are same or not. So we found that actually these 2 species are same, and so these 2 species should be synonymized, and so which makes this parasite widely distributed in South and Southeast Asian countries. Then the next problem with this parasite was there was a report by Hodges and Damania in 2018 where they reported about the neglected tropical diseases in India, and they have listed some of the tropical diseases which are found in India, but if you see, there is no mention of foodborne trematodiasis in the country, according to this report. So, but so yeah, so that's what I think the main problem with this parasite could be because of the lack of surveillance mechanisms against this parasite, where this disease, that's why the disease is underreported in the country. So for that we thought of looking for diagnostic markers in the parasite, and for that we know parasites communicate with the host through proteins in various forms like exosomes, microvesicles, or even proteins in soluble forms. So we focused on exosomes for identifying diagnostic markers in this parasite. So if you see the exosomal proteins, they are divided into three components actually. So first is the proteins which are found on the surface of the exosomes. Then there are proteins which are associated with the membrane, and there are proteins which form the cargo. So so we wanted to characterize all these different fractions of exosomes using a protocol described by Sotilo in 2022. So where the protocol briefly, it is we culture the parasite in a suitable medium, followed by isolation of the exosomes using ultracentrifugation and differential centrifugation. Then we did exosome shaving, wherein we could differentiate all these three components, and finally we did LCMS analysis for protein identification. So if you see, we isolated the exosomes and we confirmed through TEM analysis and also through DLS analysis, where we could find the size of the exosomes to be around 30 to 180 nanometer. But whereas in case of exosomes, it is said that the range should be 30 to 150 nanometer. But we got on a slightly higher side. However, we could find proteins, we could identify proteins in the exosomes around 73 in total. And if you see in each of these compartments, we could find unique proteins, which says that, or which suggests that the protocol that we used is actually good for differentiating these three components. And so for identifying diagnostic markers from this, we focused on two components specifically. One is membrane associated proteins, and the other one was surface associated proteins, which is found on the surface of the exosomes. So we could download 33 data sets from the available 13 species in the literature, and we could download data for this excretory secretory proteins and also exosomes. And out of this, when we compared, we found we could find some unique markers which was present only in our trematode species and not in any other trematode species. So there were 5 markers we could find, and we also found one conserved markers which was common to all trematode species. So this is what we got. And sorry, I went. I think it's not. Yeah. But the validation of this experiment is in progress, and we are yet to identify the diagnostic markers for the parasite. Thank you so much.
Our next participant is Ms. Geetika Kumari Ramesh Chandra.
Good afternoon, everyone. My name is Kitika. I am a PhD scholar in National Institute of Immunology, and I work under the mentorship of Dr. Tanmay Majumdar. My presentation is entitled Beta-catenin modulates macrophage alteration to regulate their immunogenic response during Toxoplasma gondii infection. So Toxoplasma gondii is an apicomplexan parasite which is responsible for causing a disease that is toxoplasmosis. It is an obligate intracellular parasite, and in the case of immunocompromised patient, it can cause encephalitis, ocular toxoplasmosis, rashes, hydrocephaly. And also being congenital, it can easily get transmitted from the mother to the fetus, causing brain defects, etc. So when a person ingests food or water that is contaminated with the parasite, the very first tissue which gets infected is the intestinal epithelium, and there the parasite uses one very well-known strategy which is called as Trojan horse mechanism, in which the parasite invades the macrophage. And as the develops a very suitable niche within itself for it to propagate and replicate. And as the parasite, we can say as the macrophage migrates along the body, so does the parasite, and hence causes invasion of the neighboring cells, resulting in parasitemia. And So in our study, we have we have seen that the parasite uses a mechanism, that is beta-catenin pathway, by hijacking the host beta-catenin pathway to establish the parasitemia. And for this, we have validated the experiment using the BMDMs from the beta-catenin flocs as well as beta-catenin knockout mice. And knockout was prepared by using two methods, that is by using genetic ablation, and second is by using a pharmacological agent, that is XAV939. It is a small molecule. So here we saw that in the presence of the parasite, the macrophage produces activated form of beta-catenin, that is the phosphorylated beta-catenin, and it prepares phosphorylated activated forms of beta-catenin by activation of PI3K and AKT. Next, it was seen that this activated form of beta-catenin forms a heterodimer complex with TCF4, and which binds at the promoter of IRF4 gene. Next, we saw that IRF4 is itself acting as a transcription factor for CYBB gene. CYBB gene is well known for ROS production, and here we observed that during infection, there was significant increase in ROS compared to the beta-catenin knockout cells which were infected. Now, when the ROS in the cell increases, the pathway which gets activated is NLRP3 inflammasome pathway, and caspase-1 of the complex is responsible for producing active forms of interleukin 18 and interleukin 1 beta, which are inflammatory and hence forms an inflammatory milieu. Also, the production of ROS is responsible for causing the disintegration of mitochondria, and hence the oxidized form of mitochondria gets released into the cytosol, which activates the AIM2 inflammasome complex. Now, when the cell can no more when the mitochondria gets disintegrated and cell can no more take care of it, it either undergoes apoptosis or pyroptosis. And here we looked for gasdermin D, and we saw an increased gasdermin D in the infected cells, and hence the parasite was actually invading the pyroptosis mechanism of the host to disseminate itself. Along with this, we have also looked for the cytokine profile of macrophage, and we saw that during inflammation there was production of inflammatory cytokine, and in the beta-catenin truncated cells, infected ones, there were anti-inflammatory cytokines. Hence, we can conclude that the parasite takes the advantage of inflammatory milieu to replicate and propagate itself within the host, and in beta-catenin truncated cell there was no replication of parasite, hence causing mitigation of disease. Hence, beta-catenin can be used as a potential therapeutic target for mitigating the disease. For more information, please visit my poster number 110. And lastly, I would like to thank all my colleagues as well as my mentor for helping me go through this process. Thank you.
Our next participant is Miss Monica Singh.
Good afternoon, everyone. I am Monica, research scholar from Punjab University under the supervision of Professor Harpreet Kaur. Today, I am here to present my presentation that entitled Taxonomic Evaluation of Diagenetic Trematodes Infecting Small Ruminants. Let's start with the brief introduction. What does diagenetic trematode means? Diagenetic trematodes are the parasitic platforms having class Trematoda, which possess a complex life cycle which involves two hosts. The first one is the intermediate host and the second one is the definitive host. If we are talking about the importance in veterinary parasitology, they causes the disease in small ruminants leading to economic loss, decreased productivity and increased treatment cost. In my case, the small ruminants were sheep and goat. Small ruminants infected by diagenetic trematodes suffered reduced productivity leading to significant economic loss. And if we are talking about the role of amphistomine in this infection, they play a significant role in this infection by causing gastrointestinal disturbance in small ruminants leading to reduced productivity and posing diagnostic challenges and parasitic management. Here is the distribution of amphistomine in India. This table is the state-wise prevalence of amphistome parasite, which shows that Rajasthan shows the highest prevalence with 83.3% and 75.63% respectively. What is the need of this study? This study helps to enhance understanding of parasite diversity, host interaction, aiding accurate diagnosis, effective treatment. It also helps in enhancing taxonomic understanding of diogenic trematodes, diagnosis, improving targeted treatments and control strategies. It also helps in facilitation of future research and policy formulation. This is the classification of superfamily Paramphistomia, which my specimen belongs to. My sample belongs to family Paramphistomia, come under the genre Paramphistomia, and my species was Paramphistomum apiclytum. If you are talking about the morphological characteristics of trematodes, basically as the name suggests, amphistomes. That means amphi means two and the stomes means sucker. These are those organisms which contain two suckers. One is present at the anterior side known as anterior sucker and the another one is present on the posterior side that is known as acetabulum or posterior sucker. Difference between amphistome and other trematodes is basically the two suckers present. First on the anterior side and another one on the posterior side. For morphological techniques, I use the light microscopy and staining. These are the aim and objective of my study. For methodology, firstly, I collected the sample from the slaughterhouse. After that, rumen was procured from amphistome infection. After that, saline wash was given. And for whole mount stretching and further preservation was done. These are the stain slide and the stain I use is Gauss carmine. These were the whole mount which show the internal morphology like oral sucker, esophagus, pharynx, intestinal cache, genital pore and other internal organs. These are the sagittal section which help in analyzing the internal organs arrangement like acetabulum type, sucker type, pharynx type, position of testes, intestinal cache under different magnified view. This table represents the morphometric measurement of whole amount of amphistome species. Now, challenges in morphological identification. The first and foremost challenge, many species of amphistome share the similar morphological traits making it difficult to distinguish. The next one is the variation in the measurement in shape, size and can lead to misidentification outcomes. The third one is the how one can preserve their specimen. To overcome these, I use the molecular approaches for correct and fast results. I used DNA extraction and PCR, and from this I get the phylogenetic tree and state that till now I have done the molecular of one species, and that species belongs to Paramphistomum apiclytum. Here is the conclusion and future perspective. The continuous evaluation of molecular techniques and imaging technology promises a deeper understanding of amphistome parasite. It acts as interdisciplinary collaboration among parasitologists, molecular biologists, and veterinaries. It enhancing knowledge on amphistome parasite and can lead to improved health outcomes in both humans and animals. Thank you.
The last participant is Ms. Sandeep Kaur.
Good afternoon, everyone. Today I am here to share my research entitled Strengthening Vaccine-Induced Immunity Not to Agonist Enhances Recombinant 70-AKDA Antigen Response Against Visceral Leishmaniasis. It is a life-threatening infectious disease that is caused by the protozoan Leishmania donovani, which is transmitted widely across by a female phlebotomine sandfly species. And the disease is caused by N. infantum in Americas, while L. donovani is responsible for infections in Asia, particularly in India. And the disease is marked by irregular bouts of fever, enlargement of the spleen and liver, loss of appetite, and the significant weight loss. So the drug therapies for visceral leishmaniasis face issues such as high toxicity, emerging resistance and restricted accessibility, especially in the resource-poor areas. So with the rising burden of leishmaniasis, there is an urgent need to develop effective human vaccines to strengthen prevention efforts and improve the control of the disease. So the core of my study is NOD2, an innate immune agonist. It belongs to the member of NOD-like receptors, NLRs, and has been identified as a key player in host protection during disease encounters. NOD2 play a vital role in controlling Leishmania species by facilitating parasite recognition, regulating its intracellular killing, and triggering both innate and adaptive immune responses. Then the highlight includes desmuramyl peptide. They are the potent NOD2 agonists reported to date, displaying nearly double the activity of MTP. They are the potent small molecule immunomodulators with promising adjuvant potential. Its functional profile includes that it enhances the production of cytokines and chemokines, which help in building strong immune environment, and it also improves antigen presentation by the activation of CD4 and CD8 T cell activation, APC activation, and memory T cell activation. It also shows good immunomodulatory and anti-inflammatory activity, so overall making it a promising adjuvant with balanced Th1 and Th2 response with unique immunomodulatory properties. So, in the present study, the role of NOD2 agonist, that is desmuramyl peptide, as an adjuvant, as well as in combination with recombinant 78 kDa antigen in mice model of visceral leishmaniasis was evaluated. These are the group of my animals, and this is the immunization schedule in which mice were sacrificed at before challenge, 8-week post challenge, and 16-week post challenge time point. So first, we have evaluated the in vivo efficacy of recombinant 78 kDa antigen in combination with DMP as an adjuvant through parasite load. And the parasite load was checked through qPCR with the help of microsatellite gene 229 base pair fragment. In the result, as it is here, in the spleen, lower parasite load was observed in the immunized groups, and addition of DMP with R78 resulted in significant protection from L. donovani with 95% of reduction at the 8-week time point and 92% reduction at the 16-week time point. Then we have enumerated the T cell subsets through flow cytometry by in different group of animals, and here you can see that the frequency of CD4 and CD8 T cells were highest in the immunized groups at the 8-week time point, and the magnitude of these cells were highest in the group that received R78 plus DMP at the at 10 microgram of the dose, and the percentage of these cells were higher in नहीं हो रहा है. So, the percentage of these cells were found higher in the R78 plus DMP group at the 10 microgram of dose at all time points. So, the elicited level shows that the vaccine formulation boosts the adaptive immune responses. Then, we have investigated the level of ROS through flow cytometry, and here it is shown that a significant increase in ROS was observed in the immunized groups in R78 at 10 microgram of dose, followed by 5 microgram, then with the MDP, and then the least ROS level was observed at the R78 alone. So, and further, we have investigated the level of NO in different group of animals through ELISA, and here the maximum nitrite levels were seen highest in the immunized group at the 8-week challenge time point when stimulated with R78 antigen, and the maximum increase were seen in the R78 plus DMP at 10 microgram of the dose. So, the elicited level indicate a stronger immune response against Leishmania donovani. Then we have investigated the expression of INOS and NF-kappa B through qPCR, and here the expression of both the genes were upregulated at the 8-week, followed by 16-week, and then at the before challenge time point, and the maximum expression of INOS were highest in the immunized group, and it was upregulated by 10-fold, and NF-kappa B gene expression was increased by 15-fold. So both show upregulation of these show stronger immune response. Further, we have evaluated the pro-inflammatory and cyto-inflammatory cytokine production in different group of animals through ELISA, and the TNF alpha and IFN were highest in the R78 groups, and anti-inflammatory cytokines were lowest in this group in comparison to the infected control. Concluding, we can say that the R78 plus DMP stands as a powerful immune shaping antigen adjuvant pair for future visceral leishmaniasis vaccine development. Thank you.
Important announcement for the participants of Young Scientists. The session will start at 2 p.m. sharp, so please be ready beforehand. Well, that concludes the Flash Talk series of 33rd NCP. We sincerely thank all the speakers and participants for active participation. Lunch has been served now. Therefore, everyone may proceed to the dining area and are requested to reassemble by 3 p.m. to resume the next session. Thank you.
Hello, 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 hello. हेलो 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 फ्रेश आवाज है कि सर थोड़ा बहुत हेलो हेलो ठीक हेलो हेलो
Hello. 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 Hello, mic test. Hello. Hello. Hello.
Hello everyone, welcome to the session of Fundamental and Translation Parasite Biology. We are honored to have Dr. Asim Mohammed from ICGEB, an expert in parasite biology, who will chair this session and guide us through insightful scientific discussion. Co-chairing this session is Professor Vishal Trivedi from IIT Guwahati, a specialist in malaria cell biology, signaling pathways and translational research in therapeutic. May I kindly request our esteemed chair to please take their seats on the dais so that we may begin the proceeding.
Okay, good afternoon and welcome to this session on fundamental and translational parasite biology. We have our first speaker, Dr. Krishna Pandey from ICMR Rajendra Prasad Memorial Research Institute, and he is leading expert on Kala Azar management and elimination strategy. Dr. Pandey.
पॉकेट में रख ले ये फॉरवर्ड है ना इधर
Good afternoon. At the outset, I'd like to thank the organizers for giving me the opportunity. And post lunch, I find very few people in the audience. Anyways, I'd be focusing on some of the treatment that we are giving, particularly for visceral leishmaniasis. As you know, at our institute, we are treating patients, we are diagnosing and treating patients of visceral leishmaniasis, post-Kala-Azar dermal leishmaniasis, and HIV leishmania co-infection. So we are one of the few centers in the country which is treating HIV leishmania co-infected patients. So over the next 25 minutes, I'll just talk about the treatment, some of the treatment that we are giving and some of the treatment guidelines that are there in the national program. So this is the brief outline of my presentation. Everybody knows what is Kala-Azar, so I'll just skip that. What are the traditional management strategies, emerging challenges in the Kala-Azar management? What are some of the recent advances in the drug therapy of Kala-Azar? What major clinical trials that we have undertaken, as well as the other centers, especially Dr. Shyam Sundar, both of us do joint collaborative projects. The reports relevant to the national program and the global initiatives and challenges. So this is all well known to everybody. It is one of the most severe forms of leishmaniasis, and of course, the 4 states of India, Bihar, Jharkhand, UP, and West Bengal do have the maximum number of cases, and of course, Brazil and a few other countries in the world, 88 countries in the world, they have the global burden of visceral leishmaniasis. There is irregular fever, then weight loss, and hepatosplenomegaly, and people with HIV and Kala-Azar, they are very most difficult to treat because they do have a lot of other opportunistic infections. And in our country, tuberculosis is the most important opportunistic infection. Otherwise, we do have a lot of fungal infections as well. And PKDL, of course. These days, the number of cases have come down. VL has come down, but PKDL on the other side is increasing. And it's quite difficult to manage PKDL. And in the lower section, you can very well see that these are cases of PKDL from our institute, and these are very, very difficult to treat because they came to our institute after getting treatment from a few other centers, even at AIMS. They were unable to diagnose the case. And once the RK39 was positive, we did the skin snip examination and found out, and it is very, very similar to neurofibromatosis, which is another disease, the autosomal dominant disease. Then these are some of the treatment options. The antimonials, sodium antimony gluconate. Urea stibamine was the first one, which was discovered by UN Brahmachari, and this year in January I'll be giving a lecture on the UN Brahmachari Oration Award, which has been given to me. So I mean, this is a tribute to him that urea stibamine was discovered by him, and it was the first antimonial long back in 1920s. So sodium stibogluconate was the first drug that was given for the treatment of visceral leishmaniasis. The dose was for 30 days, 20 milligram per kg, IM. So it is quite painful, as well as there are a lot of side effects like the cardiotoxicity, as well as the arthritis that used to occur with sodium stibogluconate, and now it has been discarded. And in the late 1980s, Dr. C.P. Thakur, he did a lot of research on this, and he found out that this drug had become resistant, and now the cure rate, now it is no more commonly used, but it is still used in some parts of the country, as well as it is quite effective in Africa. In the African subcontinent, it is still being used in combination with paromomycin. Then amphotericin B, the liposomal as well as the conventional formulation. Miltefosine, which is the oral drug, and we did the clinical drug trials. I'll just be rushing through the clinical drug trials that we did at our center, as well as Dr. Chamsundar's and Dr. C.P. Thakur center. So what are the challenges? The main challenges are, of course, the drug resistance. Even with miltefosine, we are getting drug resistance. The side effects, which with the advent of liposomal amphotericin B is quite low as compared to sodium stibogluconate. Then a high treatment cost, I won't agree with this, because we are giving it free of cost in the government setup, and limited access in the endemic areas. Again, in Bihar itself, we have got 83 centers which are giving liposomal amphotericin B. This is the same thing, drug resistance to antimonials and miltefosine. Limited diagnostics. This is a point which has to be taken care of by the program, because still in the villages and the primary health centers, they are still relying on the RK39, which is a IgG based diagnostic test. And they are not doing the splenic and the bone marrow aspiration, which is being done at the hospitals. Say at our center, we are doing the bone marrow and the splenic aspiration. Splenic aspiration is technically quite simple, but we have to look for the bleeding diatheses like the prothrombin time, the platelet count, and the total blood count. This has to be taken into consideration. So before doing the splenic count, splenic aspiration, we have to do all this. The bone marrow is a painful procedure, and previously it was being done from the manubrious sternal angle, and the arch of aorta, the thick artery which passes, so it lies just beside that. So if the guard goes just inside, the patient will bleed to death. So that is also technically quite simple. For the doctor it's quite simple, but for the patient it's quite painful. So there's a site, the iliac crest over here. So we are doing it from this. It's painful other than that, but the efficacy is quite less, about 60%, whereas in splenic aspirate it is almost about 90%. So that is still the gold standard. PCR is still not the gold standard, because it is not being done at all the centers. Of course, at our center we are doing it, but for the primary health centers and the community health centers, they are not doing it. And epidemiological shifts, of course, the follow-up is also very important, and as per the national program, we have to follow up the patient up to 2 years now. Previously, it was only 6 months. Now it is up to 2 years that we are following up the patient, because we are on the last lap of Kala-Azar elimination, and the WHO certification will be done at the end of 2027. So, I mean, we have already eliminated visceral leishmaniasis. It is less than 1 case per 10,000 population at the block level, but it has to be sustained for 3 years. So that is another very important aspect. Bangladesh in our vicinity has already eliminated visceral leishmaniasis, but we'll get the certification from WHO only at the end of 2027. and the epidemiological shifts, the changing patterns of transmission and outbreaks in new areas. The new treatment options, of course, liposomal amphotericin B, which is in the program. The oral miltefosine was given for 28 days, but we are not giving it now for VL, we are giving it for PKDL, where we are giving it for 12 weeks. And there are a lot of side effects with miltefosine. So we have come up with a new combination, and soon it will be published. Before December, we will get the WHO tag for the PKDL treatment guidelines from our center, Dr. Shyam Sundar Center, and from Bangladesh. So there are 3 centers which did the trial, and the publication I'll show. With the improved formulations, of course, liposomal amphotericin B and newly developed drugs, the LXE408, we have just completed the clinical trial of LXE408 phase 2, and now we have to go for phase 3, but we do not have enough patients, because phase 3, we do have to go for a large sample size, almost about 300 or more, which is very difficult to get at present. So it is another drug which has been developed by Novartis, and it is again a tablet. We tried out this combination, and we have all completed about 115 patients. The last patient follow-up will be in December. So after that, the data will be locked and we'll go for publication. And phase 3 trials might take place because they have already gone to DCGI for the approval for phase 3 trials. But again, I have told that we do not have enough patients for doing the phase 3 trial. So this is the treatment guideline by the NCVBDC. The main treatment guideline, of course, is the liposomal amphotericin B, 10 mg per kg single dose in 5% dextrose, given over 2 to 3 hours. It has got very few side effects. There is loin pain, and we have to look for hypokalemia. The potassium levels have to be monitored. And of course, it is nephrotoxic, but we have given liposomal amphotericin B even when the serum creatinine was greater than 2, and we did not find any side effect. The patient did not progress to any other clinical kidney disease. Then we did a lot of studies on paromycin and miltefosine combination. It was supposed to be the cheapest and has the cure rate of almost about 98%. This paper is in Lancet. We'll just be showing the Lancet publication. For the HIV co-infected patients, we have provided the treatment guideline, which was done exclusively at my center. This is a combination of liposomal amphotericin B and miltefosine for 14 days. So this is the latest publication which was published last year at our center, Dr. Shyam Sundar Center. Extremely sorry. This is the clinical trial which was done for HIV VL co-infected patients, and in this was done exclusively at my center, and I was the principal investigator. And in this, we gave the liposomal amphotericin B 40 mg total dose plus 14 days of miltefosine, and it is now the treatment guideline for HIV VL co-infected patients. In East Africa, another trial was done, and there it was for the miltefosine was given for 28 days. Otherwise, the ambisome was with the same dose. This is the treatment guideline which was published, and it is now the treatment guideline worldwide, including our country, and it has been taken by the National Center for Vector-Borne Disease Control. This is another very important publication which we did last year, and we completed last year. It was published last year, and it should be the treatment guideline because it has already been taken by the WHO GDG Guideline Development Group, and it was done simultaneously at 3 centers, our center, Dr. Shyam Sundar Center, and in Bangladesh. We treated 126 patients of PKDL with a combination of liposomal amphotericin B 20 mg over 2.5 weeks, only 2.5 weeks treatment, plus 21 days of miltefosine. So this combination worked out to be very, very good, and it was compared with 20 mg per kg of liposomal amphotericin B over 2.5 weeks. So this combination was better, and it could be the treatment guideline. It has already been accepted by the WHO as the WHO GDG. These are some of the clinical trials which I have already talked about. Miltefosine phase 2, 3 and 4. Sitamaquin, this was another 8-aminoquinoline anti-malarial drug which was used for visceral leishmaniasis, but somehow it had to be stopped because of methemoglobin, and there was nephrotoxicity with it. So we did not proceed further to the phase 3 trials. Then paromycin phase 3 and 4, it is a very good drug, but it is an injectable drug, and it is an aminoglycoside. It is an aminoglycoside, so it causes oto as well as nephrotoxicity. It is not now available in our country, although it is being manufactured by Gland Pharma, Hyderabad, but it is not available in our country at present. And we have requested the WHO that they provide us with the paromycin, because there are a lot of cases of relapses where we do need paromycin for treatment. Then amphomal, this was a drug, another drug, liposomal drug, which was not found to be very effective. Less than 90% was the cure rate at 6 months follow-up. Combination of miltefosine and paromycin, this is the best with 98.77% cure rate. A single dose ambisome, of course, this is the only drug which is being used at present for visceral leishmaniasis. Ambisome and miltefosine combination, I have already talked about it. Short course of miltefosine with ambisome, this is only for 10 days. And LXC 408, the phase 2 trials have just been completed, and the last patient follow-up will be in December. So this is the phase 4 trial of miltefosine for the treatment of visceral leishmaniasis. We treated 1132 patients, and the cure rate at that time was 94%, but it has now come down to about 85%. This is for sitamaquine. I have already talked about sitamaquine. This is for oral miltefosine for PKDL, 8 weeks versus 12 weeks therapy, and 12 weeks is still the treatment guideline for PKDL treatment in our country. But it has got side effects, like it is deposited in the retina, so it can lead to blindness, it can lead to itching, it can lead to redness of the eyes. So we have tried out another combination, and I have already talked that this has been accepted by the WHO as the guideline development group, and soon it will be published and will be taken up by the NCVBDC as the PKDL treatment guideline. This is the multi-drug treatment of comparison published in Lancet. This is the efficacy of safety of amphotericin B emulsion, ambisome plus miltefosine for PKDL. This is another study which was done at our center only, and this is for PKDL, where we tried out conventional amphotericin B for the treatment of PKDL, and conventional amphotericin B is to be given in the dose of 1 milligram per kg for 60 to 80 injections. So again, it is nephrotoxic, so we tried out 0.5 milligram per kg, and it was found to be equally effective with lesser side effects. This is the same one which I have talked about, HIV leishmania co-infection. This is pharmacovigilance of miltefosine in VL, and we tried out 1000 patients in the field where we gave miltefosine, and there were no side effects except for some in which creatinine was raised and SGPT and SGOT was raised in a few patients. 10 patients became pregnant. Since it is an anti-cancer drug, it has to be monitored in pregnant females. We do not have any data of a very large study. This is liposomal amphotericin B at my center, and the cure rate with liposomal amphotericin B at 6 months was 95%. This is again a combination of paromycin and miltefosine which I tried at my center. This is in British Journal of Dermatology for PKDL, but the cure rate at 1 year follow-up was about 83%. So it has to be more than 90% to be effective, to be called as effective treatment. This is another study which we did at our center where we compared liposomal amphotericin B with miltefosine for PKDL, and still miltefosine was better as compared to liposomal amphotericin B. This is paromycin again. Now these are some of the reports which are relevant to the program, which we have already given to the program. These are the eye complications of miltefosine, which is published in Brazilian Journal of Tropical Diseases from our hospital. So you can very well see that there is uveitis. This patient lost vision in one eye, and in the other corneal transplant was done. As a result, we have to find out new treatment options for PKDL, which is another posing another challenge to the program. This is regarding the false positivity of RK39. 6 patients of chronic myeloid leukemia also had a RK39 positive. So it is false positivity of RK39, and this we have reported to the national program as well. These are 6 patients from our center. This is for ASHA training. Once we train the ASHAs, we developed the Vaishali model. We trained the ASHAs for the diagnosis and the anybody having fever of greater than 14 days duration or hepatosplenomegaly. Of course, they cannot look for hepatosplenomegaly, but fever of more than 14 days and RK39 positive. So we taught them, and this led to a very important active case detection for the ASHAs in the field. This is again improved surveillance. This is another very important which we did for the program. Field again the same for PKDL. Now this is another challenge to the program. From our center, we have reported 6 cases in which the Kala-Azar as well as the PKDL were simultaneously coexistent in one patient. So it has to be, we reported it to the program, and of course we have to treat the visceral leishmaniasis first because it is more dangerous as compared to the PKDL. PKDL is more for the community and for epidemic rather than for the patient, because the patient does not come to you unless she is a female, she has scars on the face and all. Due to some financial constraints and all, they do not report to us. Most of the PKDL cases we have to find out in the field and then bring them, because we have to explain them that this is not very important for you, but for the program and for the community it is very, very important. This is again one study which we reported to the program, where we found out that even in urine and sweat, the RK39 positivity was about 94%, greater than 90%. So this is another test which we can do, and it is it can be done easily in the field as well. Regarding the vaccination research, it is current status at present, we do not have any widely used vaccine yet, but research of course is ongoing. Leash F1 and MPL-SE vaccine, this is showing promise in animal models and early phase human trials. These are prophylactic vaccines, and the DNA and the protein vaccines being tested for the efficacy in preventing infection, again prophylactic vaccines. But the main challenges are that we do not have any data of any large clinical drug trial based on this. And some of the challenges are ensuring safety and efficacy in endemic populations. So there was a trial which was given to us, but somehow or the other it is not now in the process. So this is some of the global initiatives and challenges. World Health Organization, of course, is aiming for the elimination of Kala-Azar as well as malaria as a public health problem by 2030. Endemic country efforts strengthening national programs to manage and control the disease, and of course the sustainability. Even if we do have one case, so we do need diagnostics and drugs, especially the oral drugs, and LXC408 could prove to be a boon for the future because it is an oral drug. We do not have to admit the patient, and as far as the phase 2 data goes, the cure rate, of course, we have followed up the patient up to 6 months, and most of them have very good efficacy, and of course they did not have any side effects other than slight headache and slight raise of LFT. So sustainability is another big problem which we have to do for up to 2027. So in conclusion, I'd say that the recent advances in Kala-Azar management have shown that they are quite promising, especially with regard to the diagnostic treatment and of course prevention. There is no drug at present for prevention and no vaccine at present, and global collaboration is essential to eliminate Kala-Azar and reduce its burden. So there are a few challenges which I'd like to again mention, that the para-KDL which I showed, the PKDL, the main treatment option is still miltefosine, so we do need combination therapy for that, and of course the active case detection, and that should also be a part of it, so that we eliminate Kala-Azar. And there are a few other endemic foci which are coming up, because we are getting patients from Kerala, we are getting patients from Himachal Pradesh, we are getting patients from Jammu and Kashmir, we are getting patients from Punjab, Rajasthan, along with CL also, because I have not seen any case of CL as far as our state is concerned, but we keep getting reports from VCRC, one of our sister institutes in Pondicherry, that there are cases of CL, and CL, is Sri Lanka is very close, and CL and VL are both caused by the Leishmania major over there. So these are some of the challenges which we do need, and we do need urgent drugs and some diagnostics. Easy antigen-based diagnostic could be important point as far as the investigation is concerned. So with this, I conclude. Thank you very much. I'd be more than glad to take a few questions.
Any question, please? Yeah. This one.
There are new drugs which are in pipeline, which are under the various stages of trials, and we know about their targets.
At present, so I don't find any new drug. Of course, they are just none of them have undergone any clinical drug trial, because you very well know that they have to undergo clinical drug trials, and especially LXC408, which I just showed, that is one of the drugs that is being considered, and we have just completed the phase 2 trial. The recruitment has been complete, and the last patient follow-up will be in December. So it could prove to be a very good drug, because it is an oral tablet, and it has very few side effects.
But are there companies working on the new drug development?
It's a neglected tropical disease, so that is, very few companies are there. So
You have a question? You have a question? Any other question? If not, I can ask a quick question. So maybe you mentioned, so there are a lot of co-infection with TB.
Yes.
So how is the prevalence, and if the treatment regime is different, because TB patients are already under huge antibiotic pressures?
No, then you must talk about HIV, because most of these patients do have HIV co-infection as well.
With TB,
And
yes.
ours is the only center where we are treating HIV-VL co-infected patients. So most of them, say about 6 or 7 out of them have TB.
Okay.
TB is the most important opportunistic infection. And then if you do have TB, treatment guideline is not very different. Suppose if you have a HIV infection, a TB infection, and a Kala-Azar. Of course, you go for Kala-Azar first, and then you start the TB treatment.
Oh,
because TB is very important, because a TB can kill immediately. HIV does not kill. So if the patient starts tolerating the TB drug, say for 8 weeks, or even less, if he does not have any complaint with regard to the tolerance, say vomiting, say any other side effect, you can start the ART as well. And there are 3 drugs which we are giving. We have a ART center as well at our institute. So we are giving tenofovir, lamivudine, and dolutegravir. So these are the three drugs which we are giving for. This is being given all over India. The same drug. So there's no difference in the treatment.
Only staggered. You have to do that.
Yes, you have to plan the treatment.
Plan it accordingly. Okay, if there is no other question, let us thank Dr. Pandey. Thank you very much. We will.
Thank you very much, sir, for that insightful and enriching presentation. May I now request our esteemed chairs of the session to kindly come forward and felicitate our distinguished speaker as a token of our appreciation.
So our next speaker is Dr. Dinesh Gupta. He is from ICGB, and he's a leading scientist in computational and generative design of anti-malarial drugs. So welcome, Dr. Dinesh. Yeah.
Thank you. Okay.
All right. Good afternoon, everyone. And today I will be talking about our recent work, which is yet incomplete, but we have published a part of it, which is about designing a novel PF-LDH inhibitor design using machine learning based library generation. So malaria is a persistent global health threat, and these are the latest statistics, which I need not emphasize to this audience. And but the fact is that there is a urgent requirement for development of novel compounds targeting new mechanisms in the parasite to overcome the resistance and eradication of malaria, which has been amply emphasized by previous speakers, including Dr. Jeremy. So this is a brief history of anti-malarial drug development. It started in 1600s with the quinine era, in which the cinchona bark was used from South America, and it was the first effective anti-malarial treatment, and it was brought to Europe by a Spanish colonist, and it remained gold standard for centuries. And then there were some synthetic breakthroughs. For example, in 1934, a German company called Bayer synthesized ricocin or chloroquine, which was discarded later on because of toxicity issues. Whereas during World War II, Americans realized the urgent need for developing an anti-malarial, and they picked up this compound to take it forward, and they had started a novel program to develop chloroquine. And with this, laid the foundation of modern anti-malarial therapy. And in 1950s and 70s, it was discovered that chloroquine resistance was occurring. It was discovered in Thai-Cambodian border, and it was also observed that resistance spreads to South America, Africa, and other parts of the world. And then combination therapies were developed in order to overcome the resistance, which was sulfadioxine pyrimethylene combination. In 1970s and 2000s, artemisinin was discovered, and 2UU received Nobel Prize for this, and this has been the choice of drug for treatment as well as the cases of resistance in malaria treatment from then on. And then in early late 2000, 2000, it was discovered that resistance against artemisinin is also happening, and this laid the foundation of modern innovations to develop new drug molecules against malaria, and new drug classes were developed, for example, atoquone and fluganil, and novel targets were also discovered, and more research was laid to find out novel targets, and many targets were explored, in fact, and also combination therapies were developed to prevent the resistance, and also next generation compounds were also tried for preventing the resistance, and many of them got into clinical trials, and many of them were mentioned by Jeremy today in his talk. So there is an ongoing challenge of drug resistance, which necessitates continuous innovation and development of new anti-malarial compounds and strategies. So this is a brief overview of my interest in plasmodium research. It started with a fellowship which I received from WHO way back in late 90s, and I got associated with Plasmodium Genome Consortium, which was a consortium to sequence and annotate various Plasmodium species which were of importance and which were also made available to malaria researchers on the website called plasmodb.org. And after coming back from this fellowship, I started working on various plasmodium proteins, because when I returned at ICGB, there is a very big group on malaria research, including Pawan and Asif, whom I started collaborating and started working on various novel protein targets and complex protein interactions on the merozoite surface. And side by side, I also started developing my own tools and databases by the experience which I gained from working in PlasmoDB. So way back in 2011, I developed a method using machine learning for predicting compounds with anti-malarial potential. And in 2017, I developed another method for prediction of arginine methylation sites in Plasmodium, because the existing methods which were predicting methylation in proteins failed to predict the already known methylation sites in Plasmodium genome. And then I also worked on deciphering transcription factors and associated factors in epicomplexans and made a database called EpicoTFDB, which was published in 2019. And then on, I have been side by side collaborating with Pawan and Asif to work on various targets. And we also started working on deciphering the post-translational modifications in Plasmodium, which were very less studied phenomena in Plasmodium. So we studied about lysine and arginine methylation in the genome. And Zishan, who is a faculty in CDRI now, also contributed to this project. And this was done in order to find out what are the various proteins which are methylated or at various positions and how they are affecting the Plasmodium biology. That was also studied. So this was published in 2017. And then on, I have been working on application of high throughput virtual screening and molecular dynamics to propose inhibitors for various validated Plasmodium targets. And recently, we are doing collaborative validation of computational predictions of the designed anti-malarial compounds, which are entering into various phases of development and optimization. And Asif and Pawan must have spoken about this in their talk. So recently, I picked up another target protein, which was very interesting because people have stopped working on this. So I decided to take up this inhibitor, which is PLDH or Plasmodium lactate dehydrogenase, which is already been identified as an essential metabolic enzyme in Plasmodium parasites. It has got a critical role in glycolysis and energy production in the parasite, which is required in anaerobic glycolysis for production of NAD+, which is required for various processes downstream. And there are several structural differences between human LDH and the parasite, which makes it attractive drug target. And someone talked about LDR. So there is one LDR which is affecting the binding site in the LDH of the parasite, which can be exploited. So this has gone into various phases of research. It was discovered in 1990s and 2000. So initial biochemical characterization of PLDH enzyme was held, and then crystal structure of the protein was solved, and development of enzyme assays were developed, and then various phases of drug discovery efforts were held, and most of them failed. And in currently, people are using or have already used various advanced computational modeling, molecular docking studies, development of allosteric inhibitor mechanisms, combination approaches with other anti-malarial targets and so on, but which have more or less failed, and people have stopped working on this target. So there is no PLDH inhibitors yet approved for any clinical use or trial. And ongoing research into novel chemical scaffolds is ongoing, and integration with multiple target drug discovery approaches is also being tried, and potential of this protein as diagnostic biomarker in addition to therapeutic target is being explored. So for this work, we explored the potential of AI in generating a diverse library of compounds which can target this protein, so that we can have diversity of compounds which can bound and which are different from the existing non-inhibitor compounds which have been tried. So this is slide just to show that AI can play a lot of role in various malaria research like vaccine development, antigen prediction, outbreak prediction, treatment response resistance monitoring, parasite biology, and diagnosis and detection and drug discovery of new compounds. So this is where rest of my talk will be. So so there is a lot of scope of machine learning as emphasized by Jeremy also in his talk. So these are partly because of traditional drug discovery have faced significant challenges, including a vast chemical space which needs to be explored. And moreover, these traditional drug discovery paths are very costly and lengthy. So machine learning offers a transformative approach which can accelerate the identification and design of novel compounds. So there are various ways machine learning can help. For example, generative compound design. So on the basis of existing chemical space, several chemical models have been developed which can predict novel entities which have not been explored yet. So efficient chemical space exploration is possible using various machine learning techniques, and you can also do data-driven insights. For example, taking stock of the compounds which are already known to inhibit plasmodium growth can be used for training various deep learning models to predict more novel entities which can be used. So in this work, we used a package called MolLib, which is a chemical trained model, which was used to generate novel molecular structures, and we evaluated their potential to be an inhibitor, but this was all in silico work. So this is how this method works. We selected 5 diverse compounds from a library of compounds that we downloaded from Campbell, and we used the transfer learning model, which has been developed in the MolLib package, and then we generated library of diverse compounds. So we generated around 3,000 compounds, and we then docked them to the target protein structure, which was already known. So this helped us in exploring the structural diversity of existing compounds. So we developed novel compounds, around 3,000 of them. We tried to computationally predict whether they could be good targets or not. And it also helped us in expanding the chemical space which can be explored for designing novel targets. So we used generative deep learning model which was there in MolLib, and it is a structure-based drug design. And we did PF-LDH specific targeting by docking each one of these 3,000 compounds. And research questions that we asked from these models, which were generated by docking all these 3,000 compounds, were whether we can generate diverse library of PF-LDH inhibitors, and will the compound show better binding? Can we identify novel chemotypes? And then we predicted these structures, and we found out that many of these compounds were indeed showing better binding than the existing inhibitors, which is one of them is oximic acid, for which the structure is available in PDB. And this is the paper which we followed. This is a MolLib paper from Gisbert Snyder's lab, and we followed their methodology. This method is available freely in their GitHub page. So essentially, this package is having a chemical language model, which is a sequential neural network, which has got 1024 and 256 units of LSTM layers. They perform batch normalization of the input compounds, and then several other parameters are given by which they made the model better in predicting diverse compounds from the input compounds. So the benefits of using transfer learning power is that it leverages chemical knowledge, which has been used in development of this chemical language model. And this we applied on 5 diverse compounds of PF-LDH, which were known to have different kinds of activities, so that we can have diversity in the output compounds. And it generates drug-like molecule because it has been trained on known inhibitors, and it explores beyond training set. So whatever is used in training set is actually used to generate diverse set of compounds which can be evaluated individually. And the benefit of using this method is that you can work with limited data. You don't need a very big set of compounds to work with. For example, in pharmacophore modeling, we need at least 200, 500 molecules which can be used for making machine learning model. But in this method, we are using already existing model which can be used for generating a novel diverse set of compounds. So this is just to show the performance of the model generation by this LSTM model. FCD scores actually tells you how diverse are the compounds from the input compounds. So you can see here that as we ran the training of the model, so each epoch is actually number of times you have trained the model with a particular set of compounds. So we used these 5 compounds to retrain in batches, and you can see here that the diversity of the compounds slightly increased, but at a certain point, say after 40 epochs of training, the difference was only stable and it was not increasing. So this is where we stopped, and we found that there were around 3000 compounds. And when we plotted UMAP of these compounds, you can see here that there were differences in source and target space after various epochs. So we generate 3000 compounds from a unique molecules of 5 seed compounds. This is another stats to show that the diversity of the compounds stopped after say 40 epochs. So these are 5 set of representative compounds after epoch 10, 20, 30, 40. So after 40, the SSE or the Shannon scaled entropy was reduced to 0.62. So that's why we stopped at this level and collected all the compounds that were generated and then evaluated them by docking it to PFLDH. So this is just to show that after evaluating sets of 3000 compounds, we came up with a best compound, which was compound 203, with a binding energy of minus 12.74 kilocals per mole. It also had best docking score versus all parent compounds. So this was better binder, and it has additional hydrogen bond to isoleucine 31. I don't know if you can see here, but it is there. And this is a novel dioxa tetracyclo group, a new scaffold which can be a potential binder to PFLDH, which was not reported earlier. And it has got a very low similarity to parent compounds. So it varied from Tanimoto coefficient of 0.31 to 0.48. And it was quite stable during 100 nanosecond MD simulation. So this was in stable conformation with the binding site of PFLDH. So we conclude that it is a novel chemotype which needs to be developed further because of various regions. So this is just to show that how stable were this compound in 100 nanosecond simulation. So these are the statistics to prove that the compound was indeed very stable. So the future perspective are that we need to modify this dioxa tetracyclo moiety in order to increase the binding and increase the selectivity as compared to the human LDH, and also improve the synthetic accessibility because we have to ensure that it can be synthesized by organic chemists. So this is where we are working right now, and experimental validation needs to be done for this compound after we modify the compound and come up with synthesizable compounds. And then in vitro PFLDH inhibition assays have to be performed to determine actual IC50 values. And of course, finally biological evaluation will have to be done on the basis of the compound that we generate further from this scaffold, and which will be done by parasite growth inhibition, cytotoxicity assessment, and resistance profiling. So these are the conclusions. We have found 3000 compounds. We came up with one final compound, and it also proves that transfer learning or the chemical language model can effectively help us in diversifying the library of a very small subset of compounds to output a highly diverse library of compounds which can be worked on further. So this preliminary work was published, and these are the main contributors of the work. Jitendra Kuldeep is actually a PhD from CDRI who joined my lab after completing his PhD here, and Neeraj also contributed this work. So my group, I wish to highlight some of my other work also, which is not related to parasitology, but recently we have developed interest in use of AI in healthcare. So we are working closely with various clinical groups from one of the major centers in New Delhi, which is All India Institute of Medical Sciences, where we have developed a deep learning model and a device which predicts presence of angle dysgenesis in ASOCT scans of glaucoma patients, which is very important for clinicians to estimate because this will help them in deciding the best treatment regimen for any patient who comes to the clinic. We can also work on histopathological slides. I don't know how relevant it to parasitology research, but in a collaboration with pathology group at AIMS, we have developed a deep learning model to predict the exact thyroid cancer type from the histopathological slides of patients. So this method is actually a method which consists of three stages because using the whole slide image as input was not possible. So in this method, we have singled out by zooming at one region of the slide, then taking the annotation from the pathologist, training the model, and then we go down further, zoom in on the regions which are important for next stage of thyroid variants, which are used for training in the next stage of the deep learning model, and then the third stage in which we have singled out on the nucleus of the cells in thyroid specimens, which are separated out, and then a deep learning model has been developed. So this is my research group at ICGEB, and is Jitender, and many of the students are working on different aspects of human health as well as plasmodium research. So this work is mainly done at the center which is known as Bioinformatics and Computational Biology Center for High Throughput Data Analysis, which is majorly supported by Department of Biotechnology. And I also acknowledge my help from my collaborators Pawan and Asif, who are sitting here. And the main purpose of my talk was to do more collaborations with parasitology groups, especially malaria, if any. So I thank for your attention.
Dr. Dinesh, wonderful talk. So, if we have one or two questions,
Arun.
Dinesh, it was a nice talk. We know that the malaria, I mean LDH, it has an additional stretch.
Yes.
I think 5 amino acids or 10 amino acids, which makes the active site very much different from the human LDH.
Exactly.
Right. Your thing that you have actually selected, does it actually bind to that place or it actually bind somewhere else?
Yes, good question. It is indeed interacting with some residues of the LCR that is there. Yeah, it is.
Have you also tried to check the docking score of the one that you showed with, I mean, human LD?
They are almost similar. That's why I said that we are going to modify this compound further
so you need
to increase the
modify it
specificity.
further. Okay.
Yeah.
Okay.
The diagnostics, two of the diagnostics assays which differentiate between Vivax and falciparum are based on LDH. And so I think once you achieve something inhibitor from that, you should try Vivax targeting also, so that you can differentiate between those kind of the drugs for the different species.
Yeah,
That
that's a
can
good
be
idea. This is where I will like to collaborate further with you.
That can be a very useful approach.
Definitely, it's a good idea, and I'll be in touch with you two guys and others if possible.
So if we don't have any more questions, thanks Dr. Dinesh for wonderful talk.
Thank you.
Thank you very much, sir, for that insightful and enrichment presentation. May I now request our esteemed chairperson to please kindly come forward and felicitate our distinguished speaker as a token of our appreciation.
Okay, our next speaker is Dr. Saurabhji Kadhirwal. He's professor at Central University of Punjab, Bhatinda, and he's an expert of artificial drug discovery and computational parasitology. Doctor.
So good evening everyone. First of all, I thank the organizer Satish Mishra and Imran for having me here. So today I would like to present some of the works that we have started a few years back. So mainly, we use various computational approaches, genomic analysis, proteomic analysis, molecular modeling, and dynamics, structure-based drug discovery, as well as in vitro screening of the compounds that we have identified using in silico techniques, as well as recombinant protein production, exploration, and initial crystallization trials that we have performed. That's what I'm going to present today. So it's my pleasure to talk our bioinformatics studies in this what is called expert biology group and get some more inputs to refine our experiments. So lymphatic filariasis, as everyone knows, is a parasitic infection that's spread by mosquitoes infected by wide variety of mosquitoes, which is one of the neglected tropical disease, which is one of the second largest cause of which causes permanent deformity as well as disability worldwide, particularly in Africa as well as in India. And this lymphatic filariasis is mainly caused by three types of nematodes. Ucheriria bancrofti, which is responsible for 90% of the cases, including India, and Burungai malai, which causes other remaining 10% of cases, especially in East as well as Southeast Asian countries. And Burungai timori is identified in isolated pockets of Timor Islands. So still it is continues over a serious concern around more than 800 million population in 44 countries according to the recent WHO statistics, and most of the lymphatic filariasis infections are concentrated in India, Indonesia, Bangladesh, Nigeria, and which causes 70% of the global burden. That is also the statistics. So to combat these conditions, WHO formed the GPLF in 2000 to eliminate lymphatic filariasis through two different strategies: mass drug administration as well as morbidity management. And in India as well, we can see it remains a severe health problem, even though various national missions we eradicated a lot, but still there are nearly so many places it occurs. Estimated of 31 million people are infected in India. Over 450 million are in risk. And this is the current statistics according to WHO. Still the status of mass drug administration is ongoing. That indicates that still that need to be completely eradicated. And here is one of the latest statistics from WHO. In 2024, the preventive chemotherapy eliminate the transmission of infection. That was required in 35 out of 72 endemic countries. At the end of, the number is 43 out of 72. That means in 2 years, at most 8 countries almost eliminated. Still, we are working for that.
and still the global burden is still mostly concentrated on the Southeast Asian countries. And as we are all know, the life cycle of lymphatic filariasis that involves with three larval stage L1, L2, L3 as well as microfilaria. So the life cycle starts from the mosquito that carries the L3, which is transmitted into the humans through blood sucking, and then adults form formation, and then microfilaria, and then again that was taken by the mosquito where the remaining cycles were, remaining larval cycles L1, L2, 3 was projected. That is a little complex process, and this is the microscopic image of adult Wuchereria bancrofti, the male one with 40 millimeters and female one with 80 to 100 millimeters. And these are the current drugs that widely used in the mass drug administration: albendazole, diethylcarbamazine, and ivermectin. So the main purpose is to reduce the density of the nematodes in the bloodstream by reducing the transmission to another healthy person which passes through the mosquito vector. So earlier it was used as a single drug, and later on several countries started with two drug regimens in combination of diethylcarbamazine as well as albendazole or ivermectin or albendazole. So that typically eliminate the microfilaria. That is one of the serious concern here, that can able to work on the microfilaria stage, which is not more effective on the adult ones. And more importantly, due to the continuous usage of this drug through various mass drug administration, the drug resistance also created to the major population. So these two issues that's made us to start work on this to identify some novel targets as well as new compounds. And this is again the latest statistic from the WHO. So interestingly, you can see that average national MDA coverage in India, particularly India, in 2023 is 82% roughly, even though there are variations in different states and districts. Even in India, 2007 we started with two drug system, and 2018 it's triple therapy. And India we use this drug elimination strategy with two approaches as WHO follows: transmission control, disability prevention and management. And in addition to that, Government of India uses enhanced strategies to remove lymphatic filariasis, and even our Government of India Ministry of Health and Family Welfare launches the national-wide mission that aim to eliminate lymphatic filariasis 2027, that is 3 years ahead the WHO goals. So in order to achieve this this particular goal, an accelerated strategy as well as more evidence-based research is required. So that's why we started working this with various bioinformatics approaches. So we started with some substantive genomics approach. So earlier, since these targets, available compounds can tackle only the adult form as well as drug resistant issues. So we try to identify some new targets. So we taken the complete proteome from Wuchereria bancrofti, which has more than 53,000 proteins, and we remove the parallax and redundant sequence, and that results nearly 18,000 entries, and then non-homologous sequences of WB bancrofti to human. That means we try to identify the protein targets that exclusively present in Wuchereria or which should have a very less sequence similarity. That should be unique to the Wuchereria. That was our idea, and we ended up with nearly 8,000 entries. And then we identify the essential proteins in this unique protein target that results 125, and then we take in for the function annotation of this 125. Some of them are hypothetical, some of them are non-annotated. So we try to start with the study with the known protein targets, and we use the KEGG interpro scan and cellular localization analysis, and based on that we identify the protein targets. And then the substratic genomics is one of the can I go back? Can I go back to this, please? Previous slide. Previous slide. No, it's going front, back. Okay. So this main interesting part in the study is we use this substrative genomics that reduce the time. Basically, the targets can be more specific to the target, and more importantly, there will be a wide variety of targets that can be selected from the genome analysis that can be prioritized. That is one of the advantage in this case. So identified 125 proteins were annotated using KEGG, and we found that 12 of them are enzyme and 28 of them non-enzymatic protein, and these 12 enzymes are in this category majority majority of the oxidative, and we mainly focus on these enzymatic proteins. Of this, we found superoxide dismutase, glutathione peroxidase, peroxiredoxin, thyrodoxine peroxidase, glutathione S transferase. All these redox enzymes are quite interesting, and we found that is more unique, and this sequence is completely different from the human host system. Yes, like other aerobic species, Wuchereria or any these nematodes that need to compare the reactive oxygen species because it has to manage both ROS. ROS produced by own nematode as well as the host ROS. So this could be one of the best strategy. That's why we started with this study. And targeting this particular enzyme may prevent the filarial worms in all stages of development because it's one of the essential antioxidant enzyme that required for the nematode. Demonstrate that more important advantage in developing the drugs against the lymphatic filariasis. So the outcomes of the studies will serve as the model for other two filarial nematodes as well. That is our idea. Since the structures of these proteins from Wuchereria bancrofti was not available, we modeled this protein using computational approaches and using the specific templates, and you can see the structures are well conserved with these different species well conserved, and we validated the model stereochemically validated using a Ramachandran plot. And we evaluate the stability of this protein target using large-scale molecular dynamics simulations. And these are some of the data that normally that we use to evaluate the performance of the molecular dynamics. The dynamics is more very nice. Its structure is more stable throughout the simulation, so that is well good enough to proceed for any analysis. That was what we can see from the studies. So we have performed molecular dynamics for all the 5 targets. Here I am going to present for one protein. We followed the same thing for other protein targets as well. So, and this is the structural superposition of structure snapshots taken every 50 nanosecond in the entire simulation. We can see the overall 3-dimensional structure is conserved during the simulation. And the binding site of these proteins are identified based on their functional importance. For example, in superoxide dismutase, the binding site is selected based on the metal binding site as well as the catalytically important arginine 143, whereas GST, we use the catalytic site, whereas in peroxiredoxin we use this N-terminal site which have C-terminal site which have an important role in PLA2 activity and peroxidation in the dimer interface, whereas GBX in some putative sites that we have used. So binding sites are identified based on their functional importance. And then we used virtual screening, large screening using Cambridge. Initially we started screening with Cambridge database. The intention is that compounds are readily available commercially we can procure. So we start with the Cambridge screening, which has nearly 1.5 million compounds. And then we use high throughput screening that utilizing extrapolation screening up to extrapolation screening that we have done, and we ended up with the top 10 hits based on the scores, docking scores. And then further we evaluated drug likeliness ADME assessment that further reduces number of identify the better compounds. Here we can able to see the compounds for each protein targets. The one in black are the compounds that passes the drug likeliness as well as the ADM qualities, whereas the red one are not qualified one. And the top 5 non-toxic leads again further taken to evaluate the stability in the protein targets, and this is the ADME profiles for the selected compounds, and this is the interaction profile of the identified compounds in the specific protein targets, and these are the 5 ligands inhibitors that identified again is superoxide dismutase. For example, here we can see the first compound is stabilized by 2 hydrogen bonding interaction with arginine 146 and histidine 63 and 1 electrostatic interaction. Mainly all these compounds are more stable. We can able to see that from the docking score. They are stabilized with the hydrogen bonding interaction, electrostatics and hydrophobic interactions. In case of superoxide dismutase, metal binding residues are more involving. Histidine 48, 63, histidine 120 and arginine 146, which is one of the important catalytic residue which forms the catalytic substrate channel to the superoxide dismutase. So all of the residues are involved. The binding, the ligands are sitting in the planned binding site in all the ligand cases, and we performed molecular dynamics for all the compounds to assess the stability of these compounds in the specific binding site, so that this particular data ensure that this compound sitting in the binding site, all the compounds sitting in the binding site as we have expected. And this is a structural superposition of various, taken from the trajectories of this 50 nanosecond simulation, and we can see the ligands are sitting in almost same position, all the data, and these are the hydrogen bonding profile. So all these interactions are maintained. So it is not the transiently bound. Throughout the simulation, the hydrogen bonding interaction with the key residues are maintained. That means the ligands are more intact with the protein binding site, and this is the dynamic stability of these ligands monitored at different time scales for the various ligands. So the ligand 1, 2, 3, 4, 5 are monitored at different time scale, which are ensured here. And further, we evaluated the what is called as affinity and specificity of the ligand in the Wuchereria bankrafti. So the model phyllidate enzymes were compared with respective human antioxidant enzymes, so that we can ensure the binding site is unique. Identified compounds are unique for the Wuchereria bankrafti. Interestingly, we can see this is the binding sites. The one shown in yellow here is the Wuchereria bankrafti binding site, whereas the one green highlighted is the human binding sites. So even though the structurally similar, the sequence variations nearly maximum we have 50% similar, 50 to 20% sequence variation similarity that we have found for the all 5 targets, and due to this sequence variations, due to the sequence variation as well as the electrostatics, binding site volume and cavity size and hydrophobicity, hydrophilic characters in the binding site, that makes the unique site for the Wuchereria bankrafti. The binding sites are, even though they are overlapping, so the ligands are more specific, whatever that you have screened are more specific to Wuchereria bankrafti. Here we can see the scores, whatever in the scores that we can see in the next drawing. So first score is 8.165 is the score that we get for Wuchereria bankrafti target. When we dock the same in the human target, the binding score is minus 3.78. Whereas similarly, you can see the drastic difference in the binding scores between the Wuchereria as well as human targets. So that emphasizes that whatever that we have identified might be more specific to the Wuchereria target, and there won't be any cross reactivity. That you can see all the scores are having very huge difference between the human and Wuchereria targets. And then we taken these compounds for the in vitro validation using CSIR digitata. This is one of the most genomically similar species with respect to Wuchereria bankrafti, that was studied extensively as an in vitro model to compare the compounds for Wuchereria bankrafti. So we collected the CSIR digitata from the peritoneal cavity of the cattle. So this compound, this CSIR digitata was further validated. Actually, we are not expertise, so validated, authenticated by the veterinary parasitology department, veterinary college in Orthanadu, Tanjavur. So where I was working earlier. So once we identify this, once we got the some worms isolated from the CSIR digitata, and we perform the standard MTT assay to test the worm viability and microfilarial cellular activity to identify the compounds which are tested using the MTT formation colorimetric assay. And this inhibition was calculated by comparing the treated worm viability to that of the untreated controls and the heat killed worms. And interestingly, we can see at the maximum dose of 20 micromolar, all the best top 3 compounds that we have identified from the in silico screening have demonstrated a better efficacy. Demonstrated the efficacy more than 84%, and more importantly, this MTT detection assay shows that the identified lead have higher inhibition better than the standard ivermectin compound, and the compounds which have better IC50 are highlighted in green, underlined the green, and red one are not compared to ivermectin standard. And nearly out of 15, we selected 3 for each target, so 15 compounds were tested. Out of that 15, 10 of them having a better IC50 compared to the standard ivermectin. And more interestingly, we identified some pharmacophore future. The compounds have different functional groups, different, and we identified some common pattern in the groups, in the compounds which have better IC50, as well as, for example, here the compounds which have better IC50 have the pharmacophore pattern of hydrogen bond acceptor, which is denoted as A3A1, and then hydrophobic as well as one aromatic moiety. 8 compounds have this pattern, whereas acceptor, aromatic moiety also have a better IC50 value, whereas the compounds which have only one acceptor, ADHR, AAH, RR compounds are showing with a lesser IC50 value. That was a common pattern that we can note from here. In addition to that, we observed some geometric features as well in these pharmacophore models. For example, here you can see the acceptor hydrophobic distance A1H1 is 3.23, whereas this compound showing better IC50 have the distance of 3.24, whereas the compound showing the poor IC50 value, the distances are very higher, 8.3 and 9.1 that we can see here. And then interestingly, you can see the angle, the aromatic acceptor and hydrophobic angle is acute, 57, 36, whatever that you can see here, even more acute, here 6, that 15 and 31. So these are all more distinct distinct pharmacophore feature, geometrical feature that we identified from the compounds. And then we try to see the structural mechanism of inhibition. So we recombinantly produce this protein using E. coli expression system. We produce this protein and purified, and we got the native crystals which are on the microcrystalline level, and we already tested some of the crystal, but still the crystallization trials are underway. And these are native crystal, and these are some co-crystals with some of the compounds we already started and identify some microcrystalline states, and we are trying to get the structures of protein ligand complexes to understand the mechanism. In addition to that, we are trying to do a large-scale genomic study to annotate the hypothetical protein. Majority of the proteins are still unknown function. We try to identify those proteins and prioritize the targets, and then we proceed the same with the same pipeline to identify the better targets as well as the lead molecules. So that is our idea. So this particular study mainly emphasizes the use of various computational approaches. Mainly substratic genomics can be one of the best way to identify the target, because due to the existing developing drug resisting issues, as well as to combat the to target the adult worms as well. So this is one of the thing that I want to propose. Some of the papers, that computational papers that we have published based on this work. And I would like to acknowledge ACRB ANRF for the funding, and ICMR, and Central University of Punjab, where I am presently working, and Shastra University, where this work was earlier started, and my research collaborator Mrigang Srivastava and Professor Sridharan Idayatullah. So mainly Mrigang we are working together to study the in vivo validation, and Sridharan try to develop some compounds, synthesize some compound to test with better efficacy, and Idayatullah helping in biophysical characterization. Thank you.
Yeah, lot of work, Dr. Saurabh.
Thank you.
Any question, please?
Yes.
Yeah, there's one.
Yes.
The inhibition on these proteins?
Yes.
Right.
The compound, yes.
And what was the IC50 like?
No, IC50 we have not checked.
So that's what I'm saying.
You've done the inhibition studies on the parasite. Not Bucararia or Brugia, but the other parasite.
Not in the CSIR we have
that CSIR-CDRI. And but you have the enzymes now, right?
Yes.
So you have purified them. So
yes.
Maybe we should also check the fact whether this actually inhibits the enzymes and their activity on the enzyme.
Yeah.
Any other question? So I have a small question or comment. So because now you have a lot of data of docking as well as pharmacophore patterns, you have structures also. So do you plan to derivatize these compounds further and more efficient compound?
Yes, that's why we identify some pharmacophore and synthesize based on this identification.
Oh, great. Sounds good. Okay, if there's no other question, then let us thank Dr. Saurabh.
Thank you very much, sir, for that insightful and enriching presentation. May I now request our esteemed chairperson, Dr. Muhammad, to kindly come forward and felicitate our distinguished guest. Hello everyone. Before moving to the next session, I have a quick announcement right now. Mr. Chandrasekhar, if you are here, please contact to the backstage immediately, because your ATM card has been found. So as soon as possible, you contact to the backstage. Thank you.
Okay. Hello. So our next speaker is Dr. Abhik Sen. Abhik Sen is working as scientist E at ICMR RMI Patna, and I think he is going to talk about the dermal fibroblast regulates immunity and influence the Leishmania progression. Thank you.
Good afternoon. It's an honor to be presenting in the National Parasitology Congress, and I thanks the organizer committee for inviting me to present this talk. So we had been listening to a lot of drug discoveries. Now I am back to some basic biology. So I will be talking about the immunoregulatory role of dermal fibroblast in visceral leishmaniasis and post-cholera dermal leishmaniasis. So we know that visceral leishmaniasis is caused by Leishmania donovani and is transmitted by the sandfly called Phlebotomus argentifus. So we have a sequel for the visceral leishmaniasis called post-cholera dermal leishmaniasis, and this happens after treatment. Most cases are occurring after cure of the VL, and mostly after like 1 to 3 years after. 10% of the VL cases mostly they get converted to PKDL, and they are manifested at hypopigmented erythematous or nodular lesions in the skin. The PKDL complicates the management of the disease, but also they serve as reservoirs. So identification of the PKDLs as well as understanding the mechanism why PKDL is happening is an important thing to know. So PKDL is characterized by dysregulated cytokine profiles and T cell activation. So next, what we thought that probably since skin is the primary route of entry for the parasite, and yet fibroblast specific mechanisms were not that much known for VL or PKDL, and these fibroblasts are key regulators for tissue repair, immune modulation, cytokine chemokine secretion, parasite encapsulation, and stromal signaling. So probably this fibroblast may be actively shaping the microenvironments which determine whether a patient gets a VL or it becomes a PKDL. So therefore, we started working on the fibroblast and related transcriptomics. So what we did, we collected punch biopsies from healthy individuals, visceral leishmaniasis patients, and PKDL patients, like a 4 mm punch biopsies, and then we cut them into small pieces and tried to grow fibroblasts from this tissue implants. So it takes almost a week or two for these implants to release the fibroblast, and they get homogeneous after like first or the second passage, and that was characterized using vimentin, which is a marker for identifying the homogeneity of the fibroblasts. Next, what we did from three individual patients from each group, we did a whole transcriptomic analysis, and so as you can see in the PCA plot, it showed a disease-specific stratification. This made us more like interested in working further with this transcriptomic assays. What we found is that if we compare the VL versus PKDL fibroblasts, 263 genes were upregulated and around 253 were downregulated in PKDL. And when they were subjected to a geo-ontology analysis, the top 10 hits were mostly either TNF signaling pathway, IL-17 pathway, or IFN gamma pathway, NF kappa B pathway, or the chemokine receptor chemokine interaction pathways. So what we next did from this whole geo-ontology analysis, we pulled out the immunologically relevant pathways and the genes involved in them, and then we did a heat map. And as you can see here, that here also the clustering is the three healthies are here, then comes the PKDL, and the VL are distant from the two groups. So in VL, what we found that the whole chemokine, the chemokine CXL1, 2, 3, 6, 8, 10, 11, they were overexpressed compared to the PKDL. The cytokines IL-1 beta, IL-6, they were elevated in the VL compared to the PKDL. And probably this gives the indication that in the VL, this immune environment is helping to enhance the monocyte, the natural killer cell, and the neutrophil recruitment to the site of the infection when the parasite gets into the skin. Well, in PKDL, what we found is that RAP7A and HLA-E, they were upregulated, while downregulated were HLA, DP1A, PSMD, and NF-kappa B. Now, RAP7A normally delays phagosome maturation. So, probably it is helping the parasite survival in the skin. And the downregulation of NF-kappa B is probably causing an environment that is immune suppressive. So, in the VL, it is indicating towards that probably it is inducing a recruitment of the immune cells, visualize the disease, whereas in PKDL, it is not recruiting that well and it is helping the parasite survive in the skin. So from the immune genes, we did a protein-protein interaction and then the proteins interacted. It is around 12 proteins that interacted for a comparison between PKDL and VL. The downregulated network proteins were MMP2, IL-1 beta, CXCL8 and IL-6, where the upregulated one were actin, HSP90 and RAP7A. And now actin being upregulated, probably it's trying to heal the wound in PKDL. So probably that is the reason why actin is upregulated in PKDL fibroblast. So this data indicates that contrasting immune pathway activation patterns. So this was from the transcriptomic data. So we tried to now validate this data with a real-time PCR experiments from the PPI network genes. So as we found in the PPI network genes, real-time PCR data also validated the same, showing that intracellular actin expression was high in PKDL, then it's VL, and finally this lowest in healthy. Intracellular IL-1 beta, IL-6, CXCL8, CAPDH expression were high in VL, more than PKDL, and finally the lowest one is in healthy. Extracellular cytokines were measured by ELISA and it gave the similar result as what we saw in the transcriptomic data or the qPCR data. So we proposed a hypothesis from this that probably in VL, hyperimmune environment is created whereby recruiting the immune cells to the site of the infection and visualizing the disease. While in PKDL, this function is not that much active and helping the parasite survive in the skin. So this first part we have published in GID in 2023. So this was a summary. Fibroblast shows a disease-specific immune signature in VL PKDL. PKDL favors a class 1 MSC response, while VL favors a NF-kappa B mediated chemokine response. Differences persist. Now this was most interesting, that even after the third passage or the fourth passage, these characteristics of fibroblast were staying down. So probably there is an imprint or either they are primed or they are having an intrinsic property. So after this, what I hypothesize is, well, the fibroblasts are behaving differently. Whether they are differentially regulating the macrophage signaling or not. So the next experiments were planned on that basis. So we set up a transwell co-culture system where we have seeded the fibroblast in the upper chamber and the THP1 cells in the lower chamber. And one set we induced them with Leishmania donovali. And then we measured the cytokines and chemokines from the fibroblast separately and the THP separately using a real-time PCR, while the soup from the combined co-culture system was measured using ELISA. So what we found is that these are the data from the fibroblast of the co-culture system. Whatever we saw initially in the transcriptomic data, we were seeing the same thing even in the co-culture system. But interesting part was that when we were inducing it with the Leishmania donovali, in VL, those expression was going down, and in some cases the PKDL, it is going up. So that was a bit confusing, but it may have an explanation that since in VL they are already, the system is primed, so they are, when we are giving them again Leishmania, they are trying to subdue that inflammation to help the parasite survive. Next, we looked into these expressions in the THPs. In the THP, what we found again that IL-1 beta, IL-6, TNF alpha, IL-12 and interferon gamma and IL-17A, they were high in VL fibroblast induced THP cells compared to the PKDL fibroblast induced THP cells. Post-infection, so post-infection with Leishmania donovani, IL-1 beta, IL-6, TNF alpha, IL-12 and IFN gamma, they go down in the VL fibroblast induced THP cells. While post-infection IL-1 beta, TNF alpha and IL-12, they go up in PKDL fibroblast induced THP cells. Now, these are for the anti-inflammatory cytokines, IL-10, TGF-beta and IL-13. They were found higher in VL fibroblast induced THP cells and compared to the PKDL fibroblast induced THP cells. And post-infection, IL-10 and IL-13, they go down in VL fibroblast induced THP cells, while post-infection IL-10 and TGF-beta, so IL-10 goes up while TGF-beta goes down in PKDL fibroblast induced THP cells. So this is for the chemokines. Excuse me. CXCL 1, 2, 3, 5, 6, 7, 5, 6, 8, 10 and 12. They were found overexpressed in VL fibroblast induced THP cells compared to the PKDL fibroblast induced THP cells. Post-infection, most of these chemokines, they got down in VL fibroblast induced THP cells except for CXCL11. But post-infection in this case, in PKDL, CXCL 1, 2, 3, 5, 8, 10 and 11, they went up. Only similarity is that CXCL 11, in both the cases post-infection, they are going up. These data were again validated for the secretory cytokines from the soup of the co-culture, and they gave almost similar results except for I think it is IL-8. Except for IL-8, sorry, yeah, except for TNF alpha, they gave similar result with the transcriptomic as well as the initial fibroblast data. So the next idea was why this is happening. So to understand why this is happening, we have not done a co-culture for this. So we grow, culture the fibroblast in a cell-free system, and then use that media to induce THP cells to see whether the soup of the conditioned media from the fibroblast, they can regulate the nuclear translocation of NF-kappa B or not. So we have seen that when we have used soup from the VL fibroblasts, they induce the nuclear translocation as a percentage of in VL in this fibroblast. Compared to the PKDL, they were down. But the P50 subunit, they did not show that much significant difference between the VL and PKDL. But RAIL A or P65, they showed a significant difference. Now, whether, if it is, what is the factor? Since IL-6 has been coming on consistently from the fibroblast till the co-culture system. So, what we did, we blocked the IL-6 or incubated the conditioned media from the fibroblast with anti-IL-6 antibody from the VL fibroblast. And when we used that one, there was a decrease in translocation of the nuclear P65. And in the PKDL system, we added recombinant IL-6 to the PKDL fibroblast soup. And then when we use that for inducing the THP cells, there is an increase compared to the only PKDL soup. So probably IL-6 from the fibroblast, they are playing this role or inducing the nuclear translocation. And this nuclear translocation of NF-kappa B is probably inducing the chemokines and the cytokines, producing a pro-inflammatory environment in case of VL, thus inducing or recruiting the cells to the site of the infection and visualizing the disease. So if we try to conclude what we found, that from the initial RNA-seq data, it's around
total 516 differential expressed genes were found in between VL and PKDL comparison in fibroblast. In PKDL, NF-kappa B, MMP2, IL-6, IL-1 beta, and CXCL8 were downregulated. Then VL fibroblasts, they promote a strong pro-inflammatory milieu. The co-culture with THP1 macrophage induced IL-6-1, IL-1 beta, IL-8, interferon gamma, and the chemokine setup, and a robust NF-kappa B activation that enhanced immune cell recruitment. While PKDL fibroblasts, they induced a balanced immune response, and it represents a controlled inflammatory regulatory state, allowing parasite persistence in a minimal tissue damage. And then Leishmania donovani infection that drives the immune exhaustion. Infection suppressed cytokine and chemokine production in already primed cells, indicating immunological fatigue in the fibroblast and macrophage interaction. And mechanistically, we can say that the IL-6 plays an important player, if not, I can't say it's the main player, but it is an important player in this NF-kappa B axis, whereas in case of VL, it induces the translocation of NF-kappa B in the immune cells, thereby producing a more inflammatory environment. If we neutralize IL-6, this NF-kappa B activation is going down, confirming the pathway. So overall, we can say that fibroblasts, they play a key role in the skin, modulating the immunity in VL and PKDL, and probably targeting this pathway for PKDL can help us preventing or like curing the disease faster. So I thank the funding agencies, ICMR and our institute for funding this research. Thank you.
Thank you for saving our 3 minutes. And now if we have very quick questions.
Good evening, sir.
Good evening.
Sir, was this fibroblast when co-cultured with the THP1 cells, they were somehow hindering any effect exerted by the parasite or not?
So, hindering the effect of the parasite or
Yes,
not?
sir.
So, we have just shown the effect of the parasites on the fibroblast. We are keeping the fibroblast, giving the parasite in the fibroblast chamber, not in the THP chamber. Just to see whether there is the parasite fibroblast interaction is changing causing a difference in the immune setup or not.
And also they were getting some connection with the resistance of parasite?
Well, we have not checked in the resistance of parasite. We have just focused on the fibroblast, macrophage or THP cell interaction.
Yes, sir, because we have reported that the resistance of parasite has been related with the host system. So that's why I asked this question.
Yeah, I mean, resistance of you won't say it as a resistance of the parasite, but probably getting relapse of the disease is probably related to the host factor. But resistance of the parasite, we won't be saying it as resistance of the parasite.
Thank you, sir.
Yeah, and
so Avik, nice presentation. So would you like to hypothesize that there are people, they are kind of predictable on the basis of how the fibroblasts behave, whether they will develop PKDL or not?
Sir, it is possible. So that is what our next project, what we got from ICMR is to see the exome mutations as well as the epigenetic regulation of this fibroblast as well as the blood system, whether they are predisposed or not. Probably the first time they're getting VL, they're tolerating it, but when they're getting the second time, either it's a epigenetic change or it's a predisposition already.
It's already there and probably escaping and going to the skin.
That is also a case. Probably the parasite is surviving somehow, and whenever that time period is coming, it is escaping and going up.
So, but you are getting normal fibroblasts from normal individuals?
No, sir. We are getting from healthy. We are getting fibroblasts from VL.
You have a large repertoire of healthy fibroblasts from healthy people.
Yeah.
And theoretically, you would expect that at least one or two will behave differently.
That is true. And that also with age, they will different behave.
Yeah, probably that will give you some indication about the predisposition.
Okay.
So you have to increase the number of
Right.
normal individuals.
Like for the relapse, we have found and we have reported also that they are like the EP300 gene. It is mutated in whatever, like 14 relapse cases we have done, 16 cured cases. So 14 out of 14 was having that mutation in the same place.
In the fibroblasts.
No, that's in the normal blood, I mean PBMC.
In which protein is that?
Is EP300.
And what's the function?
It is a histone acetyl transferase.
Okay, okay. It's epigenetic modifier.
Yeah.
Okay. Thank you.
Any more question? Okay, so if we don't have any more question, I will thank Dr. Sen.
Thank you.
Yeah.
Thank you very much, sir, for that insightful and enriching presentation. May I now request Professor Trivedi to kindly come forward and felicitate our distinguished speaker as a token of our appreciation.
Okay, our next speaker is a young warrior in the malaria field, Dr. Ankit Gupta, and he will talk about some of his work on metal ion transporters in Plasmodium.
फॉरवर्ड बैक लेजर फॉरवर्ड बैक लेजर लेजर पुश करेंगे इसको पकड़.
So first of all, I would like to thank the organizer for inviting me for the talk. Despite being a clinical biochemist, I would present a small a part of work which has been done on the same belong to basic biochemistry. So basically, this work is based on the proteins involved in magnesium transport. Those are being involved in the asexual phase of the cycle, asexual cycle of the malaria parasite. So these proteins, transporter proteins, are involved in the magnesium transport. So there are the numerous evidences that support that the magnesium is involved in the parasite development and malaria infection. Such as the serum magnesium levels were found to be elevated threefold during the uncomplicated acute falciparum malaria infections. A meta-analysis reported a significant elevation in magnesium levels in patients with fatal malaria compared with the survivors. There is also one report that suggests the magnesium deficient diet protected mice against the Plasmodium tuberculosum malaria infections. And there are also numerous studies has been done that shows the involvement of the magnesium in the parasite invasion and the zygogony. So there are the various magnesium transporter are reported in the literature. So one is the core A, which is in the mostly found in the bacteria. There are some other magnesium transporter A, B, C, and E. There is a PHOQ is the membrane receptor for magnesium, and it regulates the expression of various magnesium transporter A, B, and C. Magnesium A and B are usually unusual members of the P-type ATPase family, and there is also one magnesium transporter is reported in the mitochondria, which is known as a MRS2, which is a mitochondrial RNA splicing protein 2. So in the Plasmodium falciparum, we got 3 homologs of the metal ion transporter, which belong to a core family, and all these transporters homologs, they have a consensus GMN motif, which is a unique property of the magnesium transporters. Along with that, there is also a lysine-rich C-terminal chain. So notably, we found that PFMIT1 has the among other homologs, and the bacterial counterpart has the large C-terminus, suggesting that it probably playing some distinct role. Along with this, the PFMIT1, 2, and 3, these three homologs shares a very low similarity and identity as compared to the bacterial counterpart, which is Thermotoga meritoma. And this MIT proteins do have a C terminus, two transmembrane domains in the C terminus, and basically they are connected with a smart stretch, and this stretch basically consists of GMN motif, which acts as a ion selective ions, which selects peculiarly the magnesium ions to enter into the cell. And additionally, these proteins or the magnesium ion transporter proteins, they forms a homopentamer like assembly and are involved by making a homopentamer. They are involved in the magnesium transport. So first our objective was where these proteins are basically going in the parasite and what's their role basically. So there are genome-wide gene knockout screen done in P. Bergii reported that MIT1, 2, and 3, these homologs are dispensable for the parasites, and while deleting them, they have no changes in the asexual stages as well as in the gametogenesis. However, only the MIT2, while deleting, they found some kind of growth arrest during the sexual stage and the transmission. So here we did performed a kind of phylogenetic analysis, and we found that PFMIT2 and 3 are very closely located along with the green alga, suggesting a apicoplast origin. But MIT1 was quite away from these two homologs. Additionally, we performed some bioinformatic analysis, and we found that PFMIT1 has a apicoplast targeting peptide, although all these three proteins, they lacks the signal peptide. So only PFMIT1 is supposed to it have the apicoplast targeting peptide, suggesting that it probably targeting to apicoplast, and it also have a high mitoPro2 score, suggesting that it could be targeted to mitochondria. So here we this is the pentameric assembly which we modeled on the TM core A. So the unmodeled structures look like loops like a structure. We express this protein recombinantly with a his tag, and we did a posterior probability plot assay, which we found that presence of the two transmembrane domain towards the C terminus. In order to localize this protein, I did a transfection to overexpress this protein under the control of PFCRT promoter with a C terminus GFP tag, and we found, in contrast to wild type, this protein was successfully expressed with GFP tag at a size of 90.5 kDa. So here we found that this protein is localized to both apicoplast and mitochondria. So here we find a overlapping signal of the GFP with the NTHU, suggesting this protein is targeting to apicoplast. And additionally, we have found that the GFP signal of the protein is overlapping with the mitotracker red signal, suggesting this protein is also targeting to the mitochondria. The second homolog, MIT2, which we also expressed as a recombinant protein, and found the presence of the two C-terminus transmembrane helices using the posterior probability plot analysis. And we also made a transient transfection to tag this protein with the GFP, and we express it, and we found that the signal GFP signal is co-localizes with the NTHU signal, suggesting this protein is going to apicoplast. But we didn't find any overlapping of this signal, GFP signal, with the MR, means that protein is not going to mitochondria. So the third analog, which is the MIT3, we expressed is also as a recombinant protein. Since these proteins are the membrane proteins, so we need to solubilize with the very less percentage of a mild detergent. And we found the presence of the C-terminus transmembrane helices in this protein. So here we made a transfection. We overexpressed this protein with the GFP tag, and we found this protein is expressed with the GFP tag at the size of Day-84 kDa. We did a membrane solubilizing assay in order to see that where this protein is basically going, and we found that this protein is prominently present in the carbonate soluble fraction, and very less proportion is present in the carbonate insoluble fraction. So means that most of the protein is present in loosely associated with the membrane, and very less protein is integral to the membrane. So during the IFA analysis, we found that the anti-GFP signal of this protein is not not co-localizing with the MR signal, means this protein is not going to mitochondria. And well, this protein, we found that the signal is overlapping with the GFP signal is overlapping with the anti-HU signal, suggesting this protein is also targeting to the apicoplast. So we also performed a kind of complementation analysis in a yeast system where there is a mutant analog, the mutant yeast that lacks the magnesium transporter proteins. And we found that these two proteins, MIT2 and MIT3, even in the in the high magnesium concentration, they are somehow able to complement, but in the lower magnesium concentration, they are not being complement, suggesting that probably they alone, if we express in the yeast, they are somehow is not able to form a kind of magnesium transporter and not able to rescue the yeast strain in magnesium deficient conditions, probably. We also run the native page in order to see whether these proteins are forming the higher order structure or not. We found that MIT1 and MIT2, they form a higher order structure up to the pentameric structure, the both MIT1 and MIT3, but we do not find any kind of oligomer in the MIT2 proteins, as the protein these proteins are supposed to form a kind of homopentameric assembly. So based on the findings and their localization, we hypothesized that basically the MIT proteins are encoded by the nuclear genome, and they are targeted to the apicoplast and the mitochondria. So only MIT2 and 3 are targeted peculiar to apicoplast, whereas MIT1 is targeting, dual targeting is happening in both mitochondria and the apicoplast. So one model says whether these three transporter are individually expressing like a homopentamer in the apicoplast, or other model could be that probably they are forming a heterooligomer assembly. So means and that's our hypothesis, that if we delete a gene, other gene is overexpressed and compensate for the loss of one gene. However, we are going to test it. So there is a few inhibitors are known, which are known to be a selective inhibitors of the core magnesium transporter system in the bacteria. And there are the cobalt 3-hexamine, ruthenium 2-hexamine, and 3-hexamine. They have been shown very less IC50 in the bacterial counterparts, and we choose these three inhibitors to test that whether they are going to inhibit plasmodium system or not. So based on the bioinformatic analysis, we docked them, and we found docking scores, and the docking score for the specifically for the cobalt 3-hexamines, that binds more strongly to the PFMIT3 based on the docking basically. So we test these inhibitors on the parasite culture, and we find that they exhibited the parasitical activity. However, their IC50 is quite high, and one explanation is that since these parasites are growing in the RPMI media, which has a high concentration of the magnesium, so and generally these inhibitors, the potential activity like a physiologic conditions where they have a very limited concentration of the magnesium. In order to see whether these inhibitors are specifically targeting to our protein or not, we just choose a one inhibitor and we try to raise the drug resistance parasite. We grows the parasite in the presence of this in higher concentration of the inhibitor where all the parasites are die, and we find a 6 to 7 fold increase in the IC50 in the drug resistance clonal parasites. So this is the cloned parasite. Once the parasite died and then they appear, we just clone it and we find a 6 to 7 fold higher IC50 as compared to the wild type. Then we look for what are the changes in our protein of our interest, if whether these proteins are somehow interacting with this inhibitor or not. So this is a very preliminary result. So we found that both of the clones have an extra A basically in contrast to the wild type. However, we need to further validate, and this kind of indel, which is a kind of insertion, is present basically in the transmembrane domain 2, which causes a frame shift mutation. So the further C terminus is somehow prior terminated and the truncated proteins or non-functional proteins are being formed basically. However, we are trying to look for the other MIT1 and 2, whether these kind of mutation or the indels are present in them or not. So based on the results, we concluded that GMN motif is conserved among all MIT homologs of malaria parasite and acts as an ion selective filter which allows the entry of the hydrated magnesium. MIT1 is found to be dual targeted to both apicoplast and mitochondria, where MIT2 and MIT3 are primarily apicoplast targeting peptide proteins. MIT3 is the membrane protein that is mainly found in carbonate soluble fraction, means it is loosely associated with the membrane. And MIT1 and MIT3 proteins, they are found to be from oligomers based on the native page, and cation hexamine exhibited parasitidal activity, and cobalt 3 hexamine resistance parasite exhibited 6 times higher IC50 values compared to the sensitive parasite and supposed to carry a frame shift mutation in the transmembrane 2 helix of the PFMIT3. So this is the conclusion. So here I would like to acknowledge my small research group composed of 3 students at AIMS, and I would like to acknowledge my mentor Dr. Saman Habib for helping me especially for the confocal microscopy. So, and I also acknowledge all the funding from various agencies which I got from NRF, CST, ICMR and DHR. So, and eventually I would like to acknowledge the AIMS where all the study was done. Thank you very much.
Questions, please. Yeah, Saman.
Soluble fraction and not in the insoluble fraction?
Yeah, we found a major portion in the carbonate soluble. However, we found a little amount in the carbonate insoluble. So probably, I don't know, means somehow it is also loosely associated, and we need to see how it is playing a role. So one thing which I am supposed to do in near future, that we just pull down through anti-GFP and just probe it with the other antibody against the other homologs MIT2 and MIT3 to see whether they are forming the heterooligomers or not, in order to prove our hypothesis basically, which we are supposed to do in near future. Sorry, what? That we were supposed to do by expressing in the yeast deficient strain, but unfortunately it could not complement it. So we are trying to express it in insect cells, and just to see whether how it is involved in the magnesium transport and how in the presence of inhibitors that transport is somehow inhibiting or not, in order to see the direct relationship of these proteins with the magnesium transport and the inhibitors.
Yeah, Ankit. So this dual localization is a stage dependent or throughout the asexual life?
I just choose few pictures, but I found throughout the various stages. Basically, we pick trough and Shijon. Basically, so we found a dual localization in both
In all
kind of stages.
And then this heterocomplex, did you try in vitro? That do they form heterocomplex?
So that we need to do that. We need to pull down from using
Yeah,
a
you
one.
have recombinant protein, so you can do it.
Yeah, and use the other antibody to see whether it is coming in the pool or not. That
That
we need to do that.
Okay.
Thank you.
If no other question, we'll thank Ankit.
Thanks.
Thank you very much, sir, for that insightful and enriching presentation. May I now request Dr. Mohammed to kindly come forward and felicitate our distinguished speaker as a token of our appreciation. We extend our gratitude to the chairperson to their valuable time and support in conducting this session. Now we would like to invite Dr. Jeremy Burrows to felicitate our esteemed chairpersons. Thank you, everyone. We will now take a short tea break and join us for the refreshment at the CDRI lawn. The next two sessions will be run parallelly at 5. Session 10, Oration Award, will be conducted in the Hall 2, and the Session 11, Techno-Commercial Presentation, will be conducted in the Hall 3. Kindly be back in the time so we can start promptly. Thank you.